Hi, I'm Casey Woolabin. Good morning, good afternoon, good evening. We are so happy you are here at our Empower and Inspire Understanding and Accelerating Research for Leigh Syndrome virtual conference. We're all here because we have one thing in common, and that's Leigh Syndrome. Whether you're a parent or family member of an affected individual or affected yourself, or a researcher, doctor, or industry, we all have one goal, and that is to find a treatment or a cure for Leigh syndrome. So on behalf of Cure Mito Foundation and iCAMP, we are so happy you are here to learn more about Leigh syndrome. Let me share my screen, and I'll give you a brief introduction of Cure Mito. So Cure Mito Foundation, it was started when my son was diagnosed with Leigh syndrome. It was back in 2014. Let me go to the next slide. So Will was born December 21st, 2011. He was a perfect baby. We had no idea what was about to happen in our lives, just like many of you on this call. My heart was full. I was overwhelmed. I was a first-time mom. My life was perfect. His first year went by so quickly, and he hit all of his milestones, growing perfectly. And all of a sudden, after his second birthday, we noticed something, and that was falling episodes. So he would walk and just stumble, and we thought, wow, this is strange. What could this be? So we took him to an array of specialists, and tests were done, and they all said, no, he's fine, he's growing, he's perfect. You know, you're just a first-time mom, and don't worry, his growth will catch up. So in 2014, I gave birth to my daughter, Lauren, and became a mom for the second time. I felt like I had won the lottery. Like I had the perfect life. I had a baby boy, I had a baby girl. Little did I know that 6 weeks after she was born, he would be diagnosed with a rare disease called Leigh syndrome. So that day started as a normal day, and his stumbling just kept getting worse. And so we took him to the emergency room and asked for a CT scan or MRI, anything that could help us understand what was going on. And I knew I had that mother's tuition, like something's going on, something's not right. And we were told that he had Leigh syndrome. And our world was turned upside down, just like many of you that are on the phone right now. We grieved for what our son's life would be, and we also found strength in reaching out to others. My husband researched Leigh syndrome every day. He would email doctors and scientists to ask if they were studying Leigh syndrome, especially when we found out it was the SIRF1 mutation. Then we started identifying researchers and doctors that were working on that specific mutation. Doug would often tell me of gene therapy for monogenetic diseases, and that kind of gave us some hope that there was maybe a possibility that this could be treated, and there would be hope, right? So we were told the same. We were always told the same about gene therapy. It's going to take years and years and years. Don't waste your time. Just enjoy whatever time you have left with your son and move on, right? But for parents, that's not going to happen. So once we heard that gene therapy could be an option, and we knew we couldn't do this on our own, we connected with other SIRF1 families to start the daunting task of raising millions of dollars needed to fund necessary preclinical research. And that is when Cure SIRF1 was formed, because of these three families right here and a doctor, Dr. Stephen Gray. He is, um, oh gosh, one second. I apologize. We are having construction and the workers seem to be here right now. So if you do hear work, I apologize. Back to where we were. We are Parents on a Mission, and we worked with Dr. Stephen Gray out at UT Southwestern to build a SURF1 gene therapy program. And we were told that it would be a long road and it may not work, and it's expensive, and we had to do this to save our children. So the three families right here, so we've got Emma and Kashiti and Will, so we all came together and formed SURF1 to get to our goal of raising millions of dollars for preclinical research. And all grassroots efforts were just parents on a mission. And once we did that, once we did pave the way for SURF1, we realized it was bigger, much, much bigger. So SURF1 is the most common mutation in Leigh syndrome, but there are over 100 different mutations that cause this devastating disease. So we wanted to share our knowledge, that we have learned along the way to help other families, want to empower them, want them to come and research their child's mutation to find a treatment or a cure. So that's when we changed the name and we said we're going to encompass all of the mutations, and we're gonna do that by creating Cure Mito Foundation. And it was created, through SURF1 with the efforts of family. We are grassroots efforts. We are here to pave the way and to knock on those researchers' doors and get Leigh syndrome noticed. We want everyone to know what Leigh syndrome is, so we can find a treatment or a cure for our children. And it takes a village. It takes a huge, huge village. And we are so, so extremely blessed to have a village of our own. We have families from over 13 countries who have been with us from the beginning, sharing their knowledge, trying to find different ways to do fundraising events, to get the money needed to raise awareness and funds for research. And without these families right here, none of this would have happened. This is truly a family, a foundation. All of these families are so very important, and we could have not have done this without them. So we are so grateful for our families, and hopefully one day we will have a cure or treatment for our children. I'm proud to say that since starting Cure Mito, we have the largest Leigh syndrome global registry with over 200 patients enrolled in over 30 countries. And if you haven't enrolled, please enroll. It is so important that researchers understand what Leigh syndrome is, so we can get to that cure or treatment. We need to find out the natural history of this disease. We need to know the symptoms. We have to understand all of this to get to a cure. So it's vital that everybody participate in the global registry. We also are working with Perlara and Rarebase and drug repurposing. So we are researching drugs that are already FDA approved that we could potentially give to our children to help slow the progression of the disease while we wait for something bigger like gene therapy. We're still funding SURF1 research. We just announced that we, along with the Cure ATP6 families, are funding a 2-year, 400,000 ATP6 research project, and we are so excited. We were just at Mito Nice this past week, and just hearing the researchers talk about all this groundbreaking technology, it's exciting. And for once, we have hope that a treatment may be on the horizon. And these are just a few of our research collaborators. Without them, we wouldn't be where we are today. So we just wanted to say thanks. Thank you for allowing us to work with you. And if you are interested in working with us, please reach out and let's see what we can do to collaborate and work together to find a cure. Now, we also have, since we are a family foundation, we also have our birthday club. Our birthday club is for our Leigh Syndrome warriors, just to give them a little something special to open up on their special day. As we all know, every birthday is a huge celebration in our children's lives. So if you haven't joined, please join. They get a special surprise, and we are so just thankful for our Leigh Syndrome warriors, our heroes. They are why we are here today, each and every one of them. We also have a newly diagnosed resource guide on our website, and we also have a resource section with clinical trial updates, support groups, recommended books, and also a kids page that kind of helps explain what mitochondrial disease is for little kids. So check that out. It's awesome. I do want to end on a high note. So even though this is a devastating disease and it's terrible and we're in a nightmare, I have met the most incredible people in the world. And families, all these children that I've met throughout the years, they're like my children, and I will fight for them, and we will continue to fight. So I just want to share this little story about little Delta. She was diagnosed 1 year ago, and her mom reached out to me and said that she didn't have a wheelchair. She was wanting to be mobile and independent. And Will was given this wheelchair that you see Delta sitting in as a hand-me-down when it was his first wheelchair. So we saved it knowing that someone will need this chair. And Delta, she needed this chair. So Will and I drove to Delta's house and dropped it off. And the smile on her face just says it all. She now calls it her wheelchair instead of wheelchair, which I think is absolutely adorable. And it just reminds me that these kids are still kids, and they enjoy, they deserve a childhood too. So I just want to let that out there. I know it's depressing and whatnot, but there is a glimmer of hope, and those friendships that I have made and that you guys have all made, it just makes it worthwhile. I do want to mention that we do remember one of our Surf One partner families, daughter Lola, who passed away this past summer. There have been many, many, many children who have lost their lives too soon from this disease, and it's devastating. And I just want everybody to know that we are never going to stop fighting in honor of your children. We will never forget them. Lola was a special little girl. They've been with us since the very beginning of Cure Surf One, and we just want Christy and Josh to know that they're with us at this time, and we will never stop fighting for Lola and all those who have passed. And on that note, we are so happy you are here, and I cannot wait to start this conference so you can learn more. I want to introduce Vamsi Mutha, our keynote speaker. So let me tell you a little story. I first discovered Dr. Mutha during one of my many Google researches on mitochondria after Will was diagnosed. His name kept appearing in all these PubMed articles, and I thought, he must be important. He's been around, you know. Little did I know, Dr. Mutha is a world-renowned mitochondria expert. He has made major contributions to mitochondrial biology and genomics. He's an investigator at Howard Hughes Medical Institute, professor of systems biology and medicine at Harvard Medical School, investigator in the Department of Molecular Biology at Massachusetts General Hospital. And he's also an institute member of the Broad Institute of MIT and Harvard. We are so honored to have you here, Dr. Mutha, and we cannot wait to hear your presentation on Leigh syndrome. To the audience, if you have any questions, please put them in the Q&A box and we will get to them after Dr. Mutha's presentation. Thank you so much. And Dr. Mutha, the floor is all yours.
Thank you so much, Casey. Just a quick technical, are you able to hear me and see me properly?
Yes, we can see you. Yep.
Great. Thank you so much, Casey, for inviting me, and to your foundation for inviting me. It's a real honor for me to be giving the keynote lecture this morning on a topic that's very, very near and dear to me. And the title of my talk is Leigh Syndrome Research at 70, Sterling Past and Golden Future. I do have a few disclosures that I want to point out on this particular slide. And I want to just begin by saying that it's awesome. For those of you that don't know this, this is Mitochondrial Disease Awareness Week. And I can't think of a better way to sort of kick it off on Tuesday of this week than to be giving a talk on Leigh syndrome. So thank you again very much for having me. And hopefully this event and other events are just going to increase global awareness for this incredibly important collection of diseases. So this is the outline for my talk today. What I want to do is I want to briefly introduce mitochondria and mitochondrial disease, because of course, Leigh syndrome is a signature mito disease. And then what I want to do is I actually want to do a retrospective and actually look back at the history of Leigh syndrome, because having worked in this space for a long time, I think it's just incredible. And then what I want to do is our lab has historically worked on Leigh syndrome in multiple ways. And I want to just share with you a few vignettes over the years. And then finally, what I want to do is I want to share with you why I am so excited about the future of Leigh syndrome research and what I think may be really, really helpful for enabling significant progress in the years to come. And so, as Casey said, we're all here because we are in some way connected to Leigh syndrome. And taking a little step back, what that means is that we're all here because we are connected in some way by mitochondria, whether they're malfunctioning in a patient, or if you're a family member, or if you're an investigator investigating mitochondria. There's lots of reasons to be interested in this organelle. And this organelle is just absolutely, absolutely incredible. I mean, this is an organelle that used to be a bacterium, used to be a free-swimming bacterium 1.5 billion years ago, and now resides in virtually all of our body's cells. And it still looks like a bacterium, but it's not. It houses the machinery for what's called oxidative phosphorylation. That's a mouthful for you if you're not a scientist, but this is basically the pathway by which the food that you eat gets converted into ATP, the energy currency of the cell. And most of those really complicated reactions are taking right smack in the middle of the mitochondria through a series of macromolecular protein complexes. And so this organelle has a really interesting history. It looks like a bacterium. It still maintains a vestige of its bacterial ancestry, has its own genome. So 2 genomes have to be coordinated. There's a lot of chemistry that's happening here. One of the most interesting facets of mitochondria is that it's a very complicated organelle. And so its components, its machinery comes both from its own DNA. This is the DNA that is transmitted from mom to child. This is a maternally transmitted DNA. And this encodes 13 proteins. But the nuclear genome, the genome that comes both from mom and dad, the big genome, this is extremely, extremely important as well. And we'll get to that a little bit later in the talk. But basically, this is one of the few parts of the cell that requires 2 completely different genomic systems to be operating in sync. Now, as it turns out, there's a lot of conditions, human conditions, that are associated with some sort of a decline in mitochondrial function. And as it turns out, as all of us age, if you take mitochondria from individuals different ages and put those mitochondria into a small device, you can actually measure their rates of ATP production. Our mitochondrial activity is declining as all of us age. And the billion-dollar question in the aging field is cause and effect. Is it the case that this decline in mitochondrial function is actually driving age-associated pathology? Or do you just have sick tissue, so you have sick mitochondria when you get older? And so that's still a big unknown. But this is declining in all of us. And the good news is a mix of strength training and aerobic training can actually send you back north on this trajectory. Now, while this is a very common and relatively mild condition, at the opposite end of the extreme are a very large collection of individually rare diseases. These are rare, typically genetic mitochondrial disorders, where typically children are born with a birth defect in their mitochondria, and there is no doubt that it's the mitochondrial malfunction that's actually causing the disease. And Leigh syndrome is one of the types of rare genetic mitochondrial disorders. And our laboratory is historically focused on these rare diseases. These rare diseases merit a little bit more of an introduction, since I'm the first speaker for today's symposium. When we're talking about rare mitochondrial diseases today, we're talking about a collection of disorders that typically, not always, but typically impact this ATP producing machinery. These can be due to mutations in the mitochondrial DNA or in the nuclear DNA. And one of the real big challenges is that these disorders are associated with a lot of heterogeneity. So even though the defect may occur in the same part of a complex, some patients will only have eye disease, whereas other patients will have multisystemic disease. And we don't fully understand this pleiotropy. And I think what motivates all of us, whether we're scientists or clinicians or patients or foundations, we have zero proven therapies. And I think this is actually a mission that we can all lean in against together, as Casey was mentioning earlier. And so that's a very, very brief introduction to mitochondria. Now what I want to do is I want to do a little bit of a retrospective into Leigh syndrome. This is an incredible story. And so the initial description of Leigh syndrome was 71 years ago. It was about 70 years ago or so. And the initial title of the paper that introduced the syndrome was a bit of a mouthful. It was called Subacute Necrotizing Encephalomyelopathy in an Infant. And so this is actually what we now call Leigh syndrome because of Dennis Leigh, who was the neurologist that first described this case. And I feel a little bit of a kindred spirit with Dennis Leigh because he was a physician, but he also ended up spending about 1 year of his training in Boston at Mass General Hospital, where I work, during his training. And so I think it's really cool that he spent some time in the United States at Mass General Hospital. And this paper is remarkable. For those scientists, clinicians that haven't read this, I strongly recommend giving it a deep dive. It's a heavy read. It's only about 3 or 4 pages, but it's a heavy read. But it begins with a very modest first sentence. The following case appears to be unique, and that no references to a similar condition can be found in the literature. And in this case report, what Dennis Leigh does is he describes the case of a 6-month-old boy that, as Casey said earlier, was perfect by all measures and criteria. It was developmentally fine, behavior was normal until about 6 months of age. And then Dennis Leigh describes this case report of a patient that this boy that ends up presenting to the hospital, and he had actually been well until 6 weeks prior to this hospitalization. And at that time, although his early development and behavior was normal, his symptoms started 6 weeks earlier, right at the same time that his sister had some type of upper respiratory infection. So she had some sneezing and coughing, and that's coincident when his symptoms began. And at that time, he slowly stopped crying, he became somnolent, he stopped sucking. And this ended up progressing to the point that it necessitated hospitalization a few weeks later. And at that time, he was actually found to have optic atrophy. So he's blind, he was deaf. And at that point, he was very, very rigid and spastic. And unfortunately, over a 3-day period, he actually became comatose and died. And so unfortunately, for those of you that are familiar with Leigh syndrome, this may bear a lot of resemblance to what you're familiar with. And so this patient underwent an autopsy. And on autopsy, what was notable was that there was only CNS involvement in this particular case. That's not always true for Leigh syndrome. In this particular case, there's only CNS involvement. And what was immediately striking on gross was these focal bilaterally symmetric necrotic lesions affecting the base of the forebrain, the brainstem, the spinal cord. So all of these regions had these bilaterally symmetric lesions that were necrotic looking. And on microscopic examination, what was very, very striking was this massive capillary proliferation, astrocyte proliferation, microglial involvement. But what was noted was that despite this very necrotic appearing region, there was neurodegeneration, but there's relative neuronal sparing. And so these are all signature microscopic hallmarks of Leigh syndrome that we still appreciate today. And I love the end of this paper. Remember, this paper was written in 1951. This was before we knew that DNA was a double helix. This is before, there's an appreciation that mitochondria could be involved in neurological diseases. And so, in fact, the word DNA or genetic is not in this paper, nor is the word mitochondrion in this paper. But what this paper was a remarkable and first histopathological and clinical description of bona fide mitochondrial disease. And this paper ends with a very thoughtful speculation of the etiology. Was this viral because the sibling had some sort of a viral prodrome? Is it nutritional? Because Dennis Leigh knew that thiamine deficiency would give rise to pathologies resembling the pathology that he saw over here, but with some distinctions. Or was it toxic? And he actually pointed out that there's a few experimental medicines that had been given to non-human primates that would actually give rise to toxicities resembling what was observed over here. So it's a wonderful, wonderful paper. And I think we should always remember and be grateful to Dennis Leigh for really launching this field 70 years ago. Well, the field has advanced tremendously since then. I mean, it's been really exciting to have witnessed and to have been a part of some of the advances in Leigh syndrome research over the last 70 years. And so it begins with this paper that I just told you about. And then in the late 60s and early 70s, it became clear that in patients who subsequently had autopsy diagnoses, they had a lot of biochemical abnormalities. And so there's connections to lactic acidosis, cytochrome C oxidase deficiency. Initially, they thought that it may be a pyruvate carboxylase deficiency. Later, it was clear that it was more of a pyruvate dehydrogenase deficiency. So there are these biochemical associations that made it seem as though Leigh syndrome may actually be some sort of a neurometabolic disease. What was an absolute game changer and really distinguishes Leigh syndrome from a lot of other mitochondrial diseases is the advent of CT and MR imaging to help to identify these lesions. And so now it was possible to make antemortem diagnosis because these lesions have very, very, very specific appearances on things like MRI. And so that's been really important in helping to advance the field. And then in the 90s, early 90s, the first genetic basis of Leigh syndrome began to be established, both in the mitochondrial DNA as well as in the nuclear DNA. And then in 2008, I think one of the really important and exciting landmarks was that Richard Palmeter introduced an accurate mouse model of Leigh syndrome. And as of today, as Casey said, there's greater than 99 disease genes now that have been reported. So today we appreciate Leigh syndrome as being a largely genetic disease, largely a mitochondrial disease, because so many of these genes and pathways directly impact mitochondria. Pilar Miranda and Missy Walker in my laboratory updated the list of Leigh syndrome disease genes as of earlier this summer. And as of today, there's 99 bona fide Leigh syndrome disease genes. Most of these impact mitochondria, in particular that pathway called oxidative phosphorylation. Either those direct subunits are impacted or other pathways that help to support oxidative phosphorylation are impacted. You'll see that there are some genes that encode non-mitochondrial proteins, but again, a lot of these are related to things like thiamine metabolism. Those pathways basically help to assist a lot of these other energy metabolism pathways in the mitochondrion. The field is advanced. I mean, not only do we now have 99 disease genes, but as we'll hear later in the symposium, there's quite the clinical trial landscape right now for mitochondrial diseases in general. That's what's shown over here. Missy Walker and my group updated a list of the ongoing trials. They may not be perfect, but largely guided by clinicaltrials.gov or clinicaltrials.org. And so you can see that there's lots of drugs in phases 1, 2, 3, and 4, multiple modalities for a variety of mito diseases, and boxed are the ones that are specifically calling out Leigh syndrome as indications. And so, now we're in an era where there's lots of genes as well as active clinical trials as well. So today, and I think when the scientists and the clinicians are presenting today, when we're talking about Leigh syndrome, I think most of us are adopting the definition that was put forward by Shamima Rahman and by David Thorburn a couple of years ago in a very, very important and influential, I would say, definitive review on this topic. So I think now when we're talking about Leigh syndrome, we're talking about an early onset neurodegeneration that's characterized by psychomotor retardation, regression with progressive decline, often in a stepwise manner with frequent decompensations, often with illness. We like to see these MRI lesions that can impact the brainstem, the basal ganglia, or the spinal cord. These are what are called T2 intense lesions. And we also like to see some sort of a biochemical defect in energy metabolism. There's a variety of ways of picking that up. And today it is so straightforward to genetics, to do genetics, that we also like to see some sort of a genetic mutation as well that could explain the syndrome. And so that's what we call Leigh syndrome, but often you'll hear the term Leigh-like disease or LS spectrum. And these are cases where many of the above conditions are fulfilled, including the radiographic and histologic. So that's a bit of an introduction to mitochondria, and now we've done a little bit of a deep dive into the history of Leigh syndrome, which I think is so exciting. Now what I want to do is I want to make this a little bit more personal and actually just share with you my first encounter with Leigh syndrome. I've been working on mitochondrial biology for about 29 years at this point, but my first encounter with Leigh syndrome as a disease was about 21 years ago or so. And ever since then, my lab has consistently worked on some facet of this disease. And so I want to turn the clock back a little bit and share with you a few vignettes. And so about 20 years ago or so, I was doing a postdoctoral fellow at the Whitehead Institute with Eric Lander. He was leading the sequencing of the human genome, and I'd already developed an interest in mitochondria. And I was very interested in the nuclear genome's contribution to mitochondria. And at that time, one of my colleagues at the Whitehead named John Ryu told me about a disease that I'd never heard about before. And that was called Leigh syndrome, French-Canadian variant. And this was a form of Leigh syndrome. And at that time, this was one of the most common genetic diseases in the Saguenay-Lac-Saint-Jean region of French Quebec. It had first been described in 1993, impacted about 1 in 2,000 live births. The carrier rate was 1 in 23, and it was already appreciated that this was an autosomal recessive disease. Clinicians had just begun to describe the syndrome, and it was associated with brainstem gliosis and necrosis, so these brain Leigh syndrome features. These patients had developmental delay and ataxia. They also had liver disease and facial dysmorphism, which is actually not that common in these diseases, but in this particular form, there was facial dysmorphism. And unfortunately, these children were dying in the context of neurometabolic crises, somewhere between the ages of 6 months and 12 years of age. And so this was a disease I'd never heard of before, but my colleague, John Ryu, whose primary focus was inflammatory bowel disease, also took a special interest in this particular disease because he happened to be from that region and was interested in deciphering its genetic basis. And what I think is really, really exciting about Leigh syndrome is that Leigh syndrome French-Canadian variant was actually and I had nothing to do with this study. This particular study I'm about to tell you about predates my involvement. But what's really cool is the first successful application of a technique called genome-wide association studies, or GWAS, which is used to find genes for psychiatric disease, for neurodegeneration, for inflammatory bowel disease, for diabetes. This is the mainstay genetic methodology for common disease genetics. But as it turns out, its first successful application by Mark Daly and by John Ryu and Eric Lander was on the French-Canadian form of Leigh syndrome. And in this particular study that was reported in 2001, they're able to use this technique. Some people call it genome-wide association studies. It's also called LD mapping. they were actually able to map the disease gene to chromosome 2, and in particular, the short arm of chromosome 2. And so at that point, John Ryu approached me and said, hey, we've been able to use this new genetic method, and we've been able to zero in on the short arm of chromosome 2. But however, there's 60 either known or predicted disease genes, and we don't know which of those is actually the culprit disease gene. Can you help us? And so this is super fun for me, because at that time, I was already developing a real interest in mitochondrial proteomics, and I liked coding. And at that time, I really enjoyed taking all of the publicly available data and doing what's called co-expression analysis. So John presented me with this challenge, which is we have 60 known or predicted genes in this interval. Which of these may be important for a mitochondrial disease? And using code, I was a math and computer science major in college. I was relatively fast. I like coding. Writing relatively simple code, I integrated these 3 data sets, and out came exactly one gene called LRPPRC, which nobody had studied in the context of human mitochondria prior to this work, but it was so clear that this thing co-expressed with all known mitochondrial genes, and some of the peptides from my mitochondrial proteomics work were also landing onto this locus. And so this became the culprit. We resequenced it, and we found the causative gene in LRPPRC. At the time, this was a big deal. This is actually a method that we called integrative genomics. It was the first successful application of integrative genomics. The Wall Street Journal decided to pick up this story, and it was very, very exciting. And what I want to show over here is a painting that was actually done by the mother of a boy and a girl who lost their lives to Leigh syndrome French-Canadian variant. This, of course, was a pretty devastating set of events for her in her life. And she was a part of a foundation that helped to raise some funds to support Leigh syndrome research. And she did this painting, and after we had discovered the disease gene, she actually sent to us this print of her 2 late children that were biking around this lake as a token of appreciation for the work that we had done. And to this date, this painting actually hangs in our laboratory as a reminder of why the work that we do is so important. What I'll note is that many people in this community actually underwent genetic screening and have now successfully undergone pre-implantation genetic diagnosis and have actually had healthy children. So a very concrete consequence and benefit of finding the disease gene in a community where this particular disease was relatively common. That particular work as a postdoc reinforced upon me the importance of the nuclear genome's contribution to mitochondria. The mitochondrial DNA, which is sequenced by Fred Sanger, it only encodes 13 proteins, but all of the other business for mitochondria is encoded by the nuclear genome. And so some of the work from our laboratory over the years has been aimed at identifying all of these other 1,100 proteins, we call them mitochondria, encoded by the nuclear genome that end up in this compartment. And we used a variety of methods: mass spectrometry, computation, proximity by attenuation, a series of papers over the last 15 years or so that have yielded this protein inventory. So out of the 20,000 genes that's found in all of our human genomes, about 1,100 of them encode proteins that come and produce mitochondria. This is a inventory we call mitochondria. This is an atlas for basic scientists as well as for human geneticists and for clinicians. We make this freely available to the community. And this has helped us as well as others identify not just LRPPRC, but a number of other monogenic disease genes for Leigh syndrome, as well as for other mitochondrial diseases. Now, over the last couple of years, we've shifted a little bit from disease gene discovery because this has become relatively straightforward using next generation sequencing. And we've now shifted our attention to biomarker discovery. As I'll talk about at the very end of this presentation, I think one of the most important unmet challenges for all of Leigh syndrome research is the lack of facile biomarkers with which to monitor disease progression. This is super, super important. We need biomarkers so that we can monitor disease progression, so that we can classify diseases, and so that we can quantify therapeutic response. A biomarker is something like glucose for diabetes. Imagine if you had diabetes, if you can't measure glucose, how can you possibly manage your disease? How can a drug possibly be approved? And so what we've been trying to do is we've been trying to use a technique called tandem mass spectrometry metabolomics. Think of this as something like a glucometer, not for just glucose, but for 400 metabolites. And our goal has been to determine which of these 400 metabolites that we can measure are altered in Leigh syndrome versus controlled. And the study that we did was actually a prospective case control metabolomic studies of Leigh syndrome French Canadian variant, the very disease that I just told you about, whose genetic basis we had identified years earlier. We now partnered with Christine Desrosiers as well as the LSFC consortium, and we actually performed a prospective case control metabolomic analysis. This is a very, 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 very carefully designed study. Although it was only 9 patients and 9 controls total, they were all matched rigorously for age, gender, BMI, activity status, yada, yada, yada. And in this way, we could do metabolomics. And what was very gratifying is that on principal component analysis, we could actually perfectly separate the cases and controls on the basis of disease status, not on the basis of some other covariate. And so what are these metabolites? Most of the metabolites that distinguish cases from controls in Leigh syndrome, as well as in other mitochondrial disorders of the respiratory chain like MELAS, are metabolites like lactate and alpha hydroxybutyrate. This is not beta hydroxybutyrate, the ketone body some of you may be checking if you're on the Atkins diet. This is alpha hydroxybutyrate. It's a part of the transsulfuration pathway. We're finding that these tend to be elevated in patients with respiratory chain disease. And we can find other metabolites whose mechanistic basis we think we understand. And all of this is working towards what we call mitochondrial function tests, in the same way that if you have liver disease for that particular organ, you have liver function tests. If you have a dysfunction of the mitochondrial organelle, we want to develop organelle function tests, a panel of tests that give us a facile readout of mitochondrial function that can be useful for clinical management as well as for clinical trials. So our vision for mitochondrial medicine is that from a single tube of blood, we can establish a genetic diagnosis using something like next generation sequencing combined with a triangulation on the mitochondria inventory. And that from that same tube of blood, from the plasma or the serum, we can biochemically stage the disease. So now if we have a novel therapy that we can give to the patient, we can monitor objectively their therapeutic response. So all we need now are novel therapies. And so the last vignette from my own laboratory that I'll just share with you is some of our efforts at trying to identify therapies for mitochondrial disease. And about 6 or 7 years ago or so, we made what I think is a really, really unexpected, super interesting finding. We did a genetic screen, and we actually discovered that hypoxia or low oxygen is actually a suppressor of mitochondrial dysfunction in preclinical models. In other words, what we found is that in cultured cells, other preclinical models where we can poison mitochondria in a dish, we found that decreasing the ambient oxygen was actually extremely helpful to those cells. And so this actually led to the hypothesis that low environmental oxygen may actually be a preclinical therapeutic. Now, what I want to emphasize is that everything I'm about to show you is still in the laboratory. It's still in the preclinical realm. None of this is ready for application to humans. I want to just emphasize that this is still basic research. So this paper gave rise to a very counterintuitive idea that low oxygen may actually be good. And what we ended up doing is we ended up testing this hypothesis in a mouse model. As it turns out, our field now has excellent mouse models of mitochondrial disease, including Leigh syndrome, thanks to the work of Richard Palmer's group in Seattle. In 2008, they introduced a complex 1 deficient mouse model that develops Leigh syndrome. So it's born fine, it's developmentally okay, it weans at day 20, and then around day 50 or so, it's very sick, it's hypothermic, it's lost weight, and it develops these lesions on MRI that resemble the lesions that we observe in humans. And so we happen to have this mouse model in our laboratory, and we decided to see what happens if we decrease the ambient oxygen from 21%, which is what we're all breathing if we're in Texas or in Boston or at sea level, and we decreased it to 11%. And so this is comparable to places like Pikes Peak, Mont Blanc, Base Camp. but it's not Everest. It's something like Mont Blanc. So about 15,000 feet elevation. And again, this is, we do this by diluting the air that the mice are breathing with nitrogen. Now, for the purposes of the video, all 5 of these mice that have mouse Leigh syndrome are in the same box, but 2 of them have been breathing 11% oxygen for the last couple of weeks. So if you see if you can guess which 2 those are. So I hope that was obvious. The 2 that were running circles around the others had been breathing thin air for the few weeks prior to that video. These mice ordinarily die at day 55 from their neurological disease, but if they're breathing 11% oxygen instead of 21%, that's what the survival curve looked like at the time of publication. In fact, if we carry out the survival curve beyond the publication, these mice survive to a median of about 315 days or so. So a massive, massive improvement in survival, as well as not just lifespan, but health span as well. And so again, this is a preclinical concept that we're very, very excited about. So as you can see, both when I was a postdoc as well as subsequently in my laboratory, we've been very interested in the genetics, the biomarkers, and preclinical therapeutics for Leigh syndrome. So now in the final 5 to 10 minutes, what I want to just share with you is just take a step back and just share with you why I'm so optimistic about Leigh syndrome research today. We really are at a hockey stick moment for Leigh syndrome therapeutics. But what I also want to end with is what I think is going to be required to drive the field forward. And this is where this foundation and others are going to be playing an extremely important role. So I want to provide a little bit of context. I mean, I'm based in Boston, and this is Kendall Square. Okay, this is Cambridge, Massachusetts. This is Kendall Square. If you just go right across the bridge, you're going to be at Mass General Hospital. So I have a lab at Mass General Hospital and at the Broad Institute, and that's Boston. This is Cambridge, and this is Kendall Square. This is basically, I would say, one of the meccas of biotech in the entire world. And if you just go back into the late 60s or so, this is actually what Kendall Square looked like. And it's because of the molecular biology revolution, a lot of work from academics as well as from industry, all of this blossomed into one of the epicenters for biotech. And so I feel very fortunate because I'm surrounded by people that are really innovating at all levels to try to develop new medicines. And this is a great little video that was developed by MIT, if any of you are interested, on how the biotech boom started in part at MIT, Harvard, and in Kendall Square. I want to share with you some really exciting things that are happening more broadly. We are now in the era of definitive medicines for genetic disease. And so here's 2 drugs that I'll just call out that I think are really exciting. So Ionis developed a drug called Spinraza for spinal muscular atrophy, a really, really devastating neurological disease. The gene was cloned in 1995. Very good basic biology identified what I call a backup gene in 1999. And in 2016, a novel therapeutic modality, ASO therapies, were developed that basically reawaken that backup gene that's ordinarily not inflated. It's a really neat approach. And this leads to really, really meaningful, sizable improvements in these children. Another really remarkable story came from cystic fibrosis. The gene was cloned in 1989. The Cystic Fibrosis Foundation was crucial for helping to provide a partnership, both not just financially, but also in terms of connecting to patients and helping to support research both in academia as well as in industry. On the academic side, one of the most important things that was discovered was that the most common mutation for cystic fibrosis, it's actually a temperature-sensitive mutant. In other words, there's nothing inherently wrong with that protein if you're at the right temperature. It's just that at 37 degrees, it unfolds. And that provided a rationale for the biotech sector to develop small molecules that will basically bind to that protein and stabilize it. It confers thermal stability. And this remarkable trio of molecules was approved in 2019 and is really game-changing and literally life-changing for cystic fibrosis. These are both genetic disease. Genes were identified. There's really good basic biology. And the reasons that these programs were so successful, in my opinion, is that there's a really good understanding of the human genetics, deep understanding of the human biology, disease biology. They made clever use of emerging technology. And importantly, in these instances, there was a deep collaboration with patient research and advocacy groups. And so I will argue that this is basically where we are for Leigh syndrome. So as of today, unlike 71 years ago, we now appreciate that Leigh syndrome is a largely genetic disease. There are environmental triggers for sure, but at the root is a genetic predisposition for these diseases. We have 99 disease genes. We understand a lot of the proximal pathways that are broken subsequent to these 99 genes being disrupted. And what's really special about Leigh syndrome is we have excellent diagnostic imaging that allows us in a sensitive and specific way to identify these cases. Now, what I'll point out is that unlike those other cases like cystic fibrosis, major challenges that we face as we as a community try to take on Leigh syndrome are the genetic heterogeneity, 99 different disease genes, and in some instances, some of those genes harbor a number of different disease alleles, so genetic and allelic heterogeneity. There's a lot of pleiotropy. Sometimes a mutation in one gene only impacts the brain, and in other instances of Leigh syndrome, you'll have multiple organ systems that are impacted. And importantly, each of these individual genetic forms of Leigh syndrome is ultra rare. And so these are some unique challenges that we're going to have to take on as a field. Right? Now, I think moving forward, what's going to be very important, extremely important for Leigh syndrome is a concept that was really put forward by Victor McCusick, who wrote famously about the nosology of genetic diseases. And in particular, he described the notion of either splitting or lumping. And so if you're a lumper, you're going to take all 99 of these genetic forms of Leigh syndrome and lump them together and treat them as one. If you're a splitter, you're going to break up all of those Leigh syndromes with those T2 intense lesions and break it up into 99 different genetic forms of disease. This concept is important. In some instances, being a splitter is good, and in some instances, being a lumper is good. And so this is the high-level schematic that I'll put forward as a concept moving forward. I think that in order to be successful in the future, we're going to have to rationally and very intelligently lump some of these genetic forms of diseases and then develop gene agnostic, say, small molecule therapies that basically impact some of the common final effectors. Alternatively, we may choose to be splitters, which is fine, in which case what we're going to do is we're going to develop either gene replacement for the nuclear genes, or down the road, not immediately, because this is still very, very early technology, but maybe 5 to 10 years from now, hopefully, mtDNA editing of the mtDNA. So these are approaches that could be applied to individual genes and alleles. These can be applied in a lumped manner, if you will, to target common effector pathways. Both approaches are going to have merit. Both approaches are going to be essential for Leigh syndrome. And what I want to emphasize is that we are never going to have a single magic bullet for all forms of Leigh syndrome. They're just too heterogeneous disorders, too many organ systems that can be impacted. I think for certain forms, we may have magic bullets for individual forms of disease, but not for all of them. So we're going to need multiple approaches. So in the final 1 to 2 minutes, what I want to share with you is what I think is going to be important for driving the field forward. First and foremost, it takes a village, as Casey said early on. This is going to require collaboration. In order to solve Leigh syndrome in a really meaningful way, we're going to have to all work together. And that ecosystem is going to include the patients, the foundations, academics, companies, regulatory agencies. We all need to work together efficiently and collaboratively. One of the things I want to emphasize is that mitochondrial disease can wax and wane. And so this is a hypothetical natural history of a patient with Leigh syndrome. Okay, so patient might be fine. There's some type of a viral prodrome. There's a little bit of regression, happens again. Patient spontaneously gets better for whatever reason. and then there's another decline. This is actually what this up and down stuff is actually what a lot of these mitochondrial diseases look like. We really need to have strong scientific theses, prospective natural history studies, and rigorous clinical trial design, given this waxing and waning nature of mitochondrial disease. Because imagine a scenario in which you give a drug and you just happen to give it here. Right? Let's just say that you gave a placebo and you just gave it over here in an N of 1 drug trial. Well, the patient spontaneously got better. You may then end your drug trial and conclude from this N of 1 drug trial that your drug was actually helpful or efficacious. And if you're a parent, you may get really excited about what this particular drug did. And if you're a biotech, you may be very, very excited about it. But we have to appreciate that these drugs wax and wane. And we need to have extremely, extremely rigorous trial design, when possible, randomized controlled trials, so that we can make sure that the drugs that we are pushing forward are safe and efficacious. And in order to do that, we're really going to need to have high quality patient registries. We're going to need prospective natural history studies for Leigh syndrome. We don't have that right now. And I think what's going to be super important for the field, independent of the therapy, is we desperately need high quality biomarkers, whether they're blood-based. I'm also excited about digital biomarkers to aid in clinical trials. That's going to create a foundation. And then, lots of folks are developing exciting gene therapies, gene editing for the nuclear genome, as well as for the mtDNA. This is going to be useful for targeting specific genes or alleles. So for the splitter strategy, we also need therapies that target downstream effectors for the lumpers. We're going to need both. And finally, what I'm going to argue is that any new therapy really should be tested in preclinical models. As of 2022, we have excellent mouse models for mitochondrial disease, as well as for Leigh syndrome. There's almost no reason why a candidate therapeutic shouldn't be tested first in preclinical model. What I'll point out is that in that model that I alluded to earlier, just one of the mouse models of NF-S4 deficiency for Leigh syndrome, there's already multiple promising therapies. Again, these are in a preclinical setting also. They require a lot of work, whether it's hypoxia, whether it's a drug that targets microglia, rapamycin, doxycycline, gene therapy. So I think this is really exciting that we have good preclinical models and even some preclinical approaches that are showing promise. And so if we have this and we continue to always maintain scientific rigor and clinical rigor with the highest ethical standards, I really do think that we could see transformative medicines as early as 5 years from now, especially for things like small molecules, and maybe a decade from now when we talk about really cutting edge approaches like mitochondrial DNA editing. And so I think this is what is going to be needed for the field. And I think Casey, your foundation is going to be playing such an important role on a forward-looking basis. I want to just end by just thanking all of our patients and their families. I'm just showing Abby McCurtain and her father, Greg McCurtain, who run the Boston, he runs the Boston Marathon every year to provide support for our laboratory and also increase awareness for mitochondrial disease and Leigh syndrome. But I'm really wanting to just thank all of our patients and their families, because you are a true source of inspiration for all of us academics and others that are doing research in the space. And we want to be able to work with you to try to develop solutions and therapies. And I want to thank you for all the inspiration that you provide. I also want to just let you all know that there's a lot of us. I'm just showing my own laboratory at our 10th year anniversary, our 15th year anniversary, and our lab today. I want to thank all of my past and current members of my laboratory that have joined me in working on mitochondria and mitochondrial disease. And if you're a patient or a family member, please know that there's many of us that are working as hard as we can to try to come up with meaningful medicines for Leigh syndrome. So with that, I will stop and I'll field any questions that you may have.
Wow. Thanks, Dr. Mutha. I feel just enlightened by your presentation. I will say that I'm a splitter and now I can appreciate the lumpers. So it is going to take, like you said, a full circle. Let's get to some questions. This one came from Nikolai. It says alternating and appearing and disappearing of brain lesions are reported in literature and by parents. Have you found correlations between any factors, supplements, environmental, lack presence of seizures, and these disappearing lesions?
Yes, we actually just wrote a little review article perspective on the fact that these brain lesions can be reversible. And in our mouse model, Casey, we that we can actually take that mouse model to the brink of death, and then we can actually start hypoxia when they have advanced brain lesions. And after a few weeks of hypoxia therapy, we can actually make those lesions disappear. And so that's mouse data. But in a very recent paper that was co-led by Pilar Miranda and by Missy Walker from my laboratory, we did a case review, and there are case reports of patients that have T2 intense lesions. Their fever subsided, they just got better for whatever reason, and months later, the lesions disappear. So we actually think that there's an early part of the pathology that is visible to the MRI, that it's still reversible. And then, of course, once the neurons die, that's going to be hard to reverse. But we do think that there's very strong case reports and mouse data to suggest the reversibility of the early phases of the inflammatory disease.
That is truly amazing. We do have a follow-up question. What could hypoxia training look like for our children? And I know we're in early stages, so.
Yeah. And Casey, I want to emphasize, I really, really want to emphasize that we're still in the early stages for hypoxia therapy. It works beautifully in mouse models, but it has to be chronic, continuous hypoxia. One thing that we have reported is that if you do intermittent, it does not work. And in certain mouse models, intermittent can actually even be worse. And so we really need to understand the biology of hypoxia therapy. And then our long-term goal is to try to put that into a pill. So I would say we're not ready for any studies in human patients yet.
That's fair enough. Fair enough. Jessica, I think you have a question that you'd like to ask live. So I'm going to let you ask that question.
For a great talk. So I just wondered, the hypoxia treatment is really exciting in the animal models, and the animal model you've used has variants in the nuclear genome. I wonder if you thought at all whether this hypoxia treatment might work for diseases that involve variants in the mitochondrial genome, and have you tested it, or have you thought about whether that might be an effective therapy also for those forms of Leigh syndrome?
No, thank you, Jessica. So we haven't tested yet in any of the mtDNA based models, but I think there's no reason to think that it wouldn't work in those as well. At least based on cell culture studies and worm studies and other mouse models, the hypoxia approach does appear to generalize not to every mitochondrial disease, but a very large subset. And so I think there's reason to be cautiously optimistic that it would extend to mtDNA models in a preclinical setting as well.
And just a quick follow-up question. So with the hypoxia, did you notice any off-target effects that might result in, symptoms? And if so, or if you haven't yet, have you thought about how you might mitigate some off-target effects? Being a cardiovascular biologist, I know the heart is really sensitive to a lot of
No, and this is why we really want to try to get this right in a preclinical setting. And I think some of the toxicity to be concerned about with hypoxia is probably not off target but on target. And so one of the things that we do worry about with really chronic hypoxia is right-sided heart failure. So you can actually develop a cardiomyopathy and pulmonary hypertension. And so at least in these mouse models, we're not seeing that as a problem. But of course, if once we try to enter into humans, that's something we'll have to be anticipating and be very, very careful about.
Thank you.
Okay, we have time just for a few more questions so we can stay on schedule. Switching over to metabolomics. Are the metabolomic changes primary or secondary to Leigh syndrome? How can we be sure they are primary? This is from Isabel.
Yeah, these are great questions. We think that they're very, very, very early. We actually understand the mechanistic basis for the alterations. And so But we like to think of these as sort of ripples and responses. And so as soon as you have a proximal genetic defect in one of those genes encoding the respiratory chain, some of the earliest biochemical alterations, so we understand why, are corresponding to these elevated lactate and alpha hydroxybutyrate. Now, whether those are on the causal path to Leigh syndrome pathology, we don't know the answer to that, but these are early mechanistic markers that we're looking at.
We have a lot of questions coming in, and I know we won't get to all of them, so I will send them to you separately and see if you can answer them through email. Thank you so much, Dr. Mutha, for your time. We are so honored to have you as our keynote today, and thank you for all you do. We really appreciate all your work. Thank you.
Thank you so much, and best wishes to everyone today, this next week, and for the future.
Thank you. Now I'd like to introduce Dr. Saima Kiani. She's a pediatric neurologist at UT Southwestern here in Dallas, Texas, with additional training in genetics. She's passionate about helping children with rare disorders and finding better ways to treat these children. Saima, it's all yours.
Sorry, can you guys hear me?
Yes.
Some technical difficulties. Sorry, can you guys see my screen?
We can see it, yes.
Okay, let me go to that. Sorry. Okay, well, thank you so much, Casey, for inviting me to speak and present in this platform. Today, my talk is going to be around the clinical features of Leigh syndrome as we know it. It is just
Emma,
going. Hello.
You need to swap your display. We're seeing the presenter view. If you go up to swap display, there you go. There you go. Perfect.
Okay, perfect. So I guess as I was saying, I'm going to talk about clinical phenotype and clinical features of Leigh syndrome. And what a great talk by Dr. Mutha. I think some of my talk will have overlapping, features, but I think it was a nice segue because as we discussed that moving into a future where we want to have clinical trials for rare diseases. And one of the challenges that clinicians and clinician scientists face is when these trials or these therapies will come to the bedside, there has to be clinical outcomes and clinical endpoints. And in a disease where we have genetic heterogeneity, we have phenotypic heterogeneity, finding those clinical endpoints is going to be a daunting task. And I think understanding the natural history of the disease, whether through retrospective data or through prospective natural history, is very important. In my point of view, I think we all understand that prospective natural history study will be the gold standard because it can also provide us with a standardized cohort or a placebo, sort of like a placebo cohort for future clinical trials. But in my point of view, we start with understanding the basics, going back to the retrospective chart review, understanding the natural history of the disease from there, and then producing or proposing prospective natural history studies, which will help us with the future translation of the basic scientific therapies. So my talk will be an overview, as I discussed, of clinical features of Leigh syndrome. Leigh syndrome, as we are all aware, was first described by Dr. Dennis Lee in 1951. And it was a neuropathologic diagnosis made at that time. And another name for Leigh syndrome is subacute necrotizing encephalomyopathy. Leigh described a patient who presented in infancy with drowsiness, ended up having blindness and deafness and bilateral spasticity following a brief encephalopathic illness. As Dr. Mutha mentioned, the first link to the energy metabolism dysfunction was established in 1968 when a patient was diagnosed or when a patient was found to have lack of pyruvate carboxylase activity in their liver. Later on, the link to the mitochondrial respiratory chain dysfunction was established in 1977 in a patient who had cytochrome C oxidase deficiency. We have come a long way since 1951 and 1968 and 77, and we know that Leigh syndrome now, it's one of the most common mitochondrial disorders of childhood, which affects 1 in 40,000 children in US alone. So various groups have defined Leigh syndrome, as Dr. Mutha also alluded to, and he actually described that too. Leigh syndrome is a progressive neurometabolic disorder. It involves degeneration of central nervous system, and we see characteristic symmetric lesions in basal ganglia and subcortical brain regions, where actually the definition of Leigh syndrome and actual description of Leigh syndrome comes from. Now we know that it is connected to the mitochondrial energy production dysfunction or deficiency of respiratory chain enzymes. And we know the molecular basis of the disease as well. So combine all of these pictures together and you will get Leigh syndrome. And that's all of these 4 components define Leigh syndrome at this point. After the first description of Leigh syndrome, multiple case series have came out describing various clinical features of Leigh syndrome. As now we know that it's a multi-system disease. Failure to thrive is one of the most common findings we see in patients. Developmental delays and regression is seen in a lot of patients. Ataxia and movement disorder is also seen in many patients. Ophthalmologic disorders, oculomotor problems with abnormal motor movements of the eye muscles is also seen in these patients. Biochemically, we have elevated lactic acid in serum and CSF, and MRI abnormalities, as we discussed. A lot of these patients can have pulmonary problems, clinical issues with episodic apnea, irregular breathing, and they would need pulmonary support. So just reviewing this of all the literature review, I picked out this article which came out in 2014. It describes 130 patients. 77 at that time were had an identified genetic mutation. The rest did not. So you can see until recently, Leigh syndrome or Leigh-like syndrome, we know the genetic mutations for most of these patients, but there are probably other mitochondrial disorders or energy metabolism disorder that are just not known to us. And they present with the clinical features of Leigh syndrome, but we are not able to fully identify the underlying molecular basis in some cases. In this particular case series, 77 patients had genetic mutations, out of which 28 patients had a mutation in mitochondrial DNA, and the rest had a mutation in one of the nuclear genes linked to Leigh syndrome. Median age of onset is 7 months, according to this patient, but 80% of patients had presented by age 2. The most common clinical feature in this case series was abnormal motor finding. Again, mind you, this is not the presenting clinical symptom. This is something that patients were found to have after over a time period. So abnormal motor findings could be low muscle tone or hypotonia, abnormal deep tendon reflexes. Some of these patients had dystonia, spasticity, ataxia, which is lack of balance and coordination, weakness in their muscles, chorea, which is a form of an abnormal movement, and they can also have hyperkinetic or akinetic movements. Other features included feeding difficulties in 60% of the patients and failure to thrive in 16%. Now, it's hard to kind of like understand if it was an additive effect or there was some overlap in these numbers. But in our experience as well, we see that a lot of patients with Leigh syndrome will have feeding difficulties or some sort of GI disturbance or GI gastrointestinal system involvement. It can go anywhere from reflux to intractable reflux to motility issues to complete failure to thrive. In this particular case series, cardiac dysfunction was seen in 17% of patients, and respiratory dysfunction was seen in 50% of patients. So looking at the Leigh syndrome as a multisystem disease is an important thing when we, again, move from understanding the disease and taking it to a clinical trials, and then looking at the therapies that do not only target the brain, but also help us treat other systems as well. This graph is taken from this comprehensive review of SIRF1 deficiency, which is one of the most common forms of cytochrome C oxidase deficiency or complex 4 deficiency, and one of the most common forms of Leigh syndrome. In this case, you can see again, vomiting and feeding problems were seen in 89% of the patients, with growth failure in 95% of patients. Hypotonia and then developmental delay were the next in line, with 93% and 88% prevalence in this cohort. Developmental regression is another common finding that is seen in patients with Leigh syndrome. Gait ataxia was seen in half of these patients with movement disorders, which could be a different type of movement disorder, either myoclonus or chorea, akinetic or hyperkinetic movements in half of the patients in this case series. In these findings, we were able to do one of a retrospective chart review with Caregiver Foundation, with the help of Caregiver Foundation, and these findings are somewhat similar to what we were seeing in our cohort. Now, moving on to the neuroimaging findings, as we discussed, Leigh syndrome, at the basis of Leigh syndrome, the definition is that we see symmetric T2 hyperintensities in deeper brain regions. What does it look like? So in this slide, you can see, if you can see my arrow, this is part of the basal ganglia where we see these T2 hyperintensities. So when we say T2 hyperintensities, what does it mean? It just means that you're seeing a bright signal in a very particular sequence of the MRI, which is T2, or flare sequences, which are derived from T2 sequences. And these hyperintense signal, or these bright signals, they signify an underlying pathology or some sort of problem with the brain tissue. Over here in the midbrain, you also see that abnormal signal in the form of 2 bright dots in the back of the midbrain. In the brainstem, you see these abnormal bright signals kind of like extending towards the brainstem, extended throughout the brainstem. Now, if you look at the supratentorial white matter, in this particular case, it is relatively spare. And in most of the cases of Leigh syndrome, white matter or supratentorial gray matter can, at least initially, can look normal. Now, in cases of Leigh-like syndrome, you can see hypomyelination or abnormal maturation or myelination of white matter. And that's, again, something that is seen in patients with Leigh-like syndrome, with a similar genetic mutation, but maybe a little bit of phenotypic heterogeneity, where we are seeing not the typical Leigh syndrome lesions, but a little bit different lesions than Leigh syndrome. Keeping in mind, again, if we are treating a genetic disease with a targeted therapy or precision medicine, with the idea of a precision medicine and a targeted, maybe say gene therapy, then including, thinking about the clinical outcomes. We have a very good biochemical neuroimaging, marker, but does that include all the patients who have their genetic abnormalities? Sometimes you can see a little bit of difference in neuroimaging findings, but here we are seeing the typical Leigh syndrome lesions that define the subacute necrotizing encephalitis. In this particular patient, this first MRI was done close to 1 year of age, and in second MRI, you can see that these lesions have been progressive to where in the basal ganglia on the upper right-hand side image, you see the extension of that bright signal, extension of the bright signal abnormality, and it extends further than it was before. When we go into the midbrain and the brainstem, you see that it has extended to the point where we are now seeing some atrophy of the brainstem and with atrophy of the cerebellum as well, which is part of the brain that is connected to the brainstem through, and you can see here that we are seeing a little bit more atrophy in the cerebellum in this particular case. So the lesions in Leigh syndrome can be progressive. MR spectroscopy is another it gives another very good neuroimaging or biochemical marker for patients with Leigh syndrome. As you can see in the first patient here, we have this lactate peak. This lactate peak is a very specific marker for mitochondrial encephalopathies, including Leigh syndrome. And here in this patient with the mitochondrial DNA mutation, it's an elevated but inverted lactate peak at 1.3 parts per million, where we usually see the lactate peak. And whenever we see a lactate peak on MR spectroscopy, we usually send for genetic testing to rule in or rule out mitochondrial disorders. In the second example here, we have a 6-year-old with complex 3 deficiency and with a pretty prominent lactate peak at the same spot, which again is a very specific biomarker for Leigh syndrome. Again, it is specific, but how sensitive is this biomarker? We really don't know. And I think when we say Leigh syndrome and all these case studies, all these case series have been in a cohort of patients where we have genetic heterogeneity, which is unique to Leigh syndrome, and it is beautiful in itself, but at the same time, it kind of like poses a little bit of difficulty when you are planning to use this as a specific biomarker for a specific treatment for one underlying genetic disorder, which makes the case to understand these diseases as per their genetic basis, as well as, I think we're just, like Dr. Mutha was talking about, splitters and lumpers. So do we need to understand them from individual genetic mutation and the genetic basis standpoint, as well as from the phenotypic, the clinical phenotype standpoint? In this case review, they describe the distribution of Leigh syndrome-like lesions or Leigh syndrome lesions. And you can see in brainstem, 9 out of 17 children of the cohort had lesions in the brainstem, including the medulla oblongata, midbrain, and pons. And we can see that 11 had lesions in basal ganglia. So 11 out of 17 were in basal ganglia, 9 out of 17 were in brainstem. You can see the thalami which is sometimes considered as a part of basal ganglia, but some people would like to consider it as its own entity. 3 out of 17 patients had lesion in thalami. So, and the rest, cerebral white matter, not in this cohort, they didn't see any changes. Cortex, cerebellum, atrophy and delayed myelination was seen in some of the patients. But you can see that most of the lesions, most of the burden of the lesions, these lesions is in the brainstem and basal ganglia. So something unique about the biology or developmental biology of these brain structures that are more needy, so to speak, of the energy deficiency that is produced by Leigh syndrome. And those are the places that are impacted first before anything else. Now, keeping in mind that all these retrospective chart reviews will give you some retrospective analysis and a cross-sectional analysis in some cases, but now would these lesions progress to involve the other areas of the brain, and how these lesions really evolve over time in patients is something that's that we really need to that we don't understand completely. In this case series, 7 out of 17 children had progressive brain lesions, and I'm not clear on how many patients they actually had a repeat brain imaging on, but 7 out of 17 had progressive lesions. Now, looking into the biochemical abnormalities, and this is something that we have Dr. Mutha has also spoken about. So understanding these biochemical abnormalities and getting an overview of them. So our main oxidative phosphorylation or aerobic metabolism depends on glucose metabolism. The endpoint of glucose metabolism is pyruvic acid. Pyruvic acid gets converted into acetyl-CoA through PDH enzyme, which is one of the most important enzymes in aerobic metabolism. As it gets converted into acetyl-CoA enters the TCA cycle, which produces these very important compounds, NADH, FADH2, that enter electron transport chain. Whenever we have a dysfunction of electron transport chain, you can imagine that there will be backup of the metabolites, because there is clog in the system, so to speak. So with this clog or block in the system, you get a retrograde effect where you get intermediates of TCA cycles that can be elevated. And what are those intermediates? Lactic acid being, lactic acid not an intermediate of the TCA cycle, but going back here, pyruvic acid converts to acetyl-CoA on one side, and it converts to lactic acid on the other end. And it is in equilibrium with lactic acid. Whenever we have excess pyruvic acid, you will have excess lactic acid, and that builds up, and that's a very specific biomarker for mitochondrial disease. Again, we're using the word specific. Is it sensitive? It's hard to say, because when I see elevated lactic acid without any other reason in a patient, I think of a mitochondrial disorder. But you can have normal lactic acid values and still have a mitochondrial disease. So again, looking at the lactic acid, all these case series or retrospective chart reviews on mitochondrial disorders, mitochondrial encephalopathies, Leigh syndrome, they describe lactic acid abnormalities as a very common biochemical marker. But how common it is and how the lactic acid is drawn, how sensitive it is something that I'm not sure that we understand completely. Other biochemical markers, you can see elevated alanine, reduced citrulline in a very specific mitochondrial mutation. Then you see citric acid intermediates, oxyglutarate, succinate, fumarate, malate. Again, for all the families here, we don't have to worry about all these fancy biochemical names, but these are the intermediate molecules or compounds that come out of citric acid cycle or TCA cycle. And clinically, if I see somebody who has had maybe a borderline lactic acid in their clinical picture is concerning for mitochondrial disorder like hypotonia, or they have had developmental delays, and I see that there are intermediates of TCA cycle in acyl carnitine profile, or some of the metabolic testing that we do, serum amino acids, acyl carnitine profile, that's something that kind of like, helps me and gives me a clue to target my genetic testing toward the mitochondrial diseases. We have touched upon it many times with genetic heterogeneity of Leigh syndrome. It's well known. So we have the story of two genomes here, as one of the articles has reported it. We have mitochondrial DNA, as Dr. Mutta mentioned in his lecture. So mitochondria being an intriguing, interesting, and such an important organ that it gets to have its own DNA. And that's, how I look at it sometimes. But anyhow, mitochondrial DNA, which produces some of the mitochondrial proteins, and any dysfunction in mitochondrial DNA or any change in the spelling or any change in the instructions in mitochondrial DNA can lead to Leigh syndrome. And some nuclear genes, many nuclear genes actually by now, that we know can lead to mitochondrial dysfunction, because what they do is they make parts of this complex, intricate machinery that is present inside the mitochondria, electron transport chain. So we talked about, all these complexes because we check these complexes too. Like sometimes I send for complex 1 activity, sometimes I send for complex 4 activity. So what are these? This is kind of like a machinery that helps mitochondria build up energy. And it is connected to TCA cycle as we established. So we have complex 1, complex 2, complex 3, 4, 5, any deficiency in any of these complexes will lead to Leigh syndrome. And if we look at the distribution of genetic changes in different components of respiratory chain that can cause Leigh syndrome, we can have nuclear or mitochondrial form of complex 1, complex 2, and complex 4 is mainly autosomal recessive. But then we have complex 5, and then there are other metabolic disorders that can lead to Leigh syndrome or mitochondrial dysfunction. So if you just put in all the genes, and probably this list is not as complete as Dr. Mutta's list, but complex 1 deficiency, if we look at complex 2, 3, and 4 deficiency, there are like multiple genes that help make us part of these complexes. And whenever a part is deficient or missing and your machinery cannot function properly, you will have a defect in energy metabolism and oxidative phosphorylation. Then we have some special genes like ECHS1 or HBCH1 that are enzymes that are inside the mitochondria, and they help us metabolize some other things like valine in this case, and any disruption in valine metabolism and these intermediates collect inside the mitochondria and can cause mitochondrial toxicity. So as a clinician sitting in this time and age and looking at patients who have rare brain diseases, somebody who comes to me with a clinical phenotype that gives me clues towards Leigh syndrome. Neuroimaging findings, again, is very helpful, because once you see Leigh syndrome lesions, then you see Leigh syndrome lesions in the MRI, and then it's just a matter of finding whether we are talking about a complex deficiency, electron transport chain deficiency, or some other way where our energy metabolism is disrupted. Biochemical markers are really helpful, as we discussed earlier. Molecular testing. We have gene panels available now, so you can do targeted testing where you can send for mitochondrial DNA analysis, along with nuclear genes that are linked to energy metabolism disorder. And that when it comes back positive, then it gives you a very solid answer. Skin biopsies with fibroblast culture, and then looking at different enzyme functions in these fibroblasts can be really helpful and adds on, especially if your genetic testing is inconclusive with variant of unclear clinical significance. No proven therapies. Well, I think I'm just, I don't know how much time I have left, but I'm going to rush through these slides. And for now, I mean, again, as we have already established, there are a lot of clinical trials going on for different products, different compounds, different medications. But so far, we have not seen any proven therapies. There are no cure for mitochondrial encephalopathies in general and Leigh syndrome. Various supplements have been tried, and there is a value to trying these supplements, and we call them mitochondrial cocktail on the clinical side. So if you hear it, just know that's basically a combination of different compounds, different supplements. And as I said, there is some value while you're waiting on your genetic testing and confirmation. You can put these patients on biotin, thiamine, because there are genetic defects like biotin responsive basal ganglia disease that can look like Leigh syndrome, and it responds to biotin because there is a defect in biotin transport. And when it responds to biotin, it responds to biotin. So trying on biotin or thiamine, if there's a thiamine transporter deficiency, if we do have a specific coenzyme Q10 deficiency, you can try that specific supplement. Epi743 is a synthetic CoQ10 that has been tried in some clinical trials. Not an FDA approved drug, but something that people have talked about a lot. Riboflavin for riboflavin sensitive complex deficiency. So there are something very rare, but sometimes you can find a vitamin responsive mitochondrial encephalopathy, and it is good to diagnose those because these are treatable, and early diagnosis is the key. So mainstay of treatment for now for a lot of mitochondrial diseases is supportive care. These patients, they need feeding support. They need to be assessed for their pulmonary function. I didn't add cardiology, but they should have surveillance cardiac exams every year or 2 years based on what their cardiac health is and how their exam goes. And then therapies, PT, speech therapy, feeding therapy, occupational therapy, which helps improve overall general health of the patient, muscle health of the patient, and also helps improve communication. So that is the mainstay of the treatment again. And I think that's all I have for today. Thank you so much for your kind attention. Thank you for giving me this platform to speak. And I'm really appreciative of all the patients and families, and the resilience that you guys have is an inspiration for all of us. Thank you.
Thank you so much, Dr. Kayani. We do have a few questions. However, we only have time for one question. Performing an MRI to babies and small kids is especially challenging as they shall remain steady for a long time. This is why anesthesia is typically used. Mito kids are particularly sensitive to anesthetics. This has triggered seizures to my daughter. What is your experience with performing MRIs to Leigh syndrome kiddos?
Yes, and you're right. It's absolutely, it is tricky. We do have some guidelines for anesthesia in mitochondrial encephalopathies, but it is not complete, right? Because we talked about the genetic heterogeneity. So a patient with SERF1-related Leigh syndrome might respond differently to anesthesia than a patient who have C12 or 65 genetic defect or a mitochondrial gene defect. So I think if we do understand the genetic etiology or the underlying genetic mechanism, we have to keep that in mind. And then what I do is we convey to the anesthesiologist. I like to have these patients not be NPO for a very long time. We take precaution putting them on D10 fluids while they're getting anesthesia. And then we really have our guidance letter for the anesthesiologists to use neuroprotective anesthesias and be mindful that these patients have mitochondrial disease. And I think practicing in a center where we do have a lot of expertise in rare diseases, and our anesthesiologists are somewhat familiar. They're actually a lot familiar with mitochondrial diseases, so they are pretty good at reaching out to us to get those guidelines. One more thing that I want to say is clinically, if I do not need MRIs, I like to avoid MRIs. And what we sometimes, if we really need MRIs, what we do is we have a sequence of MRIs, which can be done within 10-15 minutes instead of like a 45-minute of MRI. And then we can, 15 minutes is still better than 45 minutes. So if we are using anesthesia, we use type of anesthesias that are, again, neuroprotective as short of a duration as possible, and avoid MRIs if we can.
Wow, that's amazing about these rapid MRIs. And thank you so much again, Saima, for your time. We appreciate all that you do for Leigh syndrome and rare diseases. I would like to ask Ethan Perlstein. He's our moderator for the next panel discussion, and he is the CEO of Perlara, whom Cure Mito works closely with. So Ethan, I'll give it away to you. Thanks.
Thanks, Casey. Hey, everybody. Let's try to jump right in here so we can try to get back some of our time. The first speaker will be Dr. Stephen Gray from UT Southwestern Medical Center, who'll actually be joining us through a video that he has shared that I will play momentarily. The title of his talk is General Overview of Nervous System Targeted Gene Therapy. So let me get his video going here.
Hello, I'm Stephen Gray. I'm an associate professor at UT Southwestern Medical Center in Dallas. And I want to give my appreciation to the Cure Mito Foundation for allowing me to give this general overview and kind of background on nervous system targeted gene therapy. Very briefly, these are my disclosures. Most notable is that I am an inventor on a gene therapy vector design for SIRF1.
Ethan, you need to share your screen.
I'll give an
video.
overview here when I'm talking about gene therapy for the nervous system. The primary vector that's being used now to do that is adeno-associated virus, or AAV. And this is an overview of the AAV virus and its use as a viral vector. It was
Ethan, we're not seeing the screen.
Discovered over 50 years ago as a contaminant.
You need to, Ethan, share your screen.
Adeno-associated virus comes from, although it's actually not related to adenovirus. Aside from that, it is a parvovirus with a very stable capsid shell, rather simple genome, and you can essentially remove all of the viral gene coding sequences out of the genome and replace it with any DNA cargo that you want it to carry, up to about 4.5 kilobases. AAV is considered a relatively safe viral vector. Some of that is owing to its natural characteristics, where it is basically a non-pathogenic virus that affects humans. There are many naturally occurring variants of this capsid that provides a sort of a toolkit of reagents for gene targeting, where some of these capsids direct to the liver, some of them direct to the muscle, some work well in the eye, others in the brain, etc. There are now hundreds of clinical trials initiated and ongoing using recombinant AAV vectors. Overall, it does have a relatively safe track record, and this led to a couple of gene therapy products now that have received full regulatory approval in US and Europe. So in terms of targeting AAV to the central nervous system, the traditional approach to target AAV vectors for the brain would have been to do a stereotaxic injection through multiple cannula inserted into the brain parenchyma. This approach has kind of mostly been supplanted. It's still used in some clinical trials, but Now the vector technology has evolved such that there are AAV vectors that have some capacity to cross the blood-brain barrier, and they can be injected IV to reach the nervous system, or else there's many ongoing clinical trials now where AAV vectors are injected intrathecally or into the cerebral ventricles. So I'm just going to talk about AAV9 because this is the vector that's being used most commonly to treat CNS diseases. And this is really exemplifies how AAV9 vectors first started getting used. If you do a tail vein injection of AAV9 into a mouse, then this is the kind of transduction pattern you'll see across the entire brain. And this is immunohistochemistry against GFP, where all the dark staining is indicating transgene expression. So this works well in a dose-responsive manner. But if you go into kind of more detail on this and look at where the vector is going across the entire body, you'll see this is the level that's going to the brain and the spinal cord, so the nervous system. But then you see that in other peripheral organs, the vector is distributing more so to those organs. And if you look at the liver and the difference in the y-axis, it's really over 100 times more vectors going to the liver as it is going to the brain. So there are approaches that are using this intravenous approach with AAV9 to treat various diseases. But I think that this distribution, even though it does translate to some extent between mice and non-human primates into humans, an obvious question was, if we're injecting AAV9, it's essentially going all over the body. Is there a way to modify the virus to get it to target more selectively to the nervous system? And there is quite a bit of ongoing work trying to do that, to try to make next generation AAV capsids that would be more efficient or more targeted to the nervous system. The other approach would be to modify the route of administration. And that would be where the intrathecal administration of the vector comes in. So, the simplest way to do this when you're injecting into the cerebral spinal fluid is to do a lumbar puncture, which can be done in animal models, can be readily done as an outpatient procedure in humans. Of course, there's other routes of administration, such as doing an intracerebral ventricular injection, or injection into the cisterna magna to enter the same CSF space. This is just a quick look at what this, visually what this looks like in mice. If you do a lumbar intrathecal injection of an AAV9 GFP vector, where you look really across the entire brain, you still get good distribution of the vector across the brain, the spinal cord. If you zoom in on this, one thing that you'll note is this is approaching a maximum feasible dose, but we're certainly not transducing every cell. We're transducing a minority of neurons and glia, but this is essentially the state of the art right now, until maybe some next generation capsids or other technical innovations come along. So and one thing to point out here is this is an intrathecal injection, so injection into the lumbar intrathecal space, but you still get vector that is distributing out to peripheral organs, including the liver, heart, muscle. But as opposed to if you inject this vector IV, the intra-CSF injections, kind of get more of a 1-to-1 ratio of nervous system tissue transduced and targeted to peripheral tissues, rather than it being skewed like 100-fold more to the liver from an IV injection. So this approach, injecting into the spinal fluid, also translates well across species. This is publication from my lab from 2013, but there's numerous publications, in the literature evaluating intra-CSF administration of AAV9 and other AAV vectors in mice, rats, pigs, dogs, non-human primates. And now this is used in a number of clinical trials. So we can view this as somewhat of a platform type of approach to try to target the nervous system, where we have a vector and we can deliver it. And so, so the design is in terms of what transgene this vector will carry and what kind of regulatory elements are built into that genome. So this is just, kind of giving an example. This is a short list of studies that my lab has initiated or that we're collaborating with other labs on to try to use this AAV9 intrathecal kind of approach to treat different diseases. And so you'll note that 7 of these have advanced and moved into first in human clinical trials. We have a number of disorders, including SIRF1, where we've generated, it's a good proof of concept in animal models. And we're working through steps now to try to move these into clinical trials, and then a number of other diseases that we think could potentially be good candidates that are kind of at the discovery stage. And there's a couple of these that I'll point out. Most of this work is in mouse models, but Krabbe disease, those studies were done in dogs, and CLN5 Batten disease, those studies were done in sheep. So if we look at clinical trials, again, viewing this as sort of a platform approach to treat neurological diseases, this is, an up-to-date list of various clinical trials being done with AAV injected into the cerebrospinal fluid. And you'll notice that most of these are using AAV9. Most of them are pediatric disorders, although there are some non-pediatric, and it's kind of a mix here where about half of these are being injected by a lumbar intrathecal injection, and half of them are being injected into the cisterna magna. And then there's a few intracerebral ventricular trials or other routes. AAV9 as a whole is being, also being administered IV in addition to some of these intra-CSF routes. And, the most notable example of this is spinal muscular atrophy, where an intravenous gene therapy trial was initiated in 2014 to treat patients under 2 years of age. And now that's become the second FDA-approved AAV product in 2019, and this is Zolgensma. And then, this AAV9 is also used for muscle diseases such as Duchenne muscular dystrophy, and mucopolysaccharidosis is another IV approach using targeting a CNS disorder. And so the question is, as far as gene therapy for mitochondrial diseases, there really aren't a lot of good examples of this out there right now in terms of using this sort of AAV platform gene therapy approach for mitochondrial disorders. Dr. Qinglong Ling in my lab did explore this for SIRF1 Leigh syndrome. We published a paper recently kind of outlining, some proof of concept studies using an AAV9 vector to deliver the SIRF1 gene to treat that form of Leigh syndrome. And, the lab is currently working through some additional steps to, with the goal to try to move this closer to an eventual human clinical trial. So, that's a kind of quick overview of gene therapy targeting the nervous system. Overall conclusions are, AAV9 and other AAV vectors administered IV or intrathecally has been validated across numerous labs for effective disease-modifying CNS gene transfer across several species, including humans. I want to emphasize here that CNS gene transfer with AAV9 still targets a minority of cells in the brain. So, I think we need to be very careful thinking about this as a cure. But what we've seen across, a variety of disease models is that level of efficiency of targeting to the brain is still, enough to provide a meaningful, significant benefit. But I also point out that, while AAV9 has been successful for a number of diseases, it's likely not efficient enough to rescue others. And also need to, make sure everybody understands that AAV gene therapy is still limited. We have to think about this as one dosing event, because the patients will get immunized against this vector after they've been treated one time, and that can preclude any future injections. But the AAV genome itself is fairly stable after you get the gene transfer. And if you're in post-mitotic cells like neurons, it should be essentially permanent. So overall, my takeaway message around this is AAV targeting for the nervous system shouldn't be viewed as a one size fits all, but it can potentially be a one size fits many. And so with that, I want to acknowledge my team works very hard in the lab every day to try to help bring these treatments forward, and give a special acknowledgement to Dr. Ling here, who's been leading all the work in my lab on gene therapy for mitochondrial disorders. So thank you very much.
All right, we're going to move right on to our next speaker. Looks like he's ready to go here. So Dr. Michał Minczuk is speaking from Pretzel Therapeutics. The title of his talk is Mitochondrial Genome Engineering in Vivo.
Hello, can everybody see my screen?
Looks good.
Yes.
Yeah, thanks very much. So thanks very much for this kind introduction, and also to invite me to give the opportunity to talk about our recent data. And although I'm involved in Pretzel Therapeutics, I'll mention that today I'm having a University of Cambridge hat on. Here are my disclosures. So before I get into the main subject of the talk, I'd like to give you a little bit of background why the mitochondrial genome is, in particular in mammalian systems, is very difficult to modify, to engineer. So at the moment, we don't have any efficient ways of delivering DNA into the organelle. So this genome is quite small. In principle, could be synthesized entirely, and then we could deliver either modified version or deliver normal version in order to correct mutations in mitochondrial DNA. But at the moment, this is not possible. CRISPR-Cas9, which is obviously a very useful to modify the nuclear genome in mammalian systems, hasn't been adopted to operate in mitochondria yet. So as a consequence, we have very few models that would precisely mimic mitochondrial DNA-associated diseases. This is contrary to the nuclear mutations that Ramsey already mentioned. However, there are some positive. There are established methods to change existing heteroplasmy, meaning the mixture of mutant and wild-type molecules within a cell. And there are emerging tools to precisely edit particular bases in the mitochondrial genome. And through the rest of my talk, I'll be concentrating on these 3 points. So the question we've been asking for over a decade now in the lab was, how can we take a sick cell which has pathogenic heteroplasmy, meaning the mixture of wild type, so green molecules here and mutant red molecules here, and can we shift them into a more favorable ratio, meaning that we'll have more wild type molecules compared to the mutant. Why would you do that? Because if we have enough wild type molecules, we'll be able to move the cell beyond the pathological threshold and basically restore its function. And we call this process heteroplasmy shift. So how do we do it? Us and others in the field came up with an idea of creating specific cuts in the mutant mitochondrial DNA. And then because the cells have pre-programmed copy numbers, so the cell sense how many copies of mitochondrial DNA should have in them, these, after cutting the mutant, the cell will repopulate itself, but mainly wild-type molecules. And the nice thing about this procedure is that you don't have to eliminate all the mutants. You have to just move it beyond the pathological threshold. Over the years, we developed several ways of cutting mitochondrial genome precisely. These tools are called programmable nucleases. So nuclease is an enzyme that cuts DNA because you can program it in order to bind any DNA sequence, hence the term. So there are zinc finger-based nucleases where you can manipulate these colorful boxes here in order to design new DNA-specific enzymes. Other nucleases involve tail DNA binders, so called mitotalens. Also, most recently, there was a paper showing that also a very old tool called meganuclease could be also adopted in order to manipulate the mitochondrial genome. So how does it look in practice? Our lab studied mainly zinc finger nucleases. So now I'm presenting an example of a mutation, 8993T2G, which is often associated with neuropathy, ataxia retinitis pigmentosa, and also if the mutant level is higher, maternal inherited Leigh syndrome. So in order to bind this mutation, we designed a pair of zinc fingers, and the red one binds specifically to the mutation. The green one here is a supporting one, and then when these two the so-called monomers come together, this is where the DNA is cut precisely in this point. In normal molecules, because the red monomer doesn't bind to the sequence which is not mutated, this cut doesn't occur. So we experimented with this system for in cells in culture. We have taken a heteroplasmic cell line, which with about 80% of mutant. This cell line have a and has a reduced oxygen consumption rate and also low OXPHOS complexes. And upon treatment and optimization of the zinc fingers, we managed to shift heteroplasmy from about 20% wild type to about 80% wild type. When we analyze these cells by proteomics analysis, so looking into steady state levels of the protein on oxidative phosphorylation system, we noticed that this cell was basically rescued. So yellow means more OXPHOS, blue is where this oxidative phosphorylation system is slower. What we also managed to do was to recover oxygen consumption, and altogether we managed to rescue this cell line. So the next important step was to try this technology in vivo. Now, as I said at the beginning, there are very few mouse models of mitochondrial disease that in particular would be heteroplasmic, so would have a mixture of wild type and mutant. However, our collaborators in Cologne developed a mouse model with a mutation in one of the tRNA genes in the mitochondrial genome. So this is sort of part of the supporting system of the oxidative phosphorylation. And in high level of heteroplasmy, this mouse model developed a rather mild but cardiomyopathy. So we decided to experiment on this animal because we didn't have any other ways of proving that our system would work in vivo. And then we designed specific zinc fingers that would bind and cut the position which was mutated in the mouse 5024, then encapsulated DNA coding for this zinc finger nuclease into a AAV vector. This was a AAV9 subtype, AAV945, which has tendency to go into the heart, so cardiotropic, and is heavily detargeted from the liver. This is where AAV viruses like going. And this is a little bit busy on the right-hand side, but what we observed that depending on the dose of AAV vector use, we managed to shift heteroplasmy in this animal. It was safe in terms of not reducing a copy number of mitochondrial genome. Also, longer treatment led to a better result. Because the mutation in this tRNA gene renders this tRNA unstable, we looked into whether stability of this molecule will be improved upon treatment. And this is indeed what we observed as well. We also, using metabolomics, which was already described, we managed to rescue a particular biomarkers of mitochondrial dysfunction. I'm showing here lactate to pyruvate ratio, which is also quite often used in the clinics. And this technology was spun out to a startup company called Pretzel Therapeutics. Pretzel launched officially last week. Please have a look on the website to read more about it if you are interested. So for the remainder of the talk, I'd like to concentrate on the following questions. So can we develop a mouse models with mtDNA mutation, which would be relevant to diseases, and then virus therapeutics could be tried preclinically, including obviously maternal inherent Leigh syndrome, relevant for this conference. So I'd like to say that at the moment, mitochondrial genome engineering is undergoing a real revolution. About 2 years ago, there was a report of using cytidine-based editing, meaning that we could take pretty much any C in the mitochondrial genome and change it into a C. And on the opposite DNA strand, this will result into G2A. This was a collaboration between University of Washington and also the Broad Institute, and VAMSI also was collaborated on this very important paper. Soon after, other types of base editors were invented. So the first ones were based on tail as DNA binding portion, but also zinc finger based and cytidine deaminases, cytidine editors were introduced as well. And this year, another type of base editors was reported, allowing for A to G mutations. However, at the moment, this tool remains a little bit less precise as compared to the cytosine editors. So basically what we want to do, we would like to take a healthy cell and then edit a particular base in DNA so that this cell will become dysfunctional due to accumulation of mutated mitochondrial genome. So quite the opposite what we've been trying to do for many, many years. Obviously, if we are very precise and efficient, this could also yield a 100% mutant, so-called homoplasmic cell lines too. These tools are also exciting because then the heteroplasmic mutations could be repaired by a cutting. However, the homoplasmic mutations, where all mtDNA molecules are mutated, were not, couldn't be repaired by cutting because there is no normal DNA. Therefore, these base editors are quite exciting for future potential precise correction of homoplasmic mutations as well. So as a part of our efforts to knock out every single gene in the mouse mitochondrial genome, I'd like to present one particular mutation which we introduced I have chosen this one because it's relevant for the Leigh syndrome. I think mutations ATP6 gene in the mitochondrial genome is the most frequent cause of the Leigh syndrome, especially in the one which is maternally inherited. So our approach here was to select a codon early on in the ATP6 gene, and it would be a tryptophan codon. And then rather than DNA coding for this particular amino acid, we converted into a stop signal, a stop codon. So from TGA to TAA by editing cytosine on the opposite strand. So after lots of tests in vitro in cells to make sure that our construct are precise and don't edit any other portions of the genome, we selected our favorite construct and then introduced it into a mouse embryo by microinjection, and then we implanted this embryo into pseudopregnant female. Our founder animal was at more or less 10% of mutation heteroplasmy. But then we could start selective breeding. So basically we could mate this female with the wild type normal male and look into the offspring and select females with the highest level of heteroplasmy. Obviously the mutation has to go through the germline. But we were lucky enough that this mutation went through the germline, and then we subjected this strain through selective breeding. And then, as you can appreciate from this graph here, in each population, in each generation, we were able to have a higher and higher level of heteroplasmy. So at the moment, we are at generation 4, and then we started to characterize this mouse. And then I'm showing here animals with these levels of heteroplasmy between 65 to 72. This is the analysis in skeletal muscle. And what we can see that the complex in which ATP6 is located, so-called ATP synthase, it's not intact. So it shows these quite typical subcomplexes which are typical for dysfunction. And these sort of subcomplexes were also found in patients affected by mutations ATP6, especially the mutations that truncate the protein. So as the next steps, and I have to admit, I'm very grateful for Cure Mito Foundation, and also Cure ATP Foundation. We'd like to follow up the studies of this animal, and then in collaboration with Stephen Gray and Alexandro Prigione, who will be our next speaker, we'd like to characterize this mouse. We'd like to apply heteroplasmy shifting technology to this mouse as well. We'd like to collaborate with Steve so that AAV therapy could be tried, and also collaborating with Alessandro so we can apply the base editing technology in induced pluripotent stem cells and organoids. And with that, I'd like to thank to all my team, especially Pedro Pinero, who's been leading the base editing program, whereas the zinc finger shifting was mainly run by Payam Gamic, but also I'd like to thank other collaborators and all the institutions for paying the bill. Thanks very much.
Thanks so much. Let's move on to our next speaker, just in the interest of time here. I do catalog the questions in the Q&A, so feel free to keep dropping them in, and we should be able to address them collectively at the end of these talks. So thank you so much, Michal. Next speaker is Dr. Jessica Federman from Boston University School of Medicine. The title of her talk is Gaining Insights into Mitochondrial Diseases Using Stem Cells.
Thank you so much. I just want to start by thanking Sophia and Casey for giving my research team the opportunity to co-host today's symposium and for the opportunity to present some of our latest research in our lab. So just to remind everyone, Vamsi already did a great job of orienting us, but there are many different organelles within the cells of our body, and there are two that are really important for my presentation today. The first is the nucleus, which is depicted in the middle in purple, and the nucleus contains a lot of the genetic information in our cells. And the other organelle of interest in my talk today are the mitochondria, as is the interest of most of us on this call today. And the mitochondria also contain genetic information in the form of the mitochondrial genome. And so just very simplistically, mitochondria take the food that we eat and convert it into a form that our cells can use called ATP. This process, much like our cars, is not completely efficient. And so some of the energy is lost in this process in the form of thermal energy or heat. And so the way that mitochondria convert the food we eat into ATP involves a process called oxidative phosphorylation. Depicted here in the middle are the oxidative phosphorylation complexes. There are 5 oxidative phosphorylation complexes, and each of these are important in catalyzing reactions that result in ATP production. Uniquely, the oxidative phosphorylation complexes are encoded by 2 genomes. They're encoded by both the mitochondrial genome as well as the nuclear genome. And genetic variants in either of these 2 genomes results in mitochondrial diseases, including Leigh syndrome. And there have been a number of different barriers to advancing our understanding of mitochondrial diseases and mitochondrial genetics in general. The rarity of carriers of very specific genetic variants has made it challenging to identify drug targets and to carry out adequately sized clinical trials. As was mentioned earlier, there are currently no approved drugs for the treatment of mitochondrial diseases. And in part, it's been particularly challenging as the therapeutic responses often differ across patients. And that's likely a result of the heterogeneity in the genetic variants that cause these diseases, as well as differences in the biochemical defects that are introduced by each of these different types of mutation or variants. In addition, the tissues that are most affected in mitochondrial diseases, especially the heart and the brain, are clinically inaccessible. So we can't easily or safely biopsy the brain or the heart of a patient to gain insights into the disease. And so we're really limited to cells that are more peripheral, such as the blood cells. A number of models have been used to study mitochondrial genetics and diseases. Some of them include animal models, such as mice, and these are really great for studying mitochondrial genes that are phylogenetically or that are conserved throughout evolution. However, the mitochondrial genome is not very well conserved, and because of interspecies differences, it makes it really challenging to use animal models to study human mitochondrial genetic defects. Another model that's been used are the cybrids. And cybrids allow us to study mitochondrial genetics in a dish. The way this works is you take a cancer cell line, such as an osteosarcoma cell line, and you deplete it of its mitochondria through either chemical or genetic manipulation. The cell with very few, if any, mitochondria left is called a row 0 cell that you then fuse with platelets from a patient. And platelets have no nucleus, so they have no nuclear genome. And so the only genetic material contributed by the platelet is the mitochondrial DNA. And so this ultimately leads to the formation of a cell that has the nuclear genome of the osteosarcoma cell line from which it originated, but the mitochondrial genetics of the platelet. And the use of cybrids has really helped us understand a lot of the basics of mitochondrial genetics, in particular heteroplasmy. But cancer cells are notorious for having metabolic and mitochondrial dysfunction that in part makes them a cancer cell. And so it's really difficult to understand how a particular mitochondrial genetic variant would alter the cell types that are most relevant to the diseases that affect patients predominantly things like the neurons and the cardiomyocytes or heart muscle cells. And so it's really time that we start to use new models to understand mitochondrial genetics and diseases. And so one of the models that our lab is using are called induced pluripotent stem cells. So induced pluripotent stem cells are derived from either a skin biopsy or from blood cells collected in a routine blood sample collection. Importantly, induced pluripotent stem cells do not require the use of embryos. So unlike embryonic stem cells, there's no ethical controversy in utilizing these cells. And of benefit to the scientific community, these cells, once you create them from a patient sample, they're indefinitely self-renewing. So we can maintain them long into the future without having to go back to the patient and recreate them. And the thing that's really important about these cells is they have the ability to become any cell type in the body under the right cell culture conditions. And so we can actually take induced pluripotent stem cells from a patient and create the cell type that makes up the tissue that's affected in that patient to better understand the disease. And so my lab has been working to take induced pluripotent stem cells and generate cardiomyocytes, or the heart muscle cells. And so pictured on your left is an immunofluorescence image of a cluster of cells in culture after having undergone directed differentiation of the stem cells to cardiomyocytes. And what we've done is we've stained the cells for cardiac troponin in green and NKX 2.5 in red and the nucleus in blue. And what you'll notice is that there are clusters of cells that are both green and red. So they are positive for both cardiac troponin and NKX 2.5. And these are two specific markers of cardiomyocytes and are telling us that we are successfully creating cardiomyocytes from the stem cells. And the really cool thing about these cells is, much like in the human heart, they actually beat in the dish in culture. And I have a video to show this. And so what you can see is in the bottom, you see the start of the contraction that then is transmitted all the way across the well of the plate. And so this really allows us to understand how genetic perturbations affect the function of these cardiomyocytes, which is reflective of what's going on in the hearts of the patients. And so we plan to use induced pluripotent stem cells to really understand mitochondrial genetics and mitochondrial diseases. And so we can do this by collecting a blood sample from patients with a mitochondrial disease. We isolate some of the cells from that blood sample, and then we reprogram those blood cells using 4 transcription factors known as the Yamanaka factors. And that generates our induced pluripotent stem cells, or iPSCs. And these cells carry the genetics of the patient themselves. And so what we can then do is we can use gene editing techniques like the 2 prior speakers discussed in order to correct the pathogenic variant, allowing us to study side by side cells from the patient that have the unedited cells that have the pathogenic variant, and the cells that the only difference is the edited correction of the pathogenic variant. We then take these cells, both the edited and unedited cells, and undergo directed differentiation to cardiomyocytes, much like what I showed you on the previous slide. The next step is deep functional characterization of these cardiomyocytes. And we can measure things like contractility and beat rate to understand cardiomyocyte function. We can look at measures of mitochondrial function, such as mitochondrial respirometry, and perform multi-omics to really deeply understand what are the abnormalities that are occurring because of the pathogenic variant. We can then take this information and identify drug screening strategies. So what we can do is we can say, okay, these are the abnormalities that we've identified. How can we target them with either existing FDA-approved drugs for other conditions or using novel and emerging drug therapies, and screen them in the patient's cells to identify the most effective therapy that would work to treat the patient from which the cells were ultimately derived. And this is really getting at creating a personalized medicine approach for every patient, where you can identify what are the specific alterations that are occurring in the cells from the patient, and how can we best target those to improve the outcomes in patients with mitochondrial disease. And if you're interested in learning more about the stem cells and how we're using them in our research program, we invite you to join us. We'll be hosting a clinic day at Boston University in the spring of 2023, where you can come see our state-of-the-art facilities for stem cell research and learn more about our research program and how you can be involved and participate in our studies. And with that, I'd just like to thank my research team as well as my many collaborators. In particular, I would like to thank Dr. Deepa Gopal, who's my MD counterpart leading our research team, as well as Jesse Morera, who is a postdoctoral fellow who created those beautiful images in the video and has really refined our protocol for directed differentiation of stem cells to cardiomyocytes. And I'd like to thank both Jesse and Karen for doing a lot of behind the scenes work today to help make it go smoothly. And with that, I will stop sharing. Thank you.
Thank you so much. I think if you're able to, Dr. Federman, there are at least one question I just saw come in the Q&A. So to all of our speakers, if you can check out the Q&A right after your talk and maybe address those questions, we can get back on track here after the break. So appreciate that. And on to our next speaker here, Dr. Alessandro Prigioni. Please go ahead and take the reins. Looks like the screen share is initiating, but not seeing any video, not seeing your presentation yet. Oh, here we go. Also, your audio, please unmute yourself, Alessandro.
Yeah, so do you see my side? Hi, everybody. Thank you very much for kind invitation. Sorry, do you see my screen?
Yes, we hear you too.
Okay. I don't see it somehow, but I can start. Sorry, I don't know what's happening.
We can see your screen. So
Okay.
maybe if you just advance the slides, even just manually with on the keyboard.
Okay, one sec. Sorry, let me try it again. Okay.
We see it now, and then I think.
Okay, perfect. Yeah, sorry, everything's fine. Yeah, thank you so much. I'm very excited to be here and also following all this great lineup of speakers. So yes, especially because in our lab we are mainly focusing on understanding and Leigh syndrome and hopefully finding therapy. So also my whole lab is connected to follow this great conference today. And also we're thankful, of course, for Casey and Cure Mito ATP6 also for the recent funding. So I'm very excited to be here. So yeah, so basically our work is focused on using induced pluripotent stem cells. We just heard a great talk from Jessica before, and we use them to make brain organoids. You see a picture of these sort of brain organoids, and these are they were called mini brain because they're small and they sort of model a brain. But now this name is discontinued because they're not actually a small brain, but they are they have some features of the brain, but they're not reproduce exactly what the brain is doing. So how are they made? So first we start, like Jessica already mentioned, we start from somatic cells. For example, in this case, we can start from fibroblast. And then we reprogram to pluripotent stem cells, and then we can differentiate to any kind of cells that we want. So in particular, brain organoids, which are these sort of aggregate of cells that contain cells of the brain. So like neurons, neuroprogenitors, astrocyte, oligodendrocyte, which are collectively known as glia cells. And then with these cells, they're not only just there, but they form organized structure. That's why it's resembling the brain. And there these can help to understand the interaction between different components, can understand specific aspects of the brain or of diseases. What is still a bit challenging are the fact that another component of the brain, microglia, which is important for the inflammatory response, is not automatically there, so it needs to be exogenously added, and also blood vasculature, which also is important for the inflammatory component or also for the response to environment of nutrients. So this is also exogenously added. So this means that it's an imperfect system, but can allow you to identify dysfunction that are primary starting in the neuronal or glial cells. So this, for example, is a picture, and you can appreciate that there's an organization. So you see here in green, these are progenitors. They organize in this neurogenic zone or embryonic-like ventricles. You can also see sort of layerings in the cortex. So there are things that can be investigated with this model. And you can look at layer formation, neuroglia interaction, region specificity, so you can make different region of the brain. And like I said, there are some challenges, but a lot of people are working on trying to improve this challenge related to vasculature, myelination, because oligodendrocyte, which makes myelin, which is this part that it's wrapping around neuronal axons. This can appear very late, so this part are still, let's say, in construction. So how do we use this model, and what do we want to achieve? So yeah, so like I said, in our lab we are mainly interested in Leigh syndrome. So we heard a lot, so I'm not gonna repeat. Just let me just stress again that there is recognized there's some specificity of the defect, like we heard the talk from Shamina, from Simon, that there's some specific defect in the basal ganglia, striatum, or midbrain. So this region seems to show some specific degeneration. We're approaching that by splitting approach, like if we follow what Vamsi was saying, so that we are generating model of a specific genetic condition. So in particular, we have a model for SURF1 and for NDFS4 now as well, and also for ATP6, which is also known as a maternal inherited Leigh syndrome. So and today I'm going to talk only about these two. So basically, for SURF1 is a nuclear gene, like I said, so that it can be approached. Well, the model that Jessica described, we can make isogenic lines. It means we can correct the mutation with CRISPR-Cas, and that's what we did. So if we generate control iPS cells, we can introduce the mutation, and from patient iPS cells, we can correct the mutations. So in this way, we can identify the specific consequence of the mutation and not of any other things that could be in the background of other genes or other confounding effect. And so we can see the SURF1 is a assembly factor of complex 4. So complex 4 activity, COX, is affected when there's mutation, whether it's in a patient iPS cells or in a control iPS cells. The important thing, the mutation has to be there. So this is to show that it's a precise model. So with this model, now we started to make these brain organoids, and we started with a more general organoid approach, which is also was defined as cerebral organoid, which is basically not region specific, but in general how the brain would form. So what we found is that when we had brain organoids carrying SURF1 mutation, whether in a control background or in a patient background, they were smaller. compared to the control organoids. And also when we look at neurons, so because this is what we can look in these organoids, we see in the control organoids that at day 40, because you need to let them grow for a long time, right? So at day 40, there are, in the control organoids, neurons are developing. At day 90, you see even more. And we see in the SURF1 organoids, this is completely disrupted. So the neurons are either not being generated or degenerated. And we can also confirm functionality that when we try to probe the functional activity of the neurons, so from these organoids, we can also grow the neurons in 2D, right? Like was also Jessica was showing, you can grow cells also in normal plates. And you can see that in the control neurons, you can see nice spiky, so the neurons can be active. But in the patient neurons, this is disrupted and there's less spiking and less postsynaptic currents. So, but now we have this model, and then we can start to understand, like I said, is it that the neurons were fine and then they start to degenerate, or they were not fine in the first place? So we look at the component of the neuroprogenitors. These they are the precursors that then give rise to neurons. And so here you see in a control organoid, is in red are the neurons, in green are the progenitors that I mentioned form these nice neurogenic areas that are organized. And also here they're very nicely organized. And in red, these are apical progenitors versus proliferative progenitors. And this again is disrupted in these organoids, so that there are progenitors you see in green, but they are not properly forming this architecture. So what we think is that the fact that we see in the neurons could come also from somehow an inability to make this neuron correctly. So we're trying now to go more into the details or to understand whether this is happening in some specific regions. So we started making midbrain organoids, which are organoids for a region of the midbrain, and they have to express a specific marker, which is TH, tyrosine hydroxylase, which is a marker of the dopaminergic neurons that reside in the midbrain. And what Carmen found is that in the control organoids, which are also bigger, we see a nice development of neurons and also of their branching. But in the patient, in the mutant midbrain organoids, we see that not only they're smaller, but also the ability of the neurons to make this projection and branching is impaired. So this seems also to be true for this specific region of the midbrain. And then we can start to understand now how this region can speak one to another. So Stephanie is making different region, cortical and striatal organoids, to assemble them. So you can try to see how they speak to each other. You can start to first make the cortical organoids and maybe label them with GFP, and then see how these labeled neurons communicate to the striatum to understand whether these projections are impaired and then would impair the communication. And then we see another experiment with a specific AAV targeting neurons, so that can we first apply it to cortical, then assemble them, and then look basically that the neurons can go to the striatum. And now we want to understand what happens when such a SORFA mutation or Leigh syndrome mutation would be present, whether this communication might be affected. So, but this altogether makes a sort of the hypothesis that when we start from pluripotent stem cells and we go to neurons, there's basically a very different demand of energy. And the cells need to adapt to that. And they adapt to that by switching to oxidative phosphorylation. So basically to mitochondrial respiration. And this is what happens in a normal scenario. There's this metabolic programming that at the level of progenitor cells, they switch to oxidative phosphorylation. When we're in a situation where this is not the case, for example, for Leigh syndrome or like I said, for SORF1 mutation in particular, these neuroprogenitor cells might not do what they're supposed to do and might not properly commit to neurons, and these neurons might not form the proper branch. So by looking at this data, we hypothesize that there are some components in Leigh syndrome that might resemble a neurodevelopmental disease. That means there's some defects that are underlying in the neuronal generation. And then, of course, there might be situation like we heard also from the case described by Leigh, that the patient might seem normal or might seem only smaller. So the children, they seem to be developing normally, but they are maybe on the lower curve of development. And then there's something that triggers something to really that cannot be go back anymore to the previous state. And we think that there's some underlying defect in how neurons are made in their let's say ability to overcome challenges, for example, inflammation or increased need of energy. And then when this challenge arrives, these progenitors and these neurons are not able to face this, and then sort of this the degeneration occurs. So, but if this is the case, can we start to target these MPCs and finding therapies? Because at the end of the day, that's what we want. Okay, we want to understand disease, but at the end, we want treatments. So we previously looked at these progenitors carrying ATP6 mutation for meals, and we found that they had mitochondrial membrane potential defect, which would then cause dysfunction of calcium homeostasis. So now we're using this as a screening target because we think that progenitors might be important and then might be important in driving the generation of neurons. So if we can find something that improve the progenitor's function, that could be important as a treatment. So we started now a new consortium called Cure Meals with several partners from Europe, and we are looking basically into this. So we have developed now several ATP6 iPS cells carrying different mutations from different patients, and we make these progenitors. And there we are carrying a large compound screening with the help of Ole Plesser, Fraunhofer, Hamburg, where they have all the automated center. And we're looking at drugs that are already either approved or safe in the clinic, but not approved for any entity. So these are sort of repurposable drugs. It's a number of drugs that can be used for another condition. And while this is ongoing, and we have several hits, but we already have one more advanced, and with this everything started with one boy that is carry a TP6 mutation that had basically this sign of Leigh syndrome and had very severe metabolic crisis. And then our clinical collaborator Markus Schulke in Berlin decided to use this FDA approved that we identify in these MPCs carrying ATP6 mutation. So since there's no animal model or not yet, and this drug is safe in children, he had a compassionate study, and this went very well. Then this boy recovered in terms of strength, ability to breathe. And so, of course, this can be anything. We heard that also when we're from BAMSID, when we heard about the curve of the disease, there's some spontaneous recovery, especially for mtDNA mutations. So we now have 4, actually 3, but then there's another 4 additional compassionate studies in Germany, and then 1 is in Italy. And all these seems to have improvement in the mainly in the basically in the preventing this further metabolic decompensation. So we see also some improve in one case of the walking distance or the fine motoric movement, but in general the patient had peripheral neuropathy. This is still there. What we see mostly is in this part related to endurance and also in sort of helping this compensation, and where we're thinking that this could act through some calcium mediated mechanisms that we are exploring. So with that, I conclude by summarizing that we think that using brain organoids from iPS cells, we can hopefully make new model system for Leigh syndrome to, to couple with available one, like for the one example, NUFS4 knockout, or the one that Michael mentioned with ATP6. And we think that using these organoids, we can understand some of the primary defects that occur into human neurons. And so maybe to understand what could be the primary things that somehow are degenerated, and then that are the ones that maybe then are important to target. So then, of course, the disease might develop a bit later because of maybe inflammation or other effect, but maybe by using this model, we can understand something that happened very early. For example, like I mentioned, this ability to make branching. And then using this system, we are also hopefully able to identify some treatments that are maybe not curing completely the disease, but maybe are ameliorating some components. And then since we use FDA approved drugs or purposeful drugs, we hope that this can be quickly repositioned to treat Leigh syndrome families. And we are basically planning this. We're in the process of planning a clinical study, although it's quite challenging, and we can discuss this. With that, I thank everybody and all of you for the attention, and of course, Cure Mito ATP6 for the recent funding that we're very excited to work with Michael and Steve on new therapeutic developments. Thank you very much.
Thank you so much, Dr. Prigioni. And again, if there are any questions in the chat, in the Q&A that you can address that are directed to you, please feel free to respond. Okay, next up is Dr. Simon Johnson from University of Washington. Title of his talk is Immune-Mediated Disease Pathogenesis in Leigh Syndrome.
All right. Hey, Ethan, can you see my slides?
Yes.
Everything looks good. Perfect. All right. Just want to quickly thank Casey and the rest of Cure Mito for inviting us to be part of this today. And of course, thank everybody who's in attendance for taking time to listen to these lectures. So I'm going to talk today about what we think in my laboratory is some sort of breakthrough work in our understanding of the pathogenesis of mitochondrial disease. This is sort of a hybrid talk for a hybrid audience. It's maybe a little bit topical for the scientists here and maybe a little bit in depth for some of the lay people. So I want to emphasize that we have some additional resources available for anybody who listens to this talk and wants to learn more. My laboratory website is just johnsonlab.science. Feel free to email me. My email address is right here. And the bulk of the work that I'm going to talk about today is published, and these papers are they recently have come out and they're open access, publicly available. So feel free to look these up, give them a deep read through at your will. All right, so my laboratory is deeply interested in elucidating the mechanisms underlying the pathogenesis of mitochondrial disease. So our real belief, kind of our mode of operation is we think that we know a lot about how mitochondria work. We know a lot about how individual proteins in the mitochondria function, but we really don't know how defects in genes that encode mitochondrial proteins actually lead to the complex syndromes that we see in human patients. We work in a lot of models, but the one I'm going to talk about today, the one we spend most of our time in, is Leigh syndrome. This is, as you know, as you've heard about through multiple talks today, the most common pediatric form of mitochondrial disease. It's early onset, typically around 2 years of age, symptoms begin to show. It's a multi-organ syndrome. It includes muscle weakness, lactic acidosis, respiratory failure, seizures, and a lot of other symptoms. The real hallmark symptom of Leigh syndrome, however, is the appearance of symmetric lesions in the brain stem and other parts of the brain. Cortex is generally spared, and importantly, lactate is often visible in MRI, MRS imaging of these lesions. There are no clinical interventions available yet. We study Leigh syndrome largely using a mouse model called the NUFS4 knockout mouse. This mouse closely resembles human Leigh syndrome, including the causal defect. So these mice have a defect in the same gene that can cause human Leigh syndrome. It's not, of course, the only gene that can cause Leigh syndrome, but it's one of the genes that can cause Leigh syndrome. As in humans, these mice are born normal, overtly normal without disease. They have a postnatal disease onset, rapid disease progression, and they show all of these same symptoms, including critically these symmetric CNS lesions, and they occur in the same exact parts of the brain in the mouse as they do in humans. This is just a quick picture to show you how we look at these lesions in a mouse brain. This is a part of the brain stem that we've stained with antibodies that mark inflammatory cells, microglia and macrophages. Those are in red here, and astrocytes. These are a supportive cell in the brain. They also respond to damage. These are in green, and you see this really huge neuroinflammatory lesion that we would typically describe as having astrocytosis and microgliosis because of the accumulation of those two cell types. So in this mouse model, progression is very rapid. As I mentioned, they're born healthy and normal. They have a sort of transient hair loss phenotype, which isn't really related to disease. And around P35, postnatal day 35, when they reach sexual maturation is when disease sets in. Rapidly after that age, they begin to display weight loss, which we call cachexia, clasping, ataxia, and circling. These are all just signs, visual signs of neurological dysfunction. And mortality in this model without intervention is by about 80 days. You can kind of think of the lifespan of this animal as two phases: no overt disease before P37, rapidly progressive neurodegenerative disease after that age. So the work I'm going to talk about today is follow-up studies to a body of work that we did back in 2013, 2014, where we found that inhibition of a particular set of proteins, which together are called mTOR, significantly extended survival in this mouse model of Leigh syndrome. It wasn't a full rescue of disease. However, at the time, it was the first small molecule drug ever shown to significantly attenuate disease in mitochondrial disease animal model. So it was quite exciting. mTOR, just a real quick intro to it, it stands for the mechanistic target of rapamycin. It's a set of proteins, a very large set of proteins that form a complex inside the cell, and they act as a hub for signaling. So they get cues from all sorts of different inputs, and they synthesize them and regulate a bunch of outputs, as you can see in this really simplified diagram here. Really what they're regulating is cues for appropriate environment for growth and proliferation for individual cells, and regulating the processes that drive proliferation versus maintenance. Thinking of mTOR in kind of the cellular context, it lies inside the cell. A lot of the signals that feed into mTOR arise from within the cell, such as amino acids, energy level, favorable conditions, but a lot also come through at the cell membrane through extracellular signaling. Some forms of signaling include insulin signaling, growth hormone signaling, things like that. So these are factors that bind something at the cell receptor, induce signaling inside, which activates mTOR. So again, we found that blocking mTOR rescued disease, but at the time we didn't know what the mechanism was. So why have we been going after this for the past nearly a decade? Well, our rationale has been that if we can determine the role of mTOR in these findings, how is it that mTOR inhibition is rescuing disease? Perhaps we can uncover some more basic mechanisms of the disease itself and maybe identify some more potent targets for intervention. So, as I mentioned, this is something we've been working on and off for nearly a decade. Our lab, as well as a number of other laboratories. And what ourselves and a number of other groups have kind of been doing is over the past few years is we've been looking downstream of mTOR and systematically looking at all the different things that mTOR regulates and kind of testing them all to see which one of them might be responsible for the benefits we saw. And after all this work, the overall result that we came to, the overall conclusion was that it didn't seem like any one of these pathways was by itself responsible for the benefits of mTOR inhibition. Some of them provided some very, very modest benefits, but nothing was really profound. So recently we started thinking about this a little bit differently. We decided to think upstream. What if the role of mTOR is not defined by what it's doing, what's downstream of it, but rather by what is activating it upstream? What if one of these inputs going into this complex of mTOR defines the role of mTOR in the setting of mitochondrial disease? We tested a number of these, and I'm going to kind of skip through all that to hone in on the one that became interesting, which is signaling through the cell membrane bound receptors, signaling through receptor tyrosine kinases in particular, which signal through PI3 kinase. So it's not important to remember all of the names of these players. Just remember that the cell membrane has receptors in it, and factors from the outside can bind to them and induce signaling on the inside. And through this particular set of pathways, through PI3 kinase and receptor tyrosine kinases, PI3 kinase mediates signaling to mTOR through those signaling factors. So when we decided to look at PI3 kinase, we initially realized that there are really 4 forms of PI3 kinase itself. They're defined by the catalytic subunit. There's an alpha, a beta, a delta, and a gamma. And fortunately, there are highly selective pharmacologic agents for each of these. So we can treat animals with mice with a drug that targets just one of these really specifically and see what happens. When we did those experiments, we found that the alpha, beta, and delta PI3 kinase forms, inhibition of them essentially did very little to nothing in terms of disease progression in these animals, whereas inhibition of the PI3K gamma isoform was extremely promising. In fact, the benefits that we observed were on par with what we saw with mTOR inhibition with rapamycin. And you can see in this lifespan curve here of animals treated with the PI3K gamma drug that it overlays quite well with the mTOR inhibitor rapamycin. We also saw a rescue of disease in the animals, and these are just 2 of the kind of measures of disease state that we looked at. One of them is performance on a rotarod assay. This is just an overall measure of kind of coordination and muscle capacity. The PI3K gamma inhibitor treated animals performed better for much longer, including past the age when untreated animals would have all been euthanized. And cachexia, or weight loss, you can see there's a significant delay in the onset of weight loss in these animals as well. We also saw similar delays in clasping, circling ataxia. You can pull up the paper to look at those if you'd like. So what became immediately interesting to us was that this isoform, the PI3K gamma isoform, what's known about it is that it's really kind of specific to the immune system, and it's involved in leukocyte activation and proliferation, whereas the other factors are involved in other types of signaling. The question that comes out of that observation is, what if mTOR inhibition is simply rescuing disease through impairing leukocyte proliferation? If this is the case, would targeting the immune system directly provide an even greater benefit? And this is getting into kind of the take-home message of the talk, which is that the evidence was starting to suggest that Leigh syndrome may be an immune-mediated disease. So to test this directly, we decided to treat animals with a drug that would hit leukocytes very potently and specifically, and we picked the drug pexidartinib. This is a pan-leukocyte inhibitor. It depletes all cells that are CSF1 receptor dependent. This is a particular immune cell receptor at the membrane. We could talk for hours about CSF1 receptor and its complexities. Just know that it depletes immune cells, that it depletes leukocytes, in particular macrophages and microglia. What we found was that treatment with this drug fully suppressed the CNS lesions in this animal model. Here on this slide, you can see on the left, an untreated mouse at P78 towards the maximum lifespan, a very large lesion in the brainstem. On the right here, a pexidartinib treated animal at nearly 200 days, complete absence of lesions. We also saw a rescue of neuroinflammation in the cortex, which hadn't previously been reported in this model. with treatment with pexidartinib. And this was a rescue of both the microgliosis, which I mentioned, and the astrocytosis. So both forms of neuroinflammation were prevented by treatment with pexidartinib, fully suppressed. We looked at a number of functional outcomes of brain activity. This is one example. This is whole body plethysmography, which looks at the ability of the brain to regulate breathing pattern. This is controlled by the part of the brainstem that degenerates in Leigh syndrome. Treatment with pexidartinib fully suppressed respiratory dysfunction in this model. It prevented gasping phenotype and allowed these mice to breathe normally and respond to hypercarboxic stresses completely normally. We also saw a rescue of exercise tolerance and ability on the rotarod, as previously mentioned, prevention of ataxia onset, and at least at an early age, P30, we also seem to see a rescuing of seizures, at least in this sort of low stress model, exercise-induced seizure model that we use. This may not carry over to other more kind of severe epileptogenic stimuli, but at least for this kind of mild stimulus for seizures, pexidartinib treatment seemed to prevent seizures. Strikingly and sort of unexpectedly, in addition to all these rescues of neurological dysfunction, we saw prevention of a number of signs of metabolic dysfunction as well. So cachexia, weight loss, was prevented, progressive hypoglycemia was prevented, and increased blood lactate levels in response to either a glucose stimulus or anesthesia. In both cases, treatment with pexidartinib, this leukocyte inhibiting drug, prevented them. And coming up on the end here, just to show you a quick video, because it's always more valuable than words. On the left, you can see about a 50-day-old end-of-spore knockout untreated, severely ataxic, uncoordinated. On the right, you can see some 150-day-ish old pexidartinib-treated knockouts. The one real off-target effect of this drug is that you can see here is that it turns your hair white. It does the same thing in both mice and humans. So these are the same type of mice, but they've been on this drug for a while, so their hair and coat has turned white. Lastly, we saw a substantial rescue of survival. This seems to be limited only by toxicity of the drug, as control animals treated with the same dose were dying at about the same age as the end-of-spore knockout disease model treated with this drug. And you can see that in the cause of death as well. The majority of animals that are untreated are euthanized due to weight loss or severe neurological signs, whereas the pexidartinib treated were all dying of unknown cause without any severe disease present. So we think we found the right target for treatment. However, this particular drug is toxic, and so we need to find a better target, a better agent. So just to give you some quick conclusions, this data suggests that at least a subset of symptoms in Leigh syndrome, the ones that we looked at, appear to be immune mediated. Disease might be attenuated with, or disease can be, as we've shown, attenuated without targeting ROS, energetic redox metabolism, or mitochondrial function at all. And this data really suggests that determining the key immune pathways involved might yield novel intervention strategies, perhaps more potent strategies and less toxic strategies. So just like to thank my lab really quick. Thank again the organizers for inviting me to talk. I'm happy to take questions when we get to the discussion period.
Thank you so much, Dr. Johnson. Great talk. And rounding us out here, Dr. Joe Bellucci from Rarebase. The title of his talk is Applying a Precision Medicine Drug Discovery Platform to SURF1 Leigh Syndrome.
Okay, thanks, Ethan. Hopefully you all can see my slides and a picture of our lab at Rarebase in Palo Alto.
Looks like it's still loading, perhaps.
Give it a second. Let me try it again. It says it's paused, so.
Yeah, it looks like the same thing happened 2 talks ago. I'm not sure how that was corrected, but maybe. Let's see.
Yeah, still saying pause, which I've never seen before.
Can you try it again now?
Still showing paused.
Yeah, not sure what the AV issue is here, unless you're prepared to do a whiteboarding session here. Maybe logging in and out. I don't know if it's one of those things, or maybe it's if you have a split screen, or it's.
Try one more thing.
Joe, did you put your talk into Dropbox?
There it is. We see it
It's
now.
working now. Yeah. Okay. Okay. So now hopefully you can see a picture of our lab. But yeah, thank you to the organizers for inviting me to speak today. First of all the talks have been amazingly interesting, and we're very excited to share some of the work that we've been doing at RareBase as it applies to SIRF1 Leigh syndrome and our drug discovery platform for precision medicine. So I wanted to talk a little bit first about RareBase's general approach, and then move into how this approach is being applied to Leigh syndrome and SIRF1 specifically. So we're taking really a multi-pronged approach to identify drug candidates for rare diseases. And we're primarily focused on trying to repurpose all of the 2,700 FDA-approved drugs across rare diseases, not just Leigh syndrome, but across the full spectrum of genetically defined rare disorders. To do that, we're developing data sets in-house, platforms, and facilities to support this. And the hope is that by looking at the connections between rare diseases, that we can find unexpected things, increase the efficiency of research, and ultimately bring effective therapeutics to patients across disorders. And RareBase really, I think, provides a path to move drugs to patients as quickly as possible in an efficient way, and really in a way that generates decision quality data that can be applied to the clinic. So what we're doing from a process standpoint is to take patient-derived or genetically engineered cells that have the background and the right context for studying a disorder. And then we're looking in our in-house database, which we call Function. And this is really a database of drug screening data and a variety of other data sources that we're generating that enables us to computationally predict candidate drugs that may be effective for a specific disorder or a specific genetic variant. And that's where the disease context of the patient-derived or the engineered cells becomes really important, because we take those cells and test the drug predictions in those cellular models of disease to generate, like I said, decision quality data that can be used in a potential clinical application. And really an important thing to emphasize, and I think this is what motivates all of us at RareBase, is that our discovery research approach begins and ends with the patient in mind. Our goal is to find effective treatments for people, and we're aiming to do that in a way that's efficient, fast, thorough, and collaborative. So we're very open to working with not only other companies, but other research groups, as well as individual families and patient advocacy organizations to get to this ultimate goal of bringing effective therapies to patients. And just to go into a little bit more detail about our drug prediction database, if you will, which we're calling Function. This is really the engine for our entire operation, and it rests and relies on three main pillars. The first is insights from those disease-relevant cell models that allows us to really measure the underlying biology in a very thorough and disease specific way using a variety of experimental methods. A second pillar, as it relates to one of those experimental methods, is insights that we're gaining from drug screens. And this is what I'll talk about in a lot of detail in the following slides. But we're generating really rich data sets across the druggable genome and using that to identify novel opportunities for drug repurposing and for novel drugs. And then finally, the third pillar of function is really insights from computational analyses. So many of the methods that we're using are generating really large data sets that require kind of next generation computational techniques to both aggregate the data and then to synthesize it in order to make those drug predictions as efficient as possible. And the combination of all these different sources of data and techniques is really what we think will maximize our ability to find effective drugs. So on the drug screening side, because this is kind of the main experimental approach that we leverage, this is really producing a set of rich transcriptomic data that enables our drug candidate prioritization. The process for the drug screening that we're doing is shown in this bubble, in this flow chart. Starting at the left side, we take iPSCs, which you've heard a lot about from previous speakers, and differentiate those into neurons. And then we treat them with a compound library. So our initial library is about 4,000 different compounds. Many of those are FDA approved. And the neurons that we're using for the screen here are derived from an iPSC line that comes from a presumably healthy volunteer. So there's no sort of disorder specificity here. We're testing these drugs on essentially healthy human neurons. We do the drug testing all in parallel. So it uses these plates that many people on the call may not be familiar with, but they're 3D4 well plates where each well is getting a specific drug, and we're treating neurons in that well with that drug. Each of these wells is about the size of the tip of a pencil, like a pencil lead. So very small, and we're able to run this with automation in a very high throughput way to be able to screen a lot of compounds in parallel. One differentiating feature of our approach is that we're not just interested in a specific gene's readout, but we're actually measuring essentially all genes in these cells and how they change in response to each drug. And we're using a technique to do this called 3'N pooled RNA-seq. And essentially, this is where kind of the power of our approach comes into play, because we can take the data that's generated and query it for a specific gene and start to identify which compounds either increase or decrease the expression of that specific gene. And we can do that across essentially all genes that can be measured in these cells. And we're reliably able to measure around 10,000 different genes at this point in these cells. The data that this is generating is really massive, I should point out. Compressed so far from this library, the compressed data set is around 30 terabytes. And just to put that in perspective, if you were to move all that data onto, audio CDs, it would be about 50,000 audio CDs just from this initial set of 4,000 compounds that we've screened. So that's where the computational analysis of this data becomes really important, because we're really dealing with truly big data sets. So I'd like to share our preliminary results for SURF1. And I'll talk about this plot in a little bit of detail in a second, but I think it's important to focus on the key takeaways first. So of those 4,000 drugs that I keep mentioning, we've screened about 2,800 of them so far, and this includes a large number of FDA-approved drugs. We're also in the process of sort of finishing out that set and have data coming in on 1,200 additional compounds, which are not shown on the plot at left. But we are identifying preliminary candidates that are able to change SURF1's gene expression. An important caveat here is that each drug in the screen has only been tested at a single concentration, and the effects of any of these candidates need to be confirmed in follow-up studies, which we're planning to do. An important feature of that is to look at a very thorough dose response characterization of each of the candidate compounds in a patient-derived cell line rather than in healthy neurons or in PCs. And so the goal here with SURF1, I also want to point out, is to be able to boost the expression of copies of SURF1 that have missense mutations that have presumably some residual activity. This strategy clearly won't work for cases where there are truncating mutations in both copies of the gene. That would essentially be like if you have a car that has a flat tire, we can put air into it, and that's sort of the missense mutation case, and the car can drive more efficiently. In the truncation case, where both copies are broken, that would be like a car with no wheel. Putting air into it isn't going to make much of a difference. So it's really for this specific use case. I should point that out. The plot is showing our initial set of results. The horizontal or x-axis is showing the fold change in SURF1 gene expression, and each point on the plot represents the response of SURF1 expression to a specific drug or chemical compound. The FDA-approved drugs are highlighted in green. Non-FDA approved drugs, these are generally clinical stage candidates, but there are some preclinical candidates as well. Those are highlighted in pink. And we are also looking for whether these any of these compounds negatively affect cell viability, and those are denoted in gray, and those would be sort of excluded from further follow-up because they're essentially toxic to these neurons. The y-axis for the vertical is showing how confident we are in each of these results. This is the minus log 10 of the p-value for those that are more technically focused. So essentially being higher on the vertical axis indicates that we have more confidence in the ability of a specific drug to move SURF1's gene expression up. And so we're very excited about these results and are planning, follow-up studies on things that are increasing SURF1, moving it to the right in this plot, and also that we have good confidence in. And I do want to present some interesting sort of aggregated analysis that we're starting to do on this data, where we've identified 2 potential classes of therapeutics that may be able to increase SURF1's expression. So the first class, target class A here, is highlighted in pink. There's actually 7 different molecules that are all of the same class that we've identified in these results that are able to significantly increase SURF1's expression. 3 of those are FDA-approved drugs and 4 are clinical stage. So this is really exciting because this is essentially reinforcing that this target may be valid as a therapeutic option. There is also another class, target class B, which is highlighted in green. One of these compounds is a tool compound where it has known toxicity, so it's not actually being clinically developed, but the other 2 are clinical stage drugs. And just to focus on that target class B here in this bubble on the top right, this is sort of trying to show an exciting feature of these 3 drug candidates. One of them is a known activator of a protein that leads to SURF1's gene's expression increasing. The other 2 candidates are actually inhibitors of an inhibitor of that SURF1 activator. So we're seeing candidates that are popping up across different protein targets of the same pathway. And we're also seeing for target class A compounds that are hitting the same target. So this is really exciting across both of these target classes. We're obviously hoping to move these forward into additional testing. And that kind of leads to the key takeaway, which is that we're seeing multiple candidate molecules with these overlapping mechanisms of action. And this is, motivating our additional characterization and development efforts. So future directions for this program are that we've generated some patient-derived iPSCs. We're hoping to generate more in the future to be able to test these drug candidates across a variety of genetic backgrounds that are specific to multiple patients. We're in the process of generating neural progenitor cells and neurons, depending on how easily those can be differentiated, which, previous talks have discussed the challenges of doing that, but we're hoping to at least get to the NPC stage and then to test these candidate drugs in these confirmatory studies that would look at not only SURF1's gene expression, but also start to assess functional improvement in neurons or NPCs via high content microscopy, which we have up and running in our facility, as well as gene-specific functional assays like mitochondrial fitness assays. Ultimately, we hope that this panel of data can be summarized for physicians and used for potential clinical application through either repurposing of FDA-approved drugs or continued development for drugs that are not FDA approved. And just to sort of highlight one of the functional assays that we're hoping to use and that we've stood up through a partner that we're working with, we are looking at mitochondrial oxygen consumption in SIRF1 affected cells. This is sort of a standard assay that many people may be familiar with that runs on Agilent's Seahorse platform. And this essentially measures how healthy the mitochondria of cells are in response to various stressors. And I think the key takeaway, to not really belabor this in the interest of time, is that we've stood this assay up with SIRF1 patient-derived fibroblasts and look at and have been able to look at an unaffected control relative to those cells, and do see a significant difference in their basal level of respiration between the two cell types. So this is the type of thing that we're thinking about as a corollary to our drug screening efforts that are really focused on gene expression and microscopy. The functional assays, I think, are a key additional layer to that hopefully inspires clinical translatability of these compounds going forward. So with that, I'd like to thank not only the Cure Mito Foundation and the organizers of this amazing conference, but also the entire team at Rarebase. We're about 25 in Palo Alto, and it's a great cross-functional team. Everyone has contributed to this work and the work that's being done across the variety of rare diseases that we're working on. So with that, I'll end, and happy to take any questions in the follow-up session.
Great, thank you so much. I think we've cut into the break now, so I think it's probably wise to give people a chance to stretch their legs here, or at least enjoy a little bit of a break before we dive into the next session. So I think most of the questions actually were answered in the Q&A. I think maybe, Joe, there's a few questions in there for you. Maybe if you can follow up in that chat in the Q&A, or maybe leave your email for folks to follow up with specific questions to you. And then I'll just pass it back to Casey or Sophia to take us into the break here.
All right. What a great first panel discussion. Awesome. I know we're running a little bit behind schedule, so we will just we'll reconvene here in 10 minutes. See you soon.
Kevin, can you see my screen?
Yes, I can.
Okay, great.
And I'm actually seeing like all of your screens. The presentation screen and your speaker notes screen.
Oh, okay. I need to swap them. Thank you.
And now I'm just seeing your desktop. So
Yeah. Confused about which screen to use. Why is it? Oh, just a minute. Yeah, I've did this before.
welcome back, everybody. My name is Kevin Fryert. I run a small company called Salem Oaks, which I started right after I finished my career at Pfizer after 30 years doing R&D in many different roles. Salem Oaks is setting out to empower patients to shape the future of medicine by educating them, preparing them to talk about drug discovery and development. I also sit on the board of Rare New England, and I'm on the advisory board for the Cure Mito registry. And I really enjoy this morning's discussions, and it's exciting to see all this work going on. we've heard a lot about the preclinical work, the basic science, learning about the mechanisms, all the different avenues people are looking at for Leigh syndrome. And now we're going to shift gears a little bit into how do we actually discover drugs for that and develop those drugs and then push those drugs through the clinic so that we can understand which are the ones that we should be moving forward. Today, I'm joined by Dr. Ann Fee, who's the Vice President of Biology at Cytokinetics, Dr. Matthew Klein, who's the COO at PTC Therapeutics, and Dr. Volkmar Weissig, who's a professor of pharmacology and biologic sciences at Midwestern University in Glendale, Arizona. So Ann, why don't you get us started?
Okay, you see the full screen?
I do.
Okay, great. Thank you so much to the organizers for inviting me. It's so wonderful to be amongst these very esteemed researchers doing very exciting research that are, making significant progress toward finding treatments for these diseases. My job here, though, is to sort of give you an overview of the drug discovery process. So it is primarily targeted toward the patients and families that are tuning in today, to sort of give you an overview of what this is like and the some of the challenges that we face when we try to develop drugs for mitochondrial disease. So I will there we go. So I don't want to go over all the details of this. What we've talked about a little bit today, what we talked about this morning was a good bit about gene therapy and the approaches that are being taken to bring gene therapy to the clinical sites to treat these patients. As you could hear, this is a fairly long and involved process to make sure that it's not only going to be effective, but also safe. The other topic that we've talked about a little bit in the previous few talks was using what we call small molecule therapy, or the conventional concept of a drug, which is a small molecule. Aspirin was the first drug to be discovered back in the 1890s. And what you can see here is the progression of the modes of therapies that have been developed over a century and how it is progressing. And of course, gene therapy is down here on this list as it's come basically to the drug marketplace only fairly recently in the 1990s, but it holds a lot of promise for patients with mitochondrial disease. So what I'm going to talk about and focus on in this talk is sort of the process of this more conventional form of the discovery of small molecule drug therapies. So here it is sort of depicted the entire process, and that is we start with an idea or a target. For instance, in Dr. Mutha's example that he gave, he did some screening to identify genes that allowed cells to escape a mitochondrial defect. And those genes then told him that one approach to treating this disease could be, for instance, lowering the oxygen tension, which is then going to enhance the ability of the cell to make ATP, which is so critical to organismal function, by the glycolytic path and not be so reliant on mitochondrial ATP production. So that process of identifying a target can be a multi-year process. So the target discovery and followed by some initial compound discovery. What you just heard in the previous talk was taking a cell model system, throwing onto a high throughput plate a whole bunch of compounds and looking for how it changed gene expression. And so that's an aspect of compound discovery, looking for exactly what the effect of the drug is going to be and could it be beneficial in the model that you're most interested in. So all that process takes 2 to 3 years, sometimes longer. Then we move on to non-clinical studies, and this period can take 3 to 5 years. Non-clinical studies means basically moving that compound that you have identified, usually in a cell or even a biochemical assay, and moving it into animals and finding whether the drug is effective or not. And I'll touch on some of the details of that in just a moment. But I'll sort of finish out the process here of then going through clinical studies, which is probably one of the longest of those phases. You just heard 2 talks that are potentially interested in using already FDA-approved drugs and repurposing them for use in these patients. And to do so then means cutting out this entire long period of clinical studies that are required to get to bring a drug to market. Takes about a year after a compound has been through clinical studies, and if it's successful, the FDA is going to make a decision that takes about a year. If you get approval, then you can launch, means take your compound to the drug market, and then it'll start be used in patients. And it's imperative that whoever the company is that developed the drug continue to monitor in those patients that are using the drug for safety and efficacy as time goes on. So, but what I want to do here is then explain, because this whole process of the laboratory and animal early kind of discovery studies can be broken down into additional steps. I talked about target identification, validation, and you got to develop an assay, some of which were described. The assay in the last talk was looking at how compounds change gene expression, and that was the assay format. And you've got, that needs to be high throughput. So he was describing they do those in 384 well plates. They've made major leaps and bounds in the huge amounts of data that are generated from that kind of experiment to pull through that to get to condensed information that's going to tell you whether your compound was effective in the direction that you wanted it to be. So when you find a compound, say it's not an FDA approved drug, then you're going to keep iterating through a process of taking that drug into lead optimization. And lead optimization means you've got to understand how the body metabolizes that drug. Our livers and other organs in our body will take a drug that you've ingested and they will change it. And it can be changed to be better, or sometimes can be changed to be toxic. And so we want to make sure that you can modify the molecule in a way that's going to get around that metabolism. You've got to modify that molecule in order to optimize the half-life in the bloodstream. Some drugs, reach the bloodstream or only high in the bloodstream for an hour or 2, and that might be enough to be beneficial for the patient. For some other indications, you need a level of drug in the bloodstream to persist for many hours in order to have efficacy. There are all sorts of other aspects, solubility. You've got to be able to get a drug into solution when it's in the bloodstream. If it falls out of solution and precipitates, it's going to cause major problems. And so there are all sorts of chemical characteristics about a small molecule that have to be optimized. And so the medicinal chemists take the data that's generated in all these assays, they look at the compound, they make some additional compounds, tweaking the structure of the compound and the physical chemical properties of the compound. And then you reiterate back through the process, back through the screening assay, and back through animal studies. And those animal studies, as you've already heard today, are very often mice, sometimes rats. And then from that point, you'll do toxicity studies, which are also done in small molecules, but the Food and Drug Administration requires you to do toxicity studies in larger animals as well, once you have a lead compound that you have confidence in. And so at that point, then you can file an application with the FDA for what's called an investigational new drug or an IND application. Once you have that IND application approved by the FDA, you can move to clinical studies. And here are the different phases of clinical studies that I think many of you have already heard about, and I thought I'd detail some of the aspects of these studies that are inherent to this process. In all of these studies, one of the primary metric is how safe is this compound. If it's never been in human beings before, the utmost importance before it goes on to be tested in a large population is that it's safe. So a phase 1 study is exactly that. It's a safety study, but you're also aiming to figure out what dose is going to be appropriate. You're going to try to figure out whether it's tolerated. Does it very often drugs will create either some nausea or a headache. Sometimes even though that's not life-threatening or health-threatening, it certainly makes patients uncomfortable, and they may not tolerate it and drop out of the study. These phase 1 studies are usually quite small. It's more likely to be 20 to 30 patients big. This 100 would be on the high end, and usually it is healthy volunteers. Not always, but sometimes it is, or most of the time it is healthy volunteers. It takes about several months. And about 70%, because of all those studies you've already done in small and larger animals, 70% of those show that they're safe and can move on to phase 2. And a phase 2 trial is then testing for efficacy. This is where you begin testing it in the diseased patients that you're intending to treat. And again, you're looking for safety, and then you're now also looking for, does it help the disease symptoms? You're also sometimes optimizing the dose. It may involve more than one dose in a phase 2 study, looking for side effects. And you've gone from 20 or 30 patients now to hundreds of patients. This may take months to years, and part of this has to do with the rate of progression of a disease. So for instance, in Alzheimer's disease, the rate of progression and loss of cognitive function is very, very slow. So in order to conduct a successful trial, that trial has to go on for many years because you can't detect a tiny change in cognitive function. The drop in cognitive function has to be big enough in the general population of the patients to be able to see a statistically significant difference as a result of drug administration. About only 33% of drugs move from phase 2 to phase 3. Then in phase 3, these are the big studies. They can take 300 to 3,000. My company ran a heart failure phase 3 trial in the past that was over 8,000 patients. And the reason to have so many patients is because you want your statistical power to detect an effect of the drug to be very high. And the more patients, the higher the chance that you're going to be able to detect an effect. Again, you're monitoring for safety. These studies can be long, again, based on the rate of progression of the disease. And only about 50% of those that make it to phase 3 go on to the FDA for approval. If you have a successful phase 3 trial, then the filing that you apply for with the FDA is fairly highly likely to be approved. So that new drug application that takes approximately a year to prepare and a year for the FDA to review, then is fairly likely to be accepted. And that's when you can then be taking your drug into the marketplace. But I need to point out, and this is part of the challenges in drug discovery, is that starting from the point of clinical studies, the likelihood of success to getting your drug to patients is only about 10%. There is a huge fallout due to safety, due to off-target effects that you didn't anticipate. I'm sorry, but mice and rats are not humans. And so you will begin sometimes to see toxic effects in human beings that did not show up in smaller species. And so this sort of summarizes some of the challenges in basic drug discovery. But I think that there are, yes.
And
Yeah.
we're seeing a little reminder screen down the corner of your screen.
Oh,
Can you
sorry.
turn that off? It's covering up some of your slides.
Yes.
Thank you.
Sorry, I can't. Put my cursor to move in the right direction. Sorry.
I guess don't worry about it because
No, I know. We had this
the
problem with
cursor's
the
not
earlier
helping
one,
you.
and I can't get it to disappear because I can't get the pointer. Excuse me, just a moment. All right. There we go. Okay, so how does this relate to then mitochondrial disease? So what I wanted to point out is that in a subset of diseases, no, it won't go. In a subset of diseases, for mitochondrial disease and neurological disease, there are basically only 2 drugs that are approved for treatment of the disease. And one is, both are actually ROS reactive oxygen species scavengers. The rest of these that are listed for metabolic disease and cardiovascular disease are also FDA approved drugs, but they may or may not relate to what we need to use to treat mitochondrial disease, but they do modify metabolism in ways that could be considered for repurposing. But only some of these would be used for that purpose. So why are there so few diseases, so few drugs approved for these diseases? The total number of FDA approved drugs is about 20,000. There are approximately 50 new drug approvals per year. And like I said before, approximately 10% of those that enter clinical trials actually become approved drugs. So why is that? Why is it so challenging? Well, I think we've heard some of those challenges in the talks that we've gone over this morning. And that is, there is a huge complexity in mitochondrial disease, in that there are 2 genomes involved in creating functional mitochondria. And so one of those is, and this may be particularly for gene therapy for mitochondrial DNA encoded mutations, that is the biggest complexity that has kept us from moving forward. But as Dr. Minjig detailed this morning, major progress is being made there. For nuclear encoded mutations, gene therapy is actually significantly easier. And there, as was detailed earlier, there are some success stories related to other monogenic diseases where there are FDA approved gene therapies. And that we all anticipate will continue to grow for the nuclear encoded mitochondrial diseases. Other challenges include the ability to design and execute robust clinical trials. This is one of the reasons why the registry that is open to these patients is so important for all the families to participate in, because what that does is allows those companies and the NIH, et cetera, that want to run a clinical trial to know where those patients are and to know what their clinical presentation is, so that they can identify the patient population that could enter a clinical trial. Numbers are an issue for those. Obviously, there are ATP6 mutations and SIRF1 mutations that are more prevalent than others. For those that are quite rare, a clinical trial may be quite challenging in that if it's a gene modification trial, it's going to be very specific for that mutation. And so the number of patients that are available to conduct that study is critical to the success of the trial. We've also talked about, Dr. Mutha and others have brought up the topic of biomarkers. We can't sample the brains of these patients, obviously. So you would prefer to have either a urine or a blood-borne marker of improved function. And I think that as time goes on, we're going to come up through efforts like what Dr. Mutha was describing and doing proteomic profiling of blood samples, metabolomic profiling of blood samples to identify biomarkers, ready biomarkers. As we heard, the MRIs are great for diagnosis. They would be difficult, in particular for young children, as a monitor of disease status. Another big component that has, I think, hampered development of drugs for these diseases is risk aversion. And the good news is we see, particularly in the last couple of years, we see significant progress in overcoming the fear of the risk of failure. Big pharma companies, for instance, like Astellas, have acquired mitochondrial-centric companies like MitoBridge. So there is an appreciation in big pharmaceutical companies that obviously have huge resources, that going after mitochondrial defects is worth the investment. And that also has been enabled by changes at the FDA for how trials can be designed for rare diseases and what is acceptable to the FDA for the patient sizes that need to be tested in order to get an approved drug. So things are changing both on the drug development side as well as at the FDA to enable this risk aversion to maybe be lessened. Limited expertise in mitochondrial function was always a problem years ago in pharma and biotech. Most mitochondriologists in those companies were relegated to being toxicologists, and that is just looking for the off-target or toxic effects of compounds that are often at mitochondria. But again, this is changing, and it seems to be changing quite rapidly. As all of you, I think, know from what has been presented today, the complexity of metabolism pathways and the complexity of mitochondria is huge. Sometimes when you add a drug to cells or an animal modeling the disease, you don't have an effect. And if you study it long enough, you may find it's because the organism has compensated for the presence of the drug. That is, our metabolic pathways are very complex, and you can tweak one pathway just to have the cell upregulate another pathway that sort of makes it as though you never added the drug at all. There are quite a few proteins that make up mitochondria for which we don't have structures of the protein, and protein structure is actually very important for drug discovery, so that our medicinal chemists can look at the structure of the protein and fit a molecule into the three-dimensional structure of the protein, like at its active site or at an allosteric site. But even more so, there are some activities in mitochondria that we know are very important. Some of these are at the mitochondrial membrane, that we still don't know what genes encode those proteins. And so we know there's a function, but we don't know the genes that encode it. When you don't know the gene, it makes it quite difficult to study in drug development. Conservation of target across species was touched on earlier as well. We run a program where a compound that we've developed works a bit differently in mice than it does in higher species. And it's because the sequence of our target is different in mice than it is in human species. And so that can be a challenge to the development of drugs for these purposes. And all right, I'm going to, in the effort to cut down on time, I'm going to skip some of these next slides. This is simply an example of where we didn't know what the calcium uniporter was made of as far as the proteins that did it when we were hunting for calcium uniporter inhibitors. And as a result, we were completely unsuccessful at finding drugs just based on a functional screen. But others have had more recent success in finding mitochondrial uniporter, calcium uniporter inhibitors, because actually Dr. Mutha was one of the ones to discover the components of the calcium uniporter. And because of that, we now know the structures of those proteins and can then more easily target molecules to them. I think I'll skip over these next so we can get to some summaries. One thing I'll say about this, and with regard to metabolism, we have been working in the area of NAD boosting, and actually boosting of NAD with niacin has been shown in a publication by a Helsinki researcher, mitochondrial disease researcher, Anna Suomalainen, who saw that NAD boosting with niacin, which is a vitamin, could help these patients. And this is one of those examples where the complexity of the pathway makes it a bit more challenging. If we want to discover something that's a bit more robust than niacin in producing the end product of the pathway, which is NAD, you can see that this metabolic pathway is really quite complex. And so that can hamper the discovery process. This is another example. When you go to modify the amount of NAD in a tissue by boosting with nicotinamide or nicotinamide riboside, look at the differences that you can boost in different tissues. In the brain, the ability to boost it is fairly low. In the kidney, it's dramatically higher. And we don't have a complete understanding of why that should be. And so these are lacks, there's a lack of knowledge about this that hampers our discovery process. So to summarize, given the great news is that given the unmet medical need in mitochondrial disease, the drug discovery efforts will continue to expand, and you've heard great examples of how that is taking place in the drug discovery world right now. The willingness to accept those risks is now decreasing. The annotation of the mitochondrial proteome, which Dr. Mutha's group and others have gone a long way to help us achieve, has improved. And we're now getting, that has not been really touched on, tissue-specific drug targeting is gaining momentum. So if we need to get a drug specifically to the brain or specifically to the heart, there are ways to contemplate doing so. And obviously, one of the biggest hopes for all of those suffering from these diseases is gene therapies. I think Vamsi made the comment, though, as well earlier, that gene therapies are probably going to still need to be combined with small molecule therapy, as most gene therapies have not been completely restorative of function. And so maybe you can tweak the treatment a bit by adding a small molecule that raises the success rate of the therapy. So I'll close now.
Thank you very much, Dr. Murphy. Dr. Klein, if you'd like to share and get started, that would be great. We are a little bit behind.
Thank you very much. I will try to pick up the pace a bit. It is truly a pleasure to be here today. I've been working in the field of mitochondrial disease for over a decade, in clinical trials of mitochondrial disease for over a decade. And when our work started many, many years ago, there really were no other programs for patients with mitochondrial disease, and there had never been before clinical trials really in patients with mitochondrial disease. And what's been an over decade-long journey has resulted in a number of really, really important learnings that I wanted to share with the group today, because we stand at a really exciting point in time now with so much new science, so much exciting science, and so many clinical trials now for people affected by mitochondrial disease, that it's important that we learn the lessons that we've gone through so we can increase the probability of success in the clinical trials that we're running now. I do want to say that it's been an incredible privilege to be able to have been working with the mitochondrial disease community and so many patients, families, and physicians over so many years. Incredibly dedicated group and really a very strong community. And I can see from on the participant tab in Zoom that there's some very familiar faces to me, and I'm grateful for all the relationships that have been formed over the work we've done together over the past decade. Just a word about PTC Therapeutics. We are a global company focused on developing drugs for patients with rare diseases. We were founded over 25 years ago with the explicit mission of using pioneering science to develop drugs for kids and adults who desperately need them. And we've grown quite a bit over the past 25 years and now have over 1,300 team members in 20 offices worldwide. And we're currently selling drugs for different diseases in over 50 countries. We have a very strong and broad portfolio of therapies for rare diseases. We have several that are in the market right now, including therapies for Duchenne muscular dystrophy, rare disorders like hATTR amyloidosis and familial hyperlipidemia, cholesterolemia in Latin America. We also have, from our splicing platform, discovered a drug for SMA currently being marketed by Roche, as well as our gene therapy Upstaza, which was approved in Europe this summer, which was the first ever gene therapy approved that's directly administered in the brain. And that's a drug that has been developed for kids with the rare disease of AADC deficiency. We do have an entire platform, our BioE platform, which is dedicated to disorders of energy, including mitochondrial disease, and our lead therapy, vatiquinone, which many of you may know as EPI-743 or now PTC-743, is currently in registration-directed trials for 2 different indications, including mitochondrial disease-associated seizures, which is a phase 2/3 global clinical trial, which is now been enrolled for kids with mitochondrial disease and associated seizures, as well as a phase 3 global trial for Friedreich ataxia. We have a number of other programs at different stages of development in our BioE platform. But today, as I said, I want to focus on our lessons that we've learned in developing drugs for kids and adults with mitochondrial disease over the past decade. I'd like to start first with just talking about the ideal disease attributes for drug development. When we think about developing drugs for a specific disease, there's certain things that we really look for that make drug development easier. First, it's very good to have a disease that's homogeneous, where there's shared genotype, phenotype, similar symptoms, and similar rate of progression. It's very important to have robust natural history with well understood disease course from the beginning of the disease to the end stage, including how quickly patients move through the disease course. It's very important to have objective metrics of disease, things that are easily measured and correlate with disease severity. It's also important to have a large number of patients who could meet the criteria for a clinical trial, which allows for rapid and easy enrollment of clinical trials. And it's also very good if there's a precedent for drug approval. If there's been already a path established for how clinical trials should be designed, what should be the endpoints in clinical trials, and also regulatory authorities like the FDA who are familiar with drug development for that specific disease. Well, I think from the discussions today and just from our own experiences, it's pretty clear that mitochondrial disease does not meet these ideal attributes. I think we know that well, and that's in part of what the challenge is in developing a drug for mitochondrial disease. We know that mitochondrial disease is a family of incredibly heterogeneous diseases. We know that 2 individuals can have the same genotype or maybe the same name syndrome, like Leigh syndrome, but have very different symptoms, very different rate of progression, and that complicates drug development. We also know that there's no established development pathway because we haven't yet had a drug get across the finish line and get approved. So we don't yet have that precedent of exactly how to do drug development. We have some challenges in terms of endpoints. We also don't have a lot of really strong natural history. There's some very nice pieces of literature which give some idea about the progression of mitochondrial disease, but we don't quite have yet the robust natural history that's really needed for drug development. Also, given the fact that we've had so few clinical trials over the years, we don't really have a strong culture yet in the community of clinical trial participation, which means that we need some strong education efforts for both patients and physicians regarding what clinical trials are about, and in particular, this idea that a clinical trials is a lot different than clinical care. And so if you're in a clinical trial, going to a doctor's visit is a little bit different than just going as if you were going for a regular checkup or examination. And of course, what we also know is that while mitochondrial disease as a whole includes a lot of people, each individual syndrome or genetic type can be quite rare, which poses some challenges in terms of enrolling and doing larger clinical studies. So I just wanted to highlight a top 5, if you will, of some key lessons that we've learned over the past decade that I think could be very helpful in our efforts now as a community in getting ready to and also participating in existing clinical trials. First is, appreciate the reality of the challenge. Second, we really do need robust natural history and patient registries. Third, it's really important to understand that drug development of mitochondrial disease is a team sport. It requires strong relationships across all stakeholders. 4, we need to really work hard to develop a culture of clinical trial participation. And finally, when we're doing clinical trials, we need to make sure that we have trials that are designed that are practical, informative in terms of demonstrating that drugs are safe and effective, but also not overly burdensome to patients and investigators. And I'm just going to walk through each of these 5 lessons in a bit more detail. First, appreciate the reality of the challenge. Mitochondrial disease drug development is hard, and there's no getting around that. And I think it's always important then to acknowledge that there's a challenge, and we have to be very sure that we don't believe anyone who's going to say that they have a shortcut or a miracle therapy without testing it. I think this is just an important in terms of setting expectations. We're going to have a lot of trials for mitochondrial disease patients. We're going to have a lot of candidate therapies, and obviously we hope for as many of them to succeed as possible, but it's always important that we understand from the beginning that this is going to be tough, and that the drug development process will have many steps along the way, as Anne alluded to in her presentation. And that means that we may need multiple studies to get a drug approved, and that as we're doing these studies, the way that the studies are designed have to be done in a way to ensure that we're getting the right information around how a drug works and its safety to lead us down that path towards approval. It's also really important that we learn from each other, and for any of my industry colleagues on the conference here, I think we need to learn from each other, which is really important. I think there's often a misconception that companies are competing with each other. We're not. We're all in this together to help get important therapies to people with mitochondrial disease, because I think it's quite clear that we're going to need not just one therapy, not just 2 therapies, but I think given the complexity of mitochondrial disease, we're going to need a lot of therapies, and we're all in this together. Second, robust disease natural history and patient registries are really, really essential to effective drug development. When we design clinical trials, it's so important to have comprehensive natural history that tells us about how symptoms play out over time. What is the rate of progression of certain symptoms? Because these help us understand how long we need to make clinical trials in order to show that a therapy is beneficial. It also helps us understand inclusion criteria, who to include in a trial, because obviously we want to include patients in trials who are most likely to show benefit to our therapies. And also we need to make sure we have the right endpoints. And endpoints are basically the way we tell that a drug is working. So for example, if we were developing a drug for blood pressure, we want to make sure that we're measuring blood pressure. And by way of contrast, if we were developing a drug for diabetes, we may not be as interested in measuring blood pressure, but something relevant to diabetes. And that's a little bit of an oversimplification, but it still holds true. If we're focused, for example, on our study of vatiquinone and mitochondrial disease-associated seizures, obviously we want to make sure we're enrolling patients who have seizures and that we have our endpoints are about seizures, because that's the story we're trying to tell. That's the evidence we're trying to gather in our clinical trial. It's also important to understand in natural history the correlation between genotype and phenotype. That's really important in mitochondrial disease, where there may be differences related based on genotype alone. It's also important to understand, to get long-term longitudinal data about the timing of symptom onset and rate of progression. It's really good to know how long it takes once a symptom appears for it to maybe get worse. That allows us to understand, can we then show in a trial that we're making a symptom better versus what would happen without any treatment, or in other words, what would happen in the natural history of the disease. And finally, as Anne mentioned, registries are incredibly important to help with trial organization, selecting study sites that would be close to where their patients are with a particular disease, and also make it easy to reach out to patients and families to let them know that a clinical trial is happening. And I want to take this opportunity to really give a shout out to Cure Mito for their incredible work in a short period of time in building the Cure Mito registry, which now has over 200 data points of 200 patients in the registry in over 32 countries. That's not easy work, and that's an incredible amount of productivity in a very short period of time that will be very, very important to us as we bring clinical trials to mitochondrial disease patients. Next, strong relationships are needed across all stakeholders. As I mentioned earlier, rare disease drug development is a team sport. No one group has a playbook on how to do it right. Not a company, not a doctor, not a patient, not a foundation. We're all in this together. And as we design trials, we need input from everyone, from patients, from doctors, from researchers, so that we're sure we're designing the best trials possible in hopes of getting therapies approved into patients. We have to be united as a community and work together and build each other up and leverage each other's expertise and knowledge and be a unified front, because this is a battle, and it's going to be best won if we're all working together and moving in the right direction. And when we talk about stakeholders, this also includes regulatory authorities like the FDA in the United States and the EMA in Europe, who are also very invested in making sure that safe and effective therapies get to patients. And there's a lot of education that we as a community can provide to the FDA that will be very helpful in their work in helping ensure that we get safe and effective therapies to patients. Fourth, we really need to work to build a culture of clinical trial participation in the community. Culture of clinical trials really help in recruitment for trials, and also for patients and families involved in clinical trials to understand what it means to be a clinical trial participant, and also to understand why clinical trials are important. Without clinical trials, we'll never get drugs approved. And it's also important to realize that clinical trials may or may not benefit someone who participates in them, but they will hopefully in the long run benefit the entire community. It's also important that we have education efforts for all stakeholders. That includes physicians, that includes patients, that includes families. This is all really, really important in getting the community in a strong position to be understanding and ready to participate in clinical studies. One important element is understanding the difference between clinical trial and clinical care, as I mentioned. Why, when you go to the physician's office for a clinical trial, it could be a bit different than when you go for a regular checkup. Also understanding why trials have a specific inclusion criteria, and just because someone, for example, may have a diagnosis of Leigh syndrome, they may not be eligible for a specific trial, and there's usually good reasons for that. It's also important to understand why trials may have placebo phases. That is, you have participants enter a trial, one may get a study drug that could work for the disease, but the other participant may get a placebo. And there's a lot of important reasons why that exists. And that's really a key part of drug development. It's also important to understand that you may go to your local physician in Biloxi, Mississippi, near your house for your mitochondrial disease care. But if you want to be in a clinical trial, you may have to travel to Houston or Atlanta to the nearest study site. That's a reality of clinical trials. And it's really important that people understand that there may be some travel required to participate in a study. It's also important for participants to know that they can also withdraw from a trial at any time. Just because in the beginning you participate if you wanted to, you actually have that right to withdraw. Now, obviously, we hope that anyone who enters a trial stays in, but it's important to know that you as a participant in a clinical trial have rights. Finally, it's also very important for patients and families to know that there's a lot of important questions to ask before entering clinical trials. Well, for example, if I have to travel from Biloxi to Atlanta, will those costs be reimbursed? Many times in clinical trials, companies like ours will reimburse travel costs so that it makes it easier to participate in the study. And also the other really important question to ask is, if I entered a clinical study and I'm put on placebo, will I ultimately get access to the drug? That's a really important question. Different companies do things differently at different stages of development. In our trials, we've always tried to ensure that participants will get study drug, but it may differ from trial to trial. And I think it's important that we know to ask those kinds of questions as we begin to participate in a clinical study. And the final lesson that I think we've learned is that clinical trials need to be practical, regardless if this is being something developed at a clinical study site, an academic institution, at the NIH, or by a company. There's a number of important considerations that should be taken into account to ensure that trials are practical and can be executed fairly easily. Picking the right duration of a trial. Everyone wants short trials, particularly if there's a placebo group, but sometimes a longer trial is needed to actually get the right answer to the question of whether a drug is safe and effective for mitochondrial disease. Really important in mitochondrial disease is understanding how supplements are dealt with in a clinical trial, because supplement regimens can differ from center to center. And if we're doing international trials, they'll be different from country to country. So there just needs to be a plan to ensure that either there's some uniform approach to supplements, either everyone can stay on their existing supplements, no one's on supplements, either way, or there's many other options. The reality is it's just something that has to be dealt with in organizing a clinical trial. Global trials can be very helpful in increasing the number of potential trial participants. This is one way we can address the fact that in any one country, a specific mitochondrial disease subtype may be quite rare, and to do a good trial, we need more patients. So going to other countries sometimes is very helpful in building up the number of eligible patients. Endpoints need to be practical, measurable, and not overly burdensome. This is important for both patients and investigators in trials. Mitochondrial disease patients have obviously easy fatigability, inability to perform certain tasks, and there's a lot of challenges that are part of being a person affected by mitochondrial disease. And so if we're going to do clinical trials and have assessments, we need to make sure that there are things that can practically be done for patients, and that also we're not asking patients to do so much that we can get tired out and not get a real accurate assessment of whether or not a drug is working. The other thing we've learned, and I think others are realizing now, is that having remote visits using telemedicine should be done whenever possible so that you reduce the burden of having to travel to study sites, and also any effects that travel could have actually on assessment. So, for example, if you spend a day or so driving several hundred miles to get to a clinical study site, you're going to be very tired. It's going to make it hard then to actually do any kind of testing that is going to help us understand if a drug is effective or not. What you want to do is erase any impact that travel can have on the disease state so that we can get a really good idea on whether or not the drug is safe and effective for a particular patient. And the last thing that's really, really important is ensuring that we have a stable baseline prior to treatment initiation. Part of being in a clinical trial, one of the benefits is sometimes you get access to a center of excellence for mitochondrial disease, where you'll also get special dietitians and physical and occupational therapists who may not only be important for the trial, but also be able to give some guidance and advice and support to just the care of a patient with mitochondrial disease. And that may change in a good way an individual's condition at any one time. So you want to make sure if there's any changes to care, those happen before starting in a trial so that we're sure that we're measuring only the drug that's given and not any other changes in care that may happen that may actually ultimately improve how someone's feeling or functioning. So that's a little bit of a quick run through some of the key lessons. There are many others, but I think these are several important ones to bear in mind because we really are at a very exciting time. I think never before have there been so many therapies in development for patients with mitochondrial disease, so many clinical trials, and as we saw this morning, so many exciting scientific advances that will bring more and more therapies forward over time. I think that over the past decade I've been in this, field. I've seen so many important advances. We've made the community is stronger and better connected than ever. We have better registries that are essential to drug development. And while we may have a long way to go, there's been tremendous progress. And indeed, the future is bright for clinical trials and development of therapies for patients with mitochondrial disease. Thank you.
Thank you very much, Dr. Klein. Dr. Weissig is getting his lighting set, and he should be ready to go.
Yes, unfortunately, I do not have a nice background you guys have. I only have my plain office here.
I couldn't get the background to work for me.
Okay, fine. Let me share the screen. Do you see my screen?
Yes.
You see the presentation mode?
Yes, we do.
Okay. Well, I also would like to thank the organizers for giving me the chance to give a talk at this extremely interesting symposium. I will talk about where we are in the development of new drugs for mitochondrial diseases, and I will focus on Leigh syndrome, of course. As of 2 years ago, there have been a total of 232 ongoing or completed clinical trials aimed at the therapy of mitochondrial diseases. 21% of these trials included new molecules, so-called new chemical entities, and I will get back to this. 12% of these trials included FDA-approved drug, repurposed drugs, that means drugs which have been approved for other indications but not for mitochondrial diseases. 18% include nutritional interventions. A large variety of available supplements are being tested. And approximately half of these 242 ongoing or completed clinical trials involved the testing of new diagnostic tools, and also very important, we investigate the compatibility of widely used general drugs with patients suffering from mitochondrial diseases. Now, I have updated this and focused on Leigh syndrome, and this compiled a few months ago. A total of 16 ongoing or completed clinical trials specifically targeted at the treatment of Leigh syndrome. Half of these trials, that means 8 trials, involve new molecules, new chemical entities. 12.5% again include old drugs, and 37.5% include observational studies, registries, as we just heard, and the investigation of tissue samples. I will exclusively focus now on these 8 trials which involve new chemical entities, NCEs. What are NCEs? These are new molecules with new chemical structure. Most important, we have never been FDA approved before for any other applications. And here are these 8 trials. Now, please don't get lost in this busy slide. I will draw your attention to 3 major aspects. If you look at the drugs, the new chemical entities which are being investigated, you see we have one which is called EPI 743, and that's actually developed by PTC Therapeutics, which we just listened to around Matthew Klein. And the other one is KH176, being developed by Condrion. That means only 2 new experimental drugs are currently being tested specifically for the treatment of Leigh symptoms. Another one at the bottom has been withdrawn, so I didn't consider this. So that's the first conclusion, first aspect, only 2 new experimental drugs. The second one is both drugs are being currently tested in clinical trials, which are still recruiting. This one, for phase 2, started in 2021, is still recruiting. And these two, for phase 2 and even phase 3, done by BTC Therapeutics, we are also recruiting. That's the aspect number 2. Now, the third aspect is, what are these drugs? Both drugs are so-called redox active molecules, antioxidants. Does it make sense to develop antioxidants for the treatment of mitochondrial diseases? Yes, of course. We all know that mitochondrial dysfunction in one way or the other leads to the elevation of reactive oxygen or reactive nitrogen species or free radicals. And we also do know that free radicals, or I will from now on only call them ROS, but ROS involves also reactive nitrogen species. They cause mitochondrial DNA damage, and they can cause a whole bunch of other clinical symptoms, neurodegenerative diseases. Alzheimer is being associated with elevated ROS levels, diabetes, metabolic disorders, chronic inflammation, and more. Subsequently, currently, actually 8 redox active NCEs, antioxidants, are being tested in clinical trials for therapy of all mitochondrial diseases. And among them, EP743 and KH176. So first conclusion is a lot of new drugs which are being tested for the treatment of mitochondrial diseases are antioxidants. And for that reason, I would like to say a little bit more about antioxidants, because there are promises, limitations, and challenges. Let's talk first about the promises of using antioxidants for the therapy of mitochondrial diseases. Antioxidants can ameliorate symptoms and slow down the progress of disease. As a result, they can and will improve the quality of living. However, antioxidants cannot cure the disease. Further, all of the 8 redox active compounds, which are just introduced, also could be tested for the treatment of Leigh disease. Likewise, these 2 drugs which are being tested for Leigh disease are currently being tested for the treatment of other mitochondrial diseases. Like KH176 is also tested for MELAS and for Leber's hereditary optic neuropathy, and EP743 also for Leber's hereditary optic neuropathy, Pearson syndrome, Friedreich ataxia, and others. What are the challenges in trying to develop antioxidants for the treatment of mitochondrial diseases, and in general also for all other diseases? There are many different reactive oxygen or reactive nitrogen species. There are superoxide radical anions, hydrogen peroxide, hydroxy radicals, and many more. That's number 1. Many, many different ROSs. Second, these RONS are selective in their reactions with biological molecules. And surprisingly, we leave most of them undamaged. Essentially, and I have highlighted this in red, the real culprits are hydroxy radicals. They attack everything. And now in the context of drug development, more important is there is no universal best antioxidant. That means each antioxidant reacts with different reactive oxygen or nitrogen species at different rates in different locations and protects different molecular targets. So developing one antioxidant to treat it all is almost seemingly impossible. Another big challenge, and that's also more my personal view, if I may offer this, and I think is still generally a little bit underappreciated. We have to look at 3 different levels of reactive oxygen nitrogen species. There are 3 different intercellular levels. The first one is the physiologic level, and this might be surprising to many. We actually need reactive oxygen nitrogen species. We need free radicals. They are serving as messengers they are widely responsible for the regulation of the entire metabolism. These are the so-called physiologic levels. Even if we increase the levels of reactive oxygen nitrogen species, for example, in response to hypoxia, in ischemia reperfusion injury, hypoxia, ROS level goes up. And the purpose is these ROS, reactive oxygen species, we actually trigger the launch of protective pathways. Now, only if we further increase the level of reactive oxygen species, we have a pathologic excess, and only at this level, again, which is being produced by an unregulated overproduction of reactive oxygen or nitrogen species, at this level, we get oxidative damage. So now the real challenge is, imagine a normal, healthy human being just has elevated levels of reactive oxygen species in order to trigger protective pathways. And now we kill all these reactive oxygen species by putting antioxidants in the body. Well, maybe it's not so a good idea. So the real challenge is antioxidants should only target the excess of reactive oxygen and nitrogen species. And how to do this? This is a huge challenge. In the second part of my talk, I would give a brief summary of clinical trial outcomes related to these two drugs, which are being tested for the treatment of Leigh disease. And now I do not read the title of the clinical trial. I don't read the aim of a clinical trial. I also would like to pay attention to the outcome regarding the efficiency, safety of the drug, in this case, KH176. So the results. First, this drug is well tolerated at certain doses, and the pharmacokinetic profile is supportive for twice daily dosing. So it's safe at certain doses. Only at high doses, this drug can cause clinically relevant changes in the cardiac electrophysiology. So at a certain dose, it's safe. Another clinical trial involving the same drug results, again, the dose was well tolerated and appeared as safe. No serious treatment emergent adverse events have been observed. However, also no significant improvements in gait parameters, for example, or other outcome measures have been observed, yet generally a positive effect on alertness and mood has been recorded. And again, I do not read the exact title and the exact aim of a clinical trial. I hope that works this way. The next three ones are dealing with EPI 743. Results, 12 patients treated with this drug have survived, 2 patients died. Out of the 12 survivors, 11 demonstrated clinical improvements, with 3 showing partial relapse. And in general, this drug has modified the disease progression in more than 90% of the patients, which is, of course, a very, very good outcome. Another one, same drug. In this trial, 4 patients demonstrated arrest of the disease progression and reversal of visual loss. 2 patients exhibited a total recovery, no adverse effects. Also very good results. And finally, another for EPI 743. It was again demonstrated to be safe and well tolerated, and at treatment for 24 months, a statistically significant improvement in neurological function and disease progression has been recorded. Now, in my final slide, I would like to show where we are with development of new drugs. These are all, I compiled this approximately 2 years ago. These are all new chemical entities. Okay, these are drugs which have not been approved by the FDA for treatment of any diseases, let alone mitochondrial diseases. And it shows where we are in terms of phase 1, phase 2, and phase 3. So you can see, as of 2 years ago, none of them has been FDA approved yet. Now, I'm updating this graph now, which I made 2.5 years ago, based on the fact, as I introduced, that EPI 743 is currently, since February 2022, recruiting for phase 3. So I upgrade this and give AP 743 and push forward However, as you can see, the front runner is still an approach which uses gene therapy for the treatment of Leber's hereditary optic neuropathy. Well, thank you very much for your attention. Hello.
Thank you. Hello. Sorry, my screen got hidden on me and couldn't find the mute button. Thank you very much. Unfortunately, we've run way over time. These have been great presentations, and there's been a lot of information passed along. I've been taking note of the questions that are coming through, and I will get them to the speakers to make sure that we can get something to put out to people so they can understand the answers to those questions. Thank you very much again, and I'm going to pass on to Danielle Boyce now for the next session.
Thank you. It looks like my video cannot be started because the host, something about the host. So if someone can help me turn my video on, that would be great. There we go. There we go. Can you see me? Hi, everyone. Thank you. First, I'd like to thank the Cure Mito for inviting me to moderate this session. What a treat. This is right in my wheelhouse. I'm Danielle Boyce. I am a researcher. I am a data scientist. Well, they didn't used to call us data scientists back when I started 30 years ago. I like to say we were just nerds back then. But I'm a data analyst and engineer and faculty member at Johns Hopkins. But more importantly, I'm the mom of a child with a rare form of epilepsy, not Leigh syndrome, but he had infantile spasms, which sometimes happens to people with this condition. And now he has Lennox-Gastaut syndrome. So Charlie has many of the same comorbidities that we talk about here today. And I'm very honored to be a member of the Cure Mito Scientific and Medical Advisory Board. I am particularly impressed about their handling of data and data standards. And Sophia and I are very like-minded in that way, because professionally, before we had children, this was an area of interest for us professionally. And so we've had many, many discussions. I'm incredibly impressed by how Cure Mito approaches everything they do, from social media, communications, marketing, graphics, but especially this area, data and accelerating research through data. So it is my great honor to introduce Sophia Zilber, who is a Cure Mito board member and the patient registry director. This presentation will give an overview of Leigh Syndrome Global Patient Registry and what has been accomplished so far and future directions. Sophia, off to you.
Thank you so much, Danielle. Can everybody hear me? I'm going to share my screen. So thank you for the introduction, and we are also so lucky to have Danielle on our team. Her expertise is incredible. Just to add to this quick introduction, so as Danielle said, I'm a board member and a patient registry director for Cure Mito. I have many years of experience working with clinical trial data. I still work as my day job in the pharmaceutical industry. And I work on statistical programming and analysis of data for clinical trials and preparing data for regulatory submission. I became a part of this community 5 years ago when I had a daughter, Miriam, who was born as a perfectly healthy baby initially, and very shortly after that became very, very sick, and within a few weeks died. I volunteer my time for this community in Miriam's memory, and I'm very, very honored to be a part of the Cure Mito. I would like to start this talk about the registry with our goals for this registry and really our vision and the reasons for starting the registry. So what were our goals? First, we wanted to have a registry that's international so that anyone, anywhere in the world could join the registry. That was one of our important goals. Second, we wanted to identify and learn about our population, again, everywhere in any country, anywhere in the world. A high priority for us was to have data that is accessible and available to researchers and to the industry, to those who want to access and work on this data. Another priority was to enable recruitment for clinical trials. As Dr. Klein and others have said before, recruitment for clinical trials in rare disease is very, very challenging. And registries can really play a huge role in finding these patients and being able to contact them. A very, very huge priority for us was sharing of the results. We really wanted to have a registry where we don't just ask patients to enroll with the hope that eventually something will come out of it, but we wanted to have a registry where we are always sharing the results. Our community is always engaged. The data is available, and we are always learning from this data. And another important thing on that, we wanted to make sure that we know exactly what's in the data, so that if we are approached by a company or a researcher, and we often are, and they are asking, do we have patients with a specific symptom, specific gene or mutation, and we can very quickly let them know basically within a few seconds. So that's very, very important to us, so that this registry is always working and always doing something for our community. And with all of this together, we want to build a worldwide community, a strong community of patients and researchers. Our registry has been started in a partnership with CORDS, Coordination of Rare Diseases at Stanford. Stanford Health is a very large healthcare system that was established a very long time ago in 1894. And CORDS registry has been around since 2010, so it's well established. It's around for more than 10 years. It's listed on clinicaltrials.gov. At this time, it's a disease agnostic registry platform. And at this time, it has data for more than 2,000 rare diseases. CORDS works with 91 partner groups. It has more than 15,000 participants from 87 countries and all 50 states in the US. This registry is completely funded by Stanford Health. So there's no cost either to us or to any patient groups or to researchers or industry who would like to get the data or contact patients. This registry is HIPAA protected because Stanford Health is subject to HIPAA, is a healthcare organization, and it's also GDPR compliant. Our registry has two surveys. One of them is a general survey, and in the CORDS registry platform, the general survey is exactly the same for all rare diseases. It's using common data elements that are recommended by the NIH, and the fact that it's the same survey for all rare diseases makes it easy to harmonize data across diseases and see what's in common between them. Additionally, there is a disease-specific survey that's developed separately for each disease. This structure makes it possible to potentially add other MITA conditions in the future. General survey always remains the same, and disease-specific survey can be developed for each individual disease, and potentially our Leigh syndrome survey could be used as a basis for that. This platform is linked to Orphanet, list of rare diseases, and any disease that's in Orphanet would be there. Joining this platform is based on disease. It's not based on a patient group. So even if a patient has a disease that's where there is no partnership with a patient group, they still can join. We have designed our survey with the input from our advisors, our registry advisors, scientific and medical advisors, and the industry. And this is some of the information we are collecting. We are collecting demographics, diagnosis and genetic mutation information, time to diagnosis, loss of milestones and the ages when the milestones were lost, disease management, what patients are using to manage their disease, first concerns that were noticed, and a history of symptoms and concerns, infections, healthcare utilization, caregiver burden, and quality of life. This is a poster. Our first results on this registry have been shared last year in February at the Mitochondria Targeted Drug Development Summit. And this is the poster that we shared. It was based on just 3 months of data at that time. And I'll just highlight just a very few things from there. At that time, we had 134 enrolled participants, and we included 93 in this analysis. We found that the interesting thing, we found that the time to diagnosis was relatively short, actually, for Leigh syndrome. It was under 6 months for 30% of patients. It was between 6 and 12 months also for 30%. And it was short overall. Only 6% had time to diagnosis greater than 5 years. We found for almost 95%, their diagnosis was determined by genetic testing. And of those, again, for 95%, around that, we knew their gene mutation. And overall data quality was good. We didn't have a lot of missing data, and our results were consistent with what we know about Leigh syndrome. So that was very important for us to know based on our initial data. And as I said, we are very much focused on sharing results widely. So we've had other opportunities to share. We shared the same poster at UMDF Symposium in June. We also just shared again the same poster at Mito conference in France. We have another poster, and I'm going to say more about it on the next slide, that's actually being presented right now at the FUSE FDA Computational Science Symposium. And we're going to have another also different poster presented at NORD conference in October. That poster is called Building a Worldwide Community, Leigh Syndrome, a Global Patient Registry. And that poster will focus on more than the numbers. It will be focusing on our entire effort to build the community through the registry. We are also working on a paper, which is actually almost completed. And it will be submitted to Biomolecules Journal in October. And we already have an announcement of the paper online. So I want to share about some of our partnerships in regards to this registry. One is a partnership with Sumptuous Data Sciences. As some of you might know, in order to submit the data to the FDA, data has to be converted to a standard called CDISC standard. That's a requirement for all regulatory submissions to FDA or PMDA. And so we are collaborating with Sumptuous Data Sciences specifically with that goal. We wanted to learn if our data is compatible with CDISC or not, and to what extent. After that, we want to convert this data first to CDASH format and then to SDTM format, which is what FDA accepts. And after my talk, Rhonda from CDISC will speak, and she will give much more details on this data standard. We wanted to achieve data that's regulatory submission ready. And of course, we wanted to share all these learning with the entire Mito community, because this is, as far as we know, this is the first attempt to take patient-reported data from a patient registry and convert to CDISC. So that's very interesting how it works out. And so our results, we actually found that more than 90% of our data maps to existing CDISC domain, which is really, really great. And we are working on this conversion. We have a press release. When we started the project, we had a press release that's on our website. And right now, we have a poster, and Parag, who is speaking in this panel after a couple of talks from me, he's going to share a lot more on this, and he's presenting the poster at the FUSE FDA Computational Science Symposium right now. And we're going to share the poster with the community in a few days as well. Our other partnership for the registry is a partnership with CPATH, Critical Path Institute. CPATH has a platform that's FDA funded and where multiple types of data could be integrated, such as patient-reported data, electronic health records data, clinical trials data. It could be all kinds of data. And this data could be combined and we can learn more from this data. And so again, this is the press release that we had with CPAS. Our goal is to integrate our Leigh syndrome data into CPAS platform. Others in MIDA community are doing the same. MIDA Action also has a similar press release, and they're sharing their data that's collected in the mobile app with CPAS as well. And Alex from CPAS, he will also speak, and he will share more about how MIDA community can work with CPAS and how we can share our data. One thing I wanted to note regarding data sharing is if we want to combine data and combine data for the same patient from different registries or different data sources, one of the ways to do it is by using a global unique identifier, GUID. And but one important detail on that is that GUID has to be developed in exactly the same way in all these different data sources. Otherwise, we would not be able to combine it. And so we are working with courts to create GUID through NIH GUID generator. It's a very commonly GUID generator in a rare disease. It's used by many in rare disease, and there are very successful cases of using it. So I wanted to share that. So now, what's our current status of the registry? Our registry is exactly a year old. We actually started it last year during MIDA Awareness Week, so it's exactly a year this week. We have more than 200 patients enrolled from 32 countries. So that's really wonderful. We are so extremely grateful to all the patients that have enrolled. It's so important. Each enrolled patient really matters for this. We have a flyer that is available on our website. It could be downloaded. Patients could bring it to their medical provider. Doctors could print it out and share it with their patients. And we really appreciate all collaboration in sharing this. We have surveys created in Spanish and Portuguese. All translations are certified and IRB approved, and please reach out to us if you need that. And if there's demand for other languages, we could add additional languages too. We also have frequently asked questions on our website as well. Again, yeah, please continue to join. It is very important. We really want patients to know that we speak to researchers. We always show aggregated results. One feedback that we're getting is that it's very important and very interesting to do data analysis by gene. So we haven't done it so far because we need more patients to enroll. We have many genetic mutations in the registry, but in some cases it's a few patients per gene. So we really, when there's more patients, we will be able to do it, and it's very important. And yeah, I just really wanted to share a quote that I thought speaks to our efforts. It's by Eleanor Roosevelt: "The future belongs to those who believe in the beauty of their dreams." So I feel that it really speaks to what we're doing. And finally, I would like to thank everyone. I want to thank Casey, most importantly. She's truly the heart of this foundation and this effort. And I would like to thank all of the patients that are enrolling in the registry. I want to thank Cure Mito partner families for helping us share and spread the word and spread awareness. I would also like to thank everyone who is working with us on the posters, on the paper. If you see any of our publications, you'll see there are many authors. Everyone who works with us volunteers their time to help, and we're so extremely grateful and for them giving us their time and their expertise. If anyone has any questions, please do not hesitate to reach out to me by email. You can also connect with me through LinkedIn, and please follow Cure Mito through any social media. We post a lot of updates, and please follow us. Thank you.
Thank you so much, Sophia. That was terrific, and how impressive in just 1 year. It's amazing how much you guys have done in just a year. Please continue to put your questions either in the Q&A or the chat, and we'll go through them at the end and try to get some of your questions answered either live, or we can get back to you as well. I'm very pleased to announce our next speaker, to introduce our next speaker, Rhonda Facile. She is the Vice President, Partnerships and Development at CDISC. That's those FDA data standards that Sophia just shared about. And her presentation is called CDISC and How Data Standards Can Help Drive Development of Mito Treatments. To you, Rhonda.
Thank you, Danielle. Let me just share my screen. Okay, can everybody see my screen? Great. Well, thank you, Sophia, and the Mito crew for inviting me to present to you today. It is amazing what's been accomplished in 1 year, so hats off for that. And just so you know, CDISC is very pleased to support and partner with Cure Mito as they move forward with their very important work. So I'm going to talk to you a little bit about CDISC and then how standards can help drive development of treatments. So I'm going to give you a quick overview. You've already introduced me. I've been with CDISC for a long time, 15 years. Prior to that, I have experience from the pharmaceutical industry and regulatory affairs and clinops. So I've been at this for a little while. So I'm going to just start with what is CDISC. Some of you might know very well, like I recognize Parag from long ago, and I know that he knows very well what CDISC is. But so pardon me if this is repetitive for some of you, but CDISC is a global standards development organization. It was founded in '97 by volunteers. And today it's a 501(c) nonprofit organization. We have over 40 employees. We're soon to hit the 50 soon. And we have over 100 contractors. Also working with us is 1,000 volunteers all over the world. And we develop training both online and virtual, online and classroom rather, for most of the standards. CDISC has 545 members. That's the primary source of funding for CDISC is the membership. But it's also a little bit from events and a little bit from education to kind of keep the lights on and the doors open. As an SDO, it's really important that CDISC reach out and work with different groups in order to push forward not only data standards, but also helping to push the science forward and making sure that the data is clear and can be used by multiple different stakeholders. So this slide just summarizes some of the areas where we have alliances and collaborations or partnerships. The first one there on the left is therapeutic area standards. And we've been working for years with CPATH, which was mentioned earlier, a great organization. We have been working with them for, as I said, many, many years. And together with them and various other stakeholders, we've developed about 45 therapeutic area standards. And what those are is they build upon foundational standards, which is those kind of concepts that you see regardless of whether or not you're visiting your physician for a checkup, a clinical encounter, or you're in a clinical trial. And that would be those things like Sophia listed on the list on her presentation: demographics, medical history, medications, things of that sort that are collected at every encounter. And so what we do with the therapeutic area standards is we layer over that therapeutic area concept so that we can give investigators a more complete package of standards for the area that they are doing their clinical trial. On the right there, you have the regulatory collaborations, and Sophia mentioned this as well, and I'll talk a little bit more about it later, but primarily it's FDA, who requires the use of SDTM, ADAM, and defined XML. PMDA, we also work with EMA, and we have some collaboration with the Chinese NMPA. On the bottom there, it's the world of standards development organizations that CDISC is active in. So we work a lot with ISO, the International Standards Organization, IHE, ICH, HL7, which is the healthcare standards organization, as well as the folks that develop the OMOP model, the Odyssey Group, DICOM, GS1, and others. That's just a few of some of the larger ones that we collaborate with. On the bottom there is FUSE, and that's a group that looks at analysis and implementation issues. And so we work very closely with them as well. On the right is sort of a different set of collaborators. And I'll speak about this later in my presentation, but as we get to a world where we are leveraging real world data in order to generate real world evidence, that requires working with academics and some of the folks that are on this call and trying to look at how do we make the CEDAS standards easier to implement so that they can be used earlier in the research process, not at the end when there's a submission to be completed. Sophia mentioned CDASH, and that's the data collection standard. So the idea is to collect it in a standard format, and then you have it there, and you can convert it to SDTM at later, and it goes much quicker because there's about a 75 to 80% overlap between those standards. So we're working with academic institutions. And one of the things I'm going to highlight later is a collaboration with Open Clinic and REDCap, where we are making the standards available in those platforms because those are platforms that academics use heavily. So a little bit more about CDISC. I mentioned we're a consensus-based standards development organization, and the standards can be used for clinical and translational research, and all of our standards are freely available on cdisc.org, all of them. So I want to make sure everybody knows that. We don't try to charge for those standards at all. And actually last week we made CDISC Library open and free, which is a repository of all the standards. So anyone can go in, create an account, and be able to access and download standards and suck them into their system and utilize them right off the bat via CDISC Library. And we have an IP policy that ensures that those standards stay open forever. Globally, we have ongoing research support in the Americas, Europe, Japan, China, India, Korea, and other regions, and it seems to be growing year by year. And our standards supporting documents are available in English, Chinese, Japanese, and Chinese. So a little bit about regulation, just following on what we spoke about earlier. What this diagram shows you is the diagrams on the left, the documents in the blue field on the left. Those are the FDA documents that created the obligation to use CEDAS standards in regulatory submission. In the middle, you have the FDA Study Data Technical Conformance Guide, which is instruction on how to use those standards. And on the right there is a screenshot from the PMDA that describes the same. So as I mentioned, they're required in FDA here in the United States and in Japan, but they're recognized for submissions by the Chinese NMPA. And increasingly, EMA is more interested in data, both getting data in for the real world data issue that I mentioned, and also they can ask for data if they have questions in their review process. And when they do, they don't object to getting it in CEDAS format. So a little bit about governance. One of the key important things about an SDO is that you have a solid governance process that ensures that, that everyone doesn't have to agree, but at least you're able to reach a consensus and that you take on the comments and objectives, you consider them, and you use fair, impartial, and open, transparent processes to develop the standards. And this is, an FDA requirement that they would not be able to advocate standards were it not for a good governance process. So on the bottom there, it's a rather simple, straightforward governance process that involves, scoping and planning, identifying the concepts that are going to be in focus, developing the standard. Then there's an internal and a public review, and those are really important. With the internal review, the idea here is not to develop standards that already exist or would be in conflict with current standards. So that's a really important step. And the next one is a public review, which is 60 days in duration, and anybody anywhere is welcome to comment on standards. In between each of these steps is a governance group that meets to ensure those things that I just described, that we're not developing standards that are elsewhere or in conflict with existing CEDAS standards. And so when we talk about standards, this is what we're talking about. We're talking about this end-to-end process, and this is where the CEDAS standards are. And so they start on the left there with preclinical, which is animal data. And then from there, we have a protocol representation model. And then we have CDASH, which is the data collection model, or the case report form standards. And then you have the organization part, or the tabulate part, which is SDTM. And I apologize, I know this is acronym soup. It'll be over soon, but that means the study data tabulation model. And then next you have analysis, and we have the model for that. And then once you've gone through all of that, hopefully you can submit or publish your report. But in between each of these stages, we have the data exchange standards, and those are in between the bars as you see going across there: ODM XML, define, dataset XML, SDM XML. And so I mentioned before, those are what we call the foundational standards, then layering over top, those are the therapeutic area user guides. And then underpinning all of that is CETUS controlled terminology and the CETUS glossary. There's also a mention there for Bridge, and that's an overarching information model that's maintained by NIH, ISO, CETUS, and NCI. So it's really important. The terms, the definitions are really important in standards. So what do the standards do? I showed you what they are, they provide a common structure in a terminology, what I just mentioned. And in that way, the data can be collected, it can be aggregated, it can be analyzed, and then it could be shared. It could be transferred from one group to another. The other big thing that standards do, that's a requirement. I think it's part 11 requirement to submit data to FDA, is this end-to-end traceability requirement that you should be able to go from protocol and then translate that into what does it look like on the case report form? What does the collected data look like? What do normalized data tabulations, what do they look like? How can you visualize the data moving through this process? Then you have analysis and analysis results. So theoretically, you could go that way and then backwards and have that complete traceability all the way back to the case report form and to the protocol. And so that's the function that CETUS standards provide. So what is the problem that CETUS seeks to address? It's this unnecessary variability at collection. And what you see in front of you is 4 different case report forms. They're all collecting sex, which is relatively straightforward, at least for the terms that are used here. And what you see is the red boxes here. You see M and F for male and female. You see it on the bottom there, 1 male, 2 female in parentheses. You see male and female spelled out. You see male also with a zero in front of it and female with a 1. And so all of these are different, which means that when you get to the data set, it's a mess. And so this is the other part of the problem. If you collect the data in an unstandard way, you're going to get that same variability when you look at the tabulated data. And so in this case, you could see each of these data set names are different. So you're going to have a tough time without spending a lot of time going down and going through the data to figure out that these are actually the same data set. And then looking at the actual table here, where you got subj ID here, you have sex here, but this one says ID and gender. Each of these is somewhat different. And then the data itself, is it 3 zeros and a 1? Is it A1? Is it M? Is it male? You get the idea. And this is a very simple example, but when you look at large amounts of complex medical data, this is a nightmare to sort out. So this is basically the use case for clinical data standards. So now I'll switch and talk to you about CETUS RWD activities. There's a lot of interest in how to leverage real world data to generate evidence. I mean, all of the regulatory authorities are looking at this. They're kicking off large projects to try to analyze it and figure out the best way to do this. And what this shows here is some of the different documents that have been generated around real world data. But it's true is that they want to use it. They want to use high quality RWD in decision making. And this is why I think Cure Mito's foresight in their registry database to go ahead and make the leap and use standards now, because that way it will facilitate the acceptance of that data later on downstream to the regulatory authorities. I think it's super smart. And indeed, recently, FDA just issued these draft RWD and registry data guidances, and they pretty much point to the use of CETUS standards for this purpose. So CETUS also is looking at real world data. And I mentioned earlier about making things easier for academics. We conducted a large Delphi survey a couple of years ago, and we just published the paper earlier this year. And out of that, we asked the community, what should CETUS do in order to make the standards easier to implement? You know, traditionally, transcribing the data into CETUS usually happened at the end when the trial was over. It's called legacy data conversion. And so that being the case, that's not something that academic investigators can really do. And that's not something we want people to do. We want them to use the standards at collection. And so this paper was asking the community, well, what should CETUS do in order to achieve that goal? How do you get people to implement the standards who might not necessarily be an expert in CETUS standards nor want to be? We want investigators to focus on science and taking care of their patients. not to necessarily be experts in the intricacies of SDTM. So, and here's some of the things that the advice that we were given and the projects that we're working on now to fulfill that goal. The first one is to create a basic SDTM implementation guide, which is just looking at those foundational domains. What would it look like for a person who doesn't know CDISC to actually implement the standards? This is a real mind shift within CDISC to think about this. We're also looking at creating a basic study setup guide, potentially in collaboration with REDCap and Open Clinica. We're also putting together a consideration document for using SDTM for observational studies, and also, exploring mapping and collaborating with the OMOP group. I mentioned the REDCap and Open Clinica C-CRFs, eCRFs with rules, so we will continue to work with them to expand that set of standards that now are available freely on both of those sites. And also putting together a C- to SDTM guide. Once you collect it, how do you take it the rest of the way into SDTM? And perhaps Parag will talk about that in his presentation. We've also reached out to HL7 and we've done a FHIR to CDISC mapping. And we're looking at expanding that. Actually, we may have a potential project working with several academic investigators to do just that. And then finally, putting together a brief overview of the CDISC model, the library, and QRS's questionnaires, ratings, and scales, just to make the standards a bit more accessible and easier to find and easier to understand for this group. So what resources are available now? Just really quickly, we have a knowledge base, and that knowledge base contains articles and various different resources that people can get to and hopefully get some of their questions answered. We have an eCRF portal, and this is an example of a demographics case report form. And then here's articles that are in the knowledge base that also address some of the issues that we know occur a lot when people are trying to implement CDISC standards. And then finally, known issues. There are some things that we know are issues that we haven't dealt with, or we don't have any solutions for them right now, but it's a good place to go, is my question one that they know about, or do I need to send them an email and make them aware of something that I've found? So hopefully they can go there and get some information as well. I've already mentioned REDCap and Open Clinica. I'm personally really excited about this. I think it's the right step forward, and we'll continue to populate both of those platforms with CDISC compliant case report forms moving forward. Yeah, this example of the demographics. And that's what I wanted to share with you today. Again, thank you for inviting me to present. I'm happy to take any questions you might have.
Thanks so much, Rhonda. I learned a lot. I'm really excited to dig into some of these resources that you shared, and very interested in this REDCap work too, but because I work with that a lot. So yeah, that's really exciting.
Thank you.
So the next speaker, I'm looking for him, is Parag Shiralkar, the president of Sumptuous Data Sciences. Hello, Parag. And his talk is on the interoperability of Leigh syndrome patient registry data with regulatory submission standards. Take it away, Parag.
Thank you. Thank you so much. And my sincere thanks to Cure Mito Foundation and Sophia and Casey for giving me this opportunity to talk and give a synopsis of the interoperability of Leigh syndrome patient registry data project that we are working in collaboration with Cure Mito Foundation and Sumptuous Data Sciences. Real quick here, I just wanted to mention that this presentation is essentially the follow-up of what Sophia mentioned about the patient registry, and then Rhonda talked about the CDISC standards. What we are trying to do here is the patient registry data, we are applying the CDISC standards and keeping the mechanism of the interoperability ready for the patient registry data. So I would do a quick recap of what is patient registry data, how the collection is designed, and how the data is stored or hosted. Sophia covered this aspect in her presentation, but I would just quickly mention it here, that this is the data collected from the questionnaire response from the patients. It is based out of NIH Common Data Element Repository, survey questionnaire design. and it is stored in the CORDS platform, which is a coordination of rare disease at Sanford. Those who want to know more about how this has been designed, how the data collection is designed, they can go to this reference and find out more about NIH common data elements. So what is the new drug application or biologic license application requirement, the filing requirement with regulatory authorities, whether it is FDA or EMA or PMDA, the Japanese agency? Rhonda gave a brief synopsis of which regulatory agencies made it mandate to use the CD standards and which are recommending to use CD standards. But really what they're looking for when you submit this data to the to the regulatory agencies, they look for traceability of data, transparency, conformance, compliance, integrity, sufficiency. I mean, you can go on and on, but what it really means is they want the data in a standard format, which is agreed upon by the industry, by the public, and by the regulatory agency. This is why they are saying that the data need to be submitted in clinical data standards. And here is some reference I have for providing regulatory submission in electronic format, standardized study data at FDA. So this is exactly why the standardization of data is important. And here are a couple of screenshots where this is the guidance by Food and Drug Administration. And here is some highlighted text from the guidance, which explicitly mentioned that the data submitted to regulatory agency need to be in CD standard. Now, in terms of interoperability, now think about it. We have the patient registry data collected at Cure Mito Foundation based on the NIH common data elements, whereas the regulatory agencies look for the data in CD standards. So we are really talking in two different languages when it comes to use of data or looking at the data is concerned. And the creating of that bridge is exactly what is interoperability establishment. So we create a mechanism to make sure that this data in common data elements of NIH gets converted into CDs based data so that it is ready for submission and analysis purpose when it comes to the new drug application. Now, Rhonda talked about it in detail, but I would just show the three basic areas where we look for the data in certain standards, which is at the collection stage, submission stage, and the analysis stage. So it's not going into too many details, but we are talking about the C collection or the C dash standards, submission or STTM standards, and ADAM or analysis data model. So when we establish the interoperability, this patient registry data is actually in these standards ready to be used or ready to be analyzed by a regulatory agency. So we have this collaborated project between Cure Mito Foundation and Sumptuous Data Sciences to establish the interoperability of the patient registry data. So if you think about these two poles, like one talk about the patient registry data and the second talk about the CDs-based data in the collection stage, data collection stage. If we think of interoperability, we are talking about four distinct aspects here. First of all, alignment of patient registry data with the C- domains. So the data in C- is collected in certain standard format, like, you have demographic data, you have concomitant medications, you have clinical findings, and so on. Whereas the patient registry data we have is questionnaire response, and that questionnaire response has everything. It has demographic information, it has clinical finding information, but this need to be segregated into different data domains when it comes to conversion to C-. So that's the first part. The second part is development of data map to align the content of data, or which is in data language, we call it as variables. Third being conducting the data transformation. So actually having the codes to convert the registry data into the C dash. And then last part is drafting the process specification. So tomorrow, any sponsor or the treatment development companies, pharmaceutical company want to use this type of data, they have a complete mechanism ready, which can show how the registry data is converted into the acceptable format, regulatory acceptable format, or C dash or CDIS format. So currently where we stand now, this is the project we are currently working on. And if you see each step here, we are done with the first part. The registry data has been analyzed and domains in C dash are being created. We have the mapping mechanism complete. And currently the data transformation is in process. We are working on actual conversion of data to C dash. And we have the preliminary assessment ready for the specifications, process specifications. So this is where we stand in terms of the interoperability project that we started back in end of May this year. Here are some key findings. Now, the good news is significant portion of the data from registry can be mapped to C dash domain. And this is a really big thing, because one thing is the data you collect does it meet the necessity and sufficiency purpose for for the clinical reporting. And second thing is how much % of that is getting actually in alignment with the CDIS standards. So the great news is that more than 90% data is actually that can be mapped to C dash, and these are some of the representative domains. And again, not to get into too many technical jargons, but these two-letter abbreviation really represent the type of data in C dash. So for instance, when we say CE, this is clinical, sorry, CE, this is a clinical events, DM is demographic, DS is disposition, then MH is medical history, QS is questionnaire. So this is a standard nomenclature developed by CDISC, and this is why the standardization is important, because we all can speak in same language when it comes to data. Another important finding is that a lot of data pertaining to pharmacogenomics and genetic findings, medical history and questionnaire domains is what we find in registry. So here is some example, like how exactly what we mean by establishing interoperability and how the data gets mapped from NIH common data elements in patient registry to the CDISC based C dash standards. So here is first example. For instance, in patient registry data, you have a column called symptom for seizures. And then you have another column stating value yes, no. Now, remember the slide that Rhonda presented about the gender, how differently the gender is represented, right? And how we can get it commonality when it comes to CDISC standards. So now the commonality here in CDISC standard, when we talk about collection or C dash, is this typical data can go into questionnaire type of data, and the variable QS test has a value, did the patient have seizure symptom, and the output is in QS ORRES variable, and it has possible value of yes and no. So think about it. If you give this column with yes, no to any regulatory authority, they won't understand what exactly to do with this, because they have their own system developed based on the CDISC. If you give this data as QS test and QS ORRES, they get it. Okay, this is how this is, and then you understand the content of the data. So another example is in patient registry, there is a variable called diagnosis gene mutation, and value is yes and no, yes or no. So when we convert this type of data to C dash, we get it into a pharmacogenomic genetic finding domains, and in C dash it is microbiology. So you talk about PF test with a genetic test value, and PFORRES or PF results with a value yes and no. So in other words, this is the language that regulatory authority would like to see the data in. So this is where we are right now working. And now where we stand and where we are heading with this interoperability establishment, and how can it help accelerate the research using this data. So we talked about 3 stages of data. One is collection, second is tabulation or submission, and third being the analysis. So as I described, the mapping mechanism is complete, data transformation is in process, and we are looking at estimated completion time for completion of transformation of data by October 2022. So by end of next month, we should be complete with the mapping mechanism as well as the data being mapped in C dash. So our next step now is to convert this C dash data with STTM. And like Rhonda mentioned, there is a very, very fine process within CDIS to get this conversion quick and seamless. And then we do compliance assessment of STTM and keep the data ready for ADAM or analysis data. So when we keep it ready for ADAM, this is basically the data is ready to be analyzed for statistical analysis. So how does this accelerate the research? We do have establishment of mechanism and process for ingestion, acquisition, and standardization of patient registry data, which is essentially a real world data. It has now with this mechanism, you have ability to share this data available through registry in a very compliant format, which can be accepted by regulatory agencies. And this can expedite the data representation, reporting, management, and analysis for faster research or faster results of the data. So I would like to thank Sophia and Casey from Cure Mito Foundation for initiating this project and collaborating with us. And from Sumptuous Data Sciences, I would like to thank Pallavi Bakare, who is really our SME in CDIS, who is driving this whole process, and Jafanya Bhupati, who is actually doing the transformation of data. So thank you, everyone. And again, thank you so much to Cure Mito Foundation for giving me this opportunity to talk. Over to you, Daniel. Any questions I would be okay to take.
Thank you so much. Looks like we're going to have Q&A with the speakers after 2 more speakers. So please continue to put those questions in the Q&A and in the chat. It is my pleasure to introduce our next speaker, someone I've worked with on and off for about 3 years, I think now, Dr. Alexander Batorin, the Scientific Director of the Critical Path Institute, or CPATH. You've heard CPATH mentioned a few times. And his talk is the Rare Disease Cures Accelerator Data and Analytics Platform, Accelerating Drug Development for Rare Mitochondrial Disorders. Over to you, Alex.
Thanks, Danielle. Nice to see you again. All right. Well, thanks again, Danielle, and thanks, Sophia and Casey, for inviting me to speak to your conference today. So I work for the Rare Disease Cures Accelerator Data and Analytics Platform. Very long name that I will just use RDCA DAP for the rest of the slides. I'm scientific director for the program. And just to start, just a quick word about the Critical Path Institute. So you've heard the name a little today, just to tell you very briefly who we are. We are a nonprofit, started in 2005, that does work to accelerate drug development for disease. The only thing that we don't do is develop drug ourselves, since we are a nonprofit. And really how we work is through an approach of developing public-private partnership to bring all the stakeholders from a given disease area to discuss together in a neutral environment. So we build the regulatory, scientific, and legal framework to convene industry, foundations, key opinion leaders, and regulatory at the same table to discuss in a safe space and maybe have discussions that they wouldn't have with each other in another scenario. So this is really the key of our CPAF work, to develop solutions and insights for disease. We have a strong connection to FDA. We have a branch of the Institute in Europe developing some interactivity with EMA. We have a collaboration with NIH that was just announced last week. And we really have a lot of collaborations ongoing with industry, but also academia and foundations. The key of what we do at CPAF in terms of science is really to leverage our core expertise in data management and standards, biomarker, quantitative modeling and analytics, clinical outcome assessments, and regulatory science to leverage data that has been collected to develop solutions and insights. So more often than not, we use patient-level data to develop these solutions. And these solutions can be a lot of different things. Again, everything that is not drug development, but that could potentially help accelerate drug development and help clinicians in their work. It could be developing models of disease progression. It could be developing a biomarker. It could be developing a novel endpoint. It could also be building a digital app to help clinicians stratify patients for a phase 1 or a phase 2 trial. So these are some examples of the things that we do, but there is way more in terms of activities. And at the moment, we are reaching 30 different programs or consortia. So more often than not, it is the public-private partnership structures that I mentioned through a consortium for a specific disease. So a lot of disease-specific consortium for Alzheimer, Parkinson, and others. We also have consortium dedicated to a specific solution. For example, a consortium in public reported outcomes that is going to focus on that these tools, the PROs, potentially applied to multiple disease. We had new announcements recently. So prior to last week, we had already consortia established for rare disease, including rare ataxias, Huntington, sickle cell disease, Duchenne, and others. We made 2 announcements recently for pre-consortium, which is the step before establishing a full consortium, one for lysosomal storage disease and one for alpha-1 antitrypsin deficiency. Last week, we also announced a new full-blown consortium for rare neurodegenerative disease, which is a partnership with FDA and NIH. The other big rare disease activity is the program that I mentioned, the RDC-ADAP. So we are not a consortium. We don't have the public-private partnership structure. We're a program and a platform that was really designed to be a neutral hub to promote sharing of rare disease data and develop solutions to accelerate understanding of disease progression and optimize the drug development for rare disease. It is a collaboration with NORD and a program entirely funded by the FDA. As a program, we're really making that program evolve into becoming a full-on collaboratory for rare disease with an intent to partner with all the stakeholders in the field. So I have a lot of discussions for collaboration with other rare disease data platforms, a lot of ongoing collaborations that are being developed, and potentially I'm going to try to bring industry to some new work that I intend to develop. We are a place for all types of data to work potentially for all rare disease. If we have already a consortium in a rare disease, I might not be involved directly, but we are going to help and facilitate these efforts within the Institute. The goal is to advance new drug development tools for rare disease, and we tend to have a program to be a program that will be evolving with the ecosystem and making changes as technological progress advances. The RDC-ADAP is also a platform. So usually at the Institute, we used to have internal databases, sometimes open to the public, sometimes not. But here for the rare disease, really, we wanted to have an effort to be wanted the effort to be very participative and really to have a place for aggregation of rare disease data, but also for sharing back some of the data that is shared to us to the entire ecosystem. So we really designed that platform from the ground up with a specific data standard. So I'm not as knowledgeable as my colleagues that talked before me. We're not using CDISC, but another format, but we're fully able to integrate CDISC data into the platform. The contribution of the data to the platform is negotiated through a legal agreement. I'm not going to go into these details, but really we're making sure we're going to enforce a few ground rules. The first thing is that ownership is not changed. So as a registry, if you share data to us, you are still the owner of the data. You can still continue to do whatever you want to do with your data. You're simply trusting us with your data, and we're going to take good care of it. We need the data to be de-identified before collecting the data into the platform. And then you're going to decide as a contributor how you want to open up your data sets to external users of the platform. And you have several selections you can make, from putting the data completely non-confidential to opening them up to anybody and making the data fully non-confidential to external users. But you can also select some other mechanism that involves the review of scientific plans from the users. The platform is really built not to be a repository, but to maximize the use and the utility of the data through that specific format. Technically, we are going to be able to integrate all sources of data into the same data standard that we established. At the moment, mostly patient-level data are in the platform from registries, trials, natural history studies, also data from a digital app that you are probably or surely familiar with. We are implementing capacities in the next 2 years for imaging and large genomic scale data, and we also have ongoing project in EHR. When we get the data, our team is going to integrate the data into the platform and then make them available to the public through the public interface of the platform. So this is the landing page that is that everybody can find. You can create a login. Everything is free and paid for. We're just going to make a verification of your email, and then you're going to get an access to the platform where you'll be able to scroll through the data content. So I'm not going to go through that, but you can really go in and create your login and take a look for yourself. So you'll be able to scroll through the data content, and it's a long curation effort, and we have a lot of data already available, but more and more is going to be available to external users. If you want to access patient-level data, you need to make a request. And as I said, depending on the wishes of the original contributor of the data set that you requested, you'll be able to access the data or not. If you get access, we are going to create a workspace for you where you will get the patient-level data. At the moment, you cannot download the data. Everything is done online in a secured and private workspace where you will have a lot of tools at your disposition to make your scientific work, including the ability to run a virtual machine and our console to make coding and develop your algorithms, but also some built-in analytical tools that you can use directly on the platform. Everything is very intuitive, and I'm putting at your disposition some classic standard visualization or statistical tools, but we're also developing more. And so you need to be urge people that are users of the platform to continue and go back to the platform, because we're developing new tools to facilitate the analytical work, but also help the users of the platform extract more knowledge out of the data. Just briefly going through our database, at the moment, we have about 25 different rare diseases with at least one data set that are in the platform. And as I said, we have more. We have other rare disease data from the Institute that are going to be available through the platform, and more will be visible month after month as the curation work goes. So we don't have a lot of mitochondrial disorders, but we do have the MitoAction data that was shared to us thanks to the effort of Sophia and the connection with Kyra at MitoAction that led to the sharing of this first set of mitochondrial disorder data from the MitoAction digital app. I really want to insist, we want users to develop work on the platform, develop solutions and insights by themselves, but also as a program, we want to do that too. So we intend to leverage the database and develop some solutions for rare disease. So we have a big work group in Friedreich ataxia called the Friedreich ataxia integrated clinical database with currently some models of disease progression that may lead to a clinical trial simulation tool. And there are new work groups that we are working on with a work group in PSP or task force in PSP that should start early next year, and additional projects ongoing. And so really sharing a data set to us, it's great, we welcome it, but also we're here to help. So we need to develop more and more projects, and really, if we want to help, we need to use the data, and that's really the whole point of the effort. And so we've been discussing with Sophia for a long time now. We actually have a memorandum of understanding, and we started a collaboration last year to really start to get things going for mitochondrial disorder overall, and try to aggregate mitochondrial data from various sources into the platform, and also start to think about unmet needs. I know there are plenty, but it's really important to think about the unmet needs, and how using patient-level data could help. As I said, MitoAction shared the data, and this was the fastest sharing of data to the platform. I think within 36 hours, MitoAction shared, signed the DCA, and then a few weeks later we had the data into the platform. So it can be done. Sharing data, everybody talks about it, but really it can be done, and it can go fast. And so we're continuing the work and the discussion with Sophia. We have ongoing conversation with courts, the CHAMPS registry. We really intend to continue to enrich the database in mitochondrial disorders, but also start thinking about the unmet needs, and what is the first line of work we could be doing, whether it's developing some models of disease progression, thinking about developing an endpoint that could be better to track Leigh syndrome patients in clinical trials. There are really a lot of things we can think about. I think my message that I really want to convey to you today is, as a community, I think Cure Mito is very active. There's a lot of things that are already happened. So I think everybody needs to help and to gather as a community to really bring more data into the platform. With more data, we're going to have more and more opportunities. So we're here for that. We're here to help. And I think there's a lot that has been developed through my first conversation with Sophia, but let's keep up the pace and continue. Thank you.
Thank you so much. One of my takeaways from one of my first meetings about your program, Alex, is that we hear so much in the rare community about silos and how we hate them. And this is the way that we're addressing that, right? So it's really exciting to have you as part of our Cure Mito community, as well as just the broader community as well. So thank you.
Thank you.
And keep those questions coming in. Casey says in the chat, data equals treatments. I work a lot with the ALS community, so we read the chat in real time, because a lot of time that is how people with ALS communicate, is with eye gaze technology. So it's hard for me to resist not butting in and reading the chat, because I was trained so well. Well, it is my pleasure to introduce our final speaker for this section or segment of our program, Shannon Rego O'Rourke, the research director of All Stripes, speaking about SURF1 related Leigh syndrome health data insights. Take it away, Shannon.
Thanks so much, Danielle. Okay. Screen sharing working okay?
Yes.
Wonderful. Thank you so much. Well, I'm so grateful for the chance to present to you today. I'm Shannon Rego O'Rourke. I am a research director at All Stripes. I'm also here with my colleague, Erin Smith, who is a director of patient engagement at All Stripes. And she'll be here and available to help with questions later. It's such a delight to get to share some data that we've collected through our partnership with Cure Mito today. So I'm just really grateful for this opportunity. I'm going to start by just telling you a little bit about what All Stripes is for anyone who doesn't know. And then I'm excited to share, again, some data that we've been able to collect through this really exciting partnership. So at All Stripes, our mission is to unlock treatments for people affected by rare diseases. And we have been fortunate to be able to do this by partnering with organizations like Cure Mito to support the development of new treatments with data. So how do we do this? We've developed a two-sided platform, and that platform has a patient-facing side that many folks here may have seen if they're participating in our research. The goal here with the platform is that we are able to consent patients and families to sign up. They sign consent through our central IRB protocol, and we're then able to go and gather patient medical records on their behalf. We're also able to survey patients, and we're able to recontact patients through the terms of our IRB and our protocol as well. Our team goes out, we gather these records at no cost to families, and then we're able to structure that data and make meaning out of it. And that's the part on the flip side of the platform, what we're able to do in terms of structuring data and getting it out into the world to help advance treatments. We have partners, including pharmaceutical companies. We also have partners that we work with, including folks at research institutions. So we're always looking for ways to get more data out there into the world. And that data can be for the purposes of supporting FDA submissions. It can be natural history studies. The goal really is to support in any way we can meaningful research that can help advance treatments. So again, all kinds of data exist in patient medical records, which are one of the sources of data that we collect in addition to surveys and other sources as well. I'm sure lots of folks here have had the chance to dig into their own medical records in the past. So when we are able to gather that information and really structure it, we can learn so much about what the natural history is of a condition, how are patients initially presenting, how are they actually getting to that point of diagnosis? What does that typical diagnostic journey look like? And then once a patient's actually diagnosed, how are they being managed? Is there like a standard of care that we're seeing emerge through records? How does the condition progress over time? These are all some of the questions that we are able to dive into with the benefit of this real world evidence in the form of patient medical records. So how that works is we at Allstripe support many different conditions, all rare conditions on our platform. And we support these conditions in partnership with amazing organizations like Cure Mito, who help us recruit patients to sign up on the platform, and for whom we then are able to gather medical records that the patients are able to access on our platform. Patients consent through the platform as well. We're able to retrieve those records, and we're able to also update those records over time and share that data out ultimately with our partners. So we've been really fortunate to be able to gather records for SURF1 in particular. So we actually started this partnership before Cure Mito was Cure Mito, when it was focused specifically on SURF1. And so we have a cohort that we were able to collect data on with SURF1 related Leigh syndrome. And I'm excited to share a little bit about what we found today. So as part of our commitment to our patients, we always want to share back information we learn from patient records. So we were able to gather some information from these records and structure it. In time, the goal is that we will fairly soon actually be sharing that information with Cure Mito directly to share out to the community. So today is a little bit of a sneak peek at what we've been learning. So who is in this SURF1 cohort? Patients who had diagnostic genetic testing confirming Leigh syndrome caused by SURF1 deficiency in their records. So, in patient records, we're usually able to get those genetic testing results that allow us to confirm that the patient really has a diagnosis. We're focusing here on patients that were located in the United States. And who is excluded? So one of the processes that we go through when we onboard new patients is that we want to make sure we're including folks in our work for whom we're able to collect a sufficient breadth and depth of medical records so that we're not leaving big gaps in the story of their journey. So anyone who didn't pass those steps because we weren't able to get sufficient records wouldn't be included in these in the data here. So here's a demographic summary. This is data on 15 patients in this SERP1 cohort. The breakdown of male to female was 9 females, so 60% of the cohort, 6 males, 40%. The median age was 7. These are all patients located within the US, and you can see the age range broken down on the right. Largest number of patients between ages 6 and 7. We have kind of a big grouping between 4 and 11 years old, and then a handful of patients that are 16 and older. So as I was saying earlier, part of our process is that our team goes out and requests the medical records for these patients after they've signed the HIPAA forms to allow us to do so. So we were able to gather more than 3,700 medical records across these 15 patients. That's a median of 220 records per patient. These folks had a median of 5 healthcare facilities that we were collecting records for, and a median of about 6.5 years of records per patient. Here you can see the breakdown of the types of documents that we're getting. So we're requesting all kinds of things. We're getting some of the data that we're especially interested in is coming from those really rich descriptions in specialist or primary care physician notes, records from hospitalizations like inpatient stays or ER visits, labs, etc. Here, this is just focusing on what types of specialists are we seeing most commonly. Not surprisingly, all the patients that we included in these 15 had seen genetics and neurology. And then you can see going down slightly less common, but still pretty common, cardiology, general practitioner, ophthalmology, physical therapy, and so on. So in terms of age of diagnosis, this is diagnosis confirmed by genetic testing. The age at which that was happening for patients varied. And the most common age range in which folks were diagnosed was between 2 and 3 years of age. But we had some folks who were diagnosed much younger than that, and then folks who were diagnosed 10 years old and beyond. The median age of diagnosis was 2.5 years. And then the years in which those diagnoses were happening were variable as well. We had the most patients in this group were diagnosed between 2018 and 2019. This data was collected up through about mid-2021. So that bar on the far right with 2020 and 2021 probably would have been higher had we had ongoing data collection included here. And then we even had a handful of patients who were diagnosed prior to 2013. One of the things I'm always interested in coming out of data from a new cohort is what the symptoms breakdown looks like. So one of the things we're interested in is how are patients presenting? What are the first symptoms we're seeing? This largely aligned with what we saw in medical literature. So vomiting and feeding problems were a common first indicator, first symptom of Leigh syndrome here. Similarly, growth issues, so failure to thrive or short stature. Slightly less common, but still common for symptoms are hypotonia, sometimes called like being a floppy baby, for example. Lactic acidosis, indicators of cognitive impairment or developmental delays, and then movement disorders or movement abnormalities. You'll see these add up to more than 100, and that's because some of these patients did have coincident symptoms. The median age of symptom onset was 1.5 years, which is interesting because it's about a year behind where the median age of diagnosis is. So, and this graph is showing symptoms that were present in patients' records at any time. So not first symptom, but just was a symptom present in the patient's record, period. Of course, these symptoms have different median ages of onset. So Some of the variability could be related to the fact that not every patient in our cohort may be at an age where we'd expect them to be manifesting certain symptoms. But given that, the most common symptoms we saw, again, not huge surprises here, hypotonia was really common, movement disorders or gait abnormalities, second most common, cognitive impairment, developmental delays and regression, also pretty prevalent. One of the things that we often look at in real world evidence, because it can help serve as a proxy for things like disease progression and other interesting endpoints, are what kinds of assistive devices are folks using. So all 15 of the patients that we included here are using assistive devices. The most common one that we saw in the records were braces. Those were used by 73% of patients. And of those brace-like devices, ankle foot orthoses were the ones that we saw most commonly. You'll also see, so standers were another common one at a little over half the cohort. And then other is the next most common category. That was a catch-all for a whole bunch of things that were kind of a mishmash and included things like communication devices, seating systems, feeding devices, and so on. So one thing I thought was really interesting here is that the median number of assistive devices documented in the record was 7, which is a fairly high number. So that is just, again, a sort of a sneak peek at some of the insights we're hoping to share back with the community quite soon. Some of the things I thought were really interesting here were just the number of facilities that folks are receiving care at. I mean, I think it really kind of gets to what I imagine is a challenge in finding specialists who are knowledgeable about the condition and capable of managing it. There's also, there was a notable gap between that median age of symptom onset, 1.4 years, and the median age of diagnosis at 2.5 years, which was making me think about that diagnostic journey that I'm sure lots of folks are undergoing. Some of the common symptoms that we saw here were generally aligned with what was in the medical record, so we didn't have any huge surprises there. Our median age of onset looked a little later than some of what I saw in the literature, but again, this is also a small cohort, so that's not necessarily a huge surprise. And then just the fact that patients are using lots of assistive devices, which to me has me wondering if there are opportunities out there for research to better understand the benefit that families are deriving there. So our next steps, as I mentioned earlier, we're going to be preparing these insights to share back with Cure Mito that can then share them out to the larger community. There will be some additional information beyond what I've included here. And this data that we've been able to collect to date, we're hoping to be able to contribute to future research should the opportunity arise. And I just want to thank Cure Mito and also the amazing families that have participated and shared their information in order to contribute to this research. We are so grateful for the opportunity to partner with you. And I think there's going to be a question period after this, so I'll hold off on asking for questions and pass it back to Danielle.
Actually, thank you very much. That was really fascinating, especially given what I know from the Cure Mito registry and some of the analysis that we've done. And then also as a mom of a kid with a rare seizure disorder, one thing that jumps out at me, and it's time for Q&A, so I'm just going to ask my question to you. One thing that jumps out at me with your list of early symptoms is that those could also be early symptoms for a lot of things, right? Hypotonia, vomiting, feeding issues. And it really strikes me how challenging it is to get a diagnosis when that is how your child is presenting, right? Because, so I think, well, I'll ask you, is that why the push for genetic testing when you start to develop, developmental delay is so important? I know that I have another child with had a developmental delay, and probably because of my older son, but not necessarily. I noticed a change where the pediatrician said, let's get a genetic test. So I wonder if that's something that you're seeing, and what are your thoughts around that?
Yeah, oh, what a great question. So I don't think I said this off at the top, but I trained as a genetic counselor, so I've seen patients in clinic in that setting a lot too. And a thing that is like kind of striking to me when I look at our data across lots of conditions is how that age of diagnosis shifts over time, because you can see the trend. I'm making an assumption here, but I think that access to genetic testing, broader genetic testing earlier, is like part of the reason for that shift that we see. And certainly I hope that the trend over time for this cohort too is to see that gap in the median age of symptom onset and the median age of diagnosis shrink and shrink and shrink.
Mm-hmm.
And I'm biased as a genetic counselor, but I hope genetic testing plays an important role in that for sure.
Thank you. Yeah, me too. And I always tell people they're concerned about a delay. Of course, I'm the lady they all ask, because of my kids. And I always say, ask for a genetic test. Even if you don't have a family history, ask for a genetic test. And yeah, I think that message is getting out there thanks to groups like Cure Mito. I have a question from Brian. Has All Stripes had a change to engage with the FDA or other health authorities on how this patient shared data is viewed for patient burden journey, external control, a chance to engage with the FDA for patient burden journey, external control, natural history, et cetera?
Yeah, that's a great question. We have been fortunate to be able to meet with the FDA, and we're always on a quest to learn how to best support the endeavors to get treatments, to get them faster. So talking to the FDA is an important way to do that. And then we're working with partners as well who are engaging with the FDA, and so learning lots through them as well.
Great. And Devin Sherman in the audience says, as another genetic counselor, I definitely second that bias and recommendation.
Yay, go genetic counselors.
I'm really glad to hear that, though. I think that's a really important thing, that an easy thing for parents to advocate for. Let me check the Q&A real quick, and then we'll go to a break. Does signing up to Cure Mito registry allow to transfer our data to other databases mentioned today, meaning All Stripes and RDCA DAP, or should we register to all of them? Sophia, maybe that's something you can answer.
Sure. Yeah, no, Cure Mito patient-reported data with courts that's separate from All Stripes. So there are two separate registrations, although we would like to combine the data, but you have to register in both. Regarding RDCA DAP, you don't need to register. It's a platform where we would share a whole data set. Individual patients don't need to register, and we would share the identified information, so your name wouldn't be shared with them. I hope that helps.
Okay. Well, if there are any more questions, we'll do our best to answer them in the chat or in the Q&A. I want to sincerely thank all of our wonderful speakers and our attentive audience today. I know I learned a lot. I always do at Cure Mito meetings and discussions. And thank you. We're taking a break until 3:20 our Eastern time. So 5-minute break.
And we're back from the quick break. Thank you so much for all of you who have stayed with us throughout the day. We really appreciate it. We're on to the next session and very, very important session, caring for the caregiver. I want to introduce Liz Morris, who is our moderator for this session. Liz, take it away.
Hi there. Welcome back from your break. I apologize that there is a phone ringing in the background. It should stop shortly. My name is Liz Morris, and I apologize for the dog. I will be the moderator for the next session, which is focused on caring for the caregiver. This topic is really close to my heart, as my son Colson lived with mitochondrial disease until he was 4 years old. And the stories from families and other caregivers living with mito were really critical lifelines to me as I adjusted to the many phases of our journey, to his diagnosis shortly after his birth, to his rapidly changing needs during his life, and now, how to cope since he passed away in 2020. I now work as a parent and a caregiver collaborator with Courageous Parents Network, and we have been able to feature stories from Cure Mito families, as well as many other families caring for children with serious illness. So today you'll get to hear from 3 speakers who are currently caring for children with mitochondrial diseases. Our international panel of amazing caregivers includes Christina Ferenc from Romania, Jennifer Lindenbach from the United States, and Georgelina Alvarez Baral from Argentina. Following the caregiver remarks, we'll also hear from Rachel Kramer, who is a clinical psychologist, on how caregivers can best care for themselves under the really challenging circumstances that mito presents, as well as how providers can support caregivers and families in that journey. And then we'll open it up to Q&A time, so please do continue to use that Q&A feature, and we'll track questions as people go. So with that, I will apologize again for the dog on the phone and turn it over to Christina. Welcome, Christina.
Hi, hello everyone. Thank you, Liz. I'm Christina from Romania, the Eastern Europe, the region of Transylvania, which you might know as Dracula land. I am the mother of 2 children diagnosed with ECHS1 deficiency. Leigh syndrome. I would like to share my screen now. Okay, so life with ECHS1 deficiency. My firstborn, Anna, she was apparently healthy. But after 3 days, she started having problems with her breathing, and she was misdiagnosed with hypoxia. We were a healthy young couple with no genetic diseases and no medical history. So we did no genetic tests. Unfortunately, Anna spent most of her very short life in hospitals. And
I'm so sorry to interrupt. We cannot see your screen.
Okay.
Okay.
Is it okay now?
Perfect. We can see. Thank you.
Okay. So sorry for that. So unfortunately, our first daughter, Anna, passed away at age 1 after virus, which caused respiratory failure. And she was only diagnosed 7 years later, when we found out that her sister is also carrying the disease. Nora was born in 2015. She was developing normally in the first 3 months. But after that, she just stopped developing, and she started having symptoms which resembled her sister's. This time we detected some high lactate levels and some basal ganglia lesions, which led us to her diagnosis of ECHS1 deficiency at age 1, which was confirmed in the Netherlands by a whole exome sequencing test. Nora is currently age 7. She's on a low protein diet. She's fully disabled, but on a stable condition, and she's a very lovely and happy child. Unfortunately, with this disease, we are facing many challenges every day. These are some medical challenges, like it was really hard to get the right diagnosis. We have to find specialists and expertise in a country where there are no metabolic wards or specialists in mitochondrial diseases. We are having trouble accessing care in other European countries, struggling with finding the right symptomatic treatments, which needs lots of traveling with a medically fragile child. We have to find our own team of doctors, and also having trouble finding the right medication and special formulas. Some other challenges that we are facing are the emotional ones. First of all, it was very difficult to accept the diagnosis that this is a genetic disease which our children inherited from us, and we are carriers without symptoms. This was a shock to us. We didn't know that this could occur to anyone. After that, we had to accept that there's no cure for this disease, and currently there are no treatments either. So we have to find the strength to go on every day. Some other challenges include finding understanding and support from family and friends, dealing with physical and emotional burnout, lots of sleepless nights, finding balance in everyday life. We were lucky enough to find a very supportive rare disease community, which helped us a lot. We are trying to find new purposes and opportunities all the time, and we are hoping to find a treatment or a cure someday. Financial and physical challenges that we have to face are handling a physically disabled child, finding the right medical equipment, mobility and disability aids, providing financially and covering medical bills for our children. This includes analysis tests, special formulas that are not covered by the National Health Insurance Program in most rare diseases in Eastern Europe. You have to find help in caregiving, help with special education, homeschooling, therapists. There's a huge challenge with the lack of sleep, as you all experienced, I believe. We are trying to stay informed and keeping up with new findings, studies, getting funds for research, treatments, and medical care. Spreading awareness is one of our constant activities, and we are hoping that we can make the right decisions all the time. In our case, finding the right diagnosis gave us some sort of closure and the strength to go on and fight for Nora. She taught us that there are no two similar cases, and that early diagnosis and the right symptomatic treatments do make a difference in their condition. Nora's smile is sort of contagious, and this gives us so much hope and the ability to believe that we will have the power to change the course of this terrible disease. We are determined to continue and enjoy every moment and every smile, and spread awareness and hope and inspire for a cure. Thank you so much.
Christina, thank you so much for sharing your family with us and for sharing both Anna and Nora with us. We have had a couple comments in the chat wishing a happy birthday week to Nora. So she's getting love from all around the world. I appreciate how you spoke to the really compounding nature of the different challenges that come with this disease, the logistics of care, the emotional challenges, the impact on the family finances and dynamic. Just really, really powerful. Thank you for sharing that with us. We don't have any questions yet, so I'm going to go ahead and turn it over to Jennifer to share more about her family. Welcome, Jennifer.
Hi, how are you guys? That was a wonderful talk, and Christina really went through a lot of things. So let me just start by kind of introducing myself. I'm Jennifer. I am the mother of 2 beautiful children who have SIRF1 Leigh syndrome, which we've obviously talked about a lot here. Evangeline is 7 and Malachi is 2. Quick story about our diagnosis. Evangeline was diagnosed when she was 18 months. We were caught completely by surprise. We thought it was GERD. It was very much not on our radar. I was pregnant with Malachi while we got that diagnosis. So Malachi was diagnosed actually at 2 weeks old through genetic testing, long before he had any symptoms. What that meant for us was that Evangeline had the sort of the big sister sacrifice in many ways. We made a lot of mistakes with her that we discovered were mistakes that we just didn't know were not what you were supposed to do, that mostly we were able to change with Malachi. Right now, they both have tracheotomies, they both have G-tubes, they both have ventilators at night. They both have a lot of the accoutrement that we've talked about, that they were talking about as far as adaptive equipment and things like that. So I wanted to just talk a little bit, rather than talking too much about the emotional toll. I wanted to talk about advocating for your children in the medical field, and especially if there are any clinicians here, as well as any families that are new to this journey. This is one of those things that can be very difficult to figure out because, as has been discussed so much here, Leigh syndrome is very complex and it's not that well known, especially just in broad, even in medical fields. There are a lot of times as a parent of someone with Leigh syndrome, or especially 2 kids with Leigh syndrome, that I'm in a room full of doctors and none of them have ever seen another patient that has Leigh's outside of my children. So that makes it very interesting because it means that the internet research that you do as a parent has to be figured out through medical journals and much more intensive things, which is good and bad. But advocating for your child, really one of the big things I've found is that whether a doctor is super familiar with Leigh syndrome or not familiar at all, starting with a place of respect, recognizing that doctors are trying to do the best that they can do. They might not know Leigh syndrome, but they are medical experts, but also remembering that doctors are not gods, they are human. And there's a lot of mistakes that can be made if we allow allow ourselves to be pushed aside when we're following our gut on something. So I'd really say for parents to follow your gut as much as possible. If you feel like something is going on with your child, pursue it as much as you can, with respect to the doctors, and be respectful and cautious, but continue asking those questions and continue kind of saying, what do you think about this? What do you think about this? Why do you think that we can we do this? I think it's really important to kind of have that as a standard, but also if something happens, I'm going to stop for a second and tell a quick story. So we actually just came home from the hospital with our daughter Evangeline, where I really had to do this exact thing. She had these transient hypoxic episodes where she was kind of dropping her oxygen saturation. We didn't know why. We thought it was all very respiratory related. And I just kept on thinking that maybe there's something about the GI involved, which is so weird, but that's what I thought. I kept on pushing that, and we found out that she actually had a hiatal hernia that was pushing on her lungs. And once we got that surgery, it was drastic, night and day difference. But really the big part of that is having people on your team that trust you and recognize that that you actually sort of know what you're talking about a little bit. You are the expert in your child. Just making that be a something that you pursue as much as possible. Don't allow the fear of progression to stop you from pursuing possible treatments that can happen. And if you're here, you're a part of the mitochondrial community, and that's a big part of what has helped me walk through this journey. There have been lots of times when we have been able to reach out to various families who have mitochondrial disorders and get their intake, get their idea of what doctors are doing, what doctors have done in different situations. And it's very helpful for you and for your medical professionals to have other people around. I mean, even as we're talking about these cohorts and different things where they're 15, 20 people, it's not a large group of people. And so it's not uncommon for a doctor to not be super familiar with Leigh syndrome. I also wanted to just, with that, I think that it's very easy for us to feel like if there are mistakes that happen, to feel like we're focused on those mistakes, whether they're medical mistakes or whether there's something that we did or something that, there's just so much of that. There's so much guilt and what if-ism in this journey. And I think really trying your best to focus on the future and not focus on the past is really, really important, and just really helps move things forward, because we have to remember that this is a progressive disease, and we'll never know what would have happened if something had been done differently. So I think it's just really important to focus on the future, trying to learn from those mistakes, but not spending all of our time paying attention to that. I think that also when you are in doctor's visits and things like that, something that I've really learned has been super helpful is to ask as many questions as you can of the doctors when you're in that visit. Write down your questions before you go to the doctor. And then if a doctor is explaining something to you, write that down. Sometimes you need to go back and research what they're telling you, because they're talking in medicalese and you might not understand that. So bring I have a big binder that I usually bring with me to appointments that has all of my questions that I have that need to be asked, has space for them to answer it, as well as any pertinent medical information that that those doctors need to know. And it's really important to have something like that, and really beneficial. But I think the last thing just to take away is try as much as you can to find your advocates in the medical field, as well as in the larger mitochondrial community. The people that can help you and support you is just super, super important. That's all I've got. So thank you so much.
Thank you, Jennifer. That was wonderful. I really appreciate you acknowledging what I think is a difficult truth to acknowledge when our children are medically complex, which is that their treatment is really a lot of trial and error.
Yeah.
And the need for trust and collaboration with providers when you're operating in that place of total ambiguity is so critical. So thank you for sharing more about Evangeline and Malachi and how you've done that with them and on behalf of them. We don't have any questions yet, so I will turn it over to Georgelina in Argentina. She will be sharing a pre-recorded talk, and go ahead and take it away, Georgelina.
Thank you. Thank you. Good evening, everybody. I will now share a video I have created, subtitled in English.
Hola, mi nombre es Jorgelina Barral, vivo en Argentina, soy mamá de 4 hijos.
Georgelina, we lost sound, so if you muted your computer, please unmute it.
Thank you. Sorry.
That's okay.
1 minute, please.
Yeah. We heard sound at the very beginning, and I don't know if you then muted your Zoom, but if you leave your Zoom unmuted and play it, I think we should be able to hear it.
Perfect. Okay.
Sorry, bye.
Hola, mi nombre es Jorgelina Barral, vivo en Argentina, soy mamá de 4 hijos, Alejo, Atilio, Amalia y Benito. 2 de ellos fueron diagnosticados con el síndrome de Leigh Syndrome 1. Atilio hoy tiene 7 años y tenía 3 años cuando fue diagnosticado con el síndrome de Leigh. En esa oportunidad nuestra primera sensación fue de aceptación y un poco de resignación. Queríamos disfrutarlo el tiempo que estuviese con nosotros y si bien nos angustiaba la posibilidad de la muerte, también creíamos que podíamos darle una vida hermosa durante el tiempo que estuviese junto a nosotros. Cuando recibimos el diagnóstico del gen SURF1, yo estaba embarazada de 4 meses y venían mellizos en camino. No pudimos ser conscientes de eso hasta que Benito con 7 meses empezó con los síntomas. Fue diagnosticado con la enfermedad a temprana edad y tratado con el cóctel mitocondrial a los 12 meses. Lo que empezamos a sentir a partir de ahí fue desazón, injusticia. No creíamos posibles ser fuertes para soportar 2 diagnósticos. En una visita a nuestra médica del Hospital Ludovica Norma Espécola, le preguntábamos qué podíamos hacer, le pedíamos ayuda y le pregunté específicamente qué hacemos, qué hacíamos. Ella nos respondió algo así: señora, no hay nada, usted si quiere puede ir por todo. Al menos así lo tomamos nosotros. Esa habilitación, ese permiso nos motivó a pensar en la oportunidad de construir lo que teníamos hasta ese momento y construir una oportunidad nueva. Cuando no hay nada, uno puede elegir quedarse con lo que tiene o puede ir por más. Y creo que nosotros elegimos probar suerte con lo segundo. Ahí mismo fue que empezamos a investigar, a leer, a aprender más sobre la enfermedad. Nos frustramos muchas veces, nos enojamos, pero creo que todo eso nos empoderó. Ahí fue que conocimos a la comunidad Surf 1, conocimos a Casey de Cure Surf 1 en ese momento, y creo que todo eso hizo que nosotros hoy estemos acá, luchando. Decidimos este año comenzar un proyecto en Argentina con nuestra familia y amigos, al que le llamamos Fundación Sonríe Surf 1. Hoy vemos esperanza para el futuro de esta enfermedad y estamos decididos a seguir adelante por nuestros hijos. Hoy siento que la familia es el centro de todo y es la que marca verdaderamente la diferencia. La familia es el sostén del paciente, es el sostén de los médicos, de los profesionales que los acompañan, es la que pide, ofrece, insiste, guía. Necesitamos empoderar a estas familias y para esto pedimos que los médicos o profesionales de la salud nos ayuden cuando comunican. Necesitamos que pongan en la rueda a estos padres. Esta rueda ya está girando, necesitamos que los habiliten, que los hagan protagonistas de su propia historia. En la rueda estaremos nosotros para recibirlos y acompañarlos, las familias que ya hemos recorrido este camino. Esto se logrará paso a paso y lo haremos juntos. Pero también sé que los médicos, profesionales de la salud, investigadores, científicos, necesitan que se los apoye, necesitan ese sostén. Necesitamos que los estados de cada país, de cada ciudad, tomen este rol importante y acompañen la tarea con herramientas, inversiones, leyes, lo que sea necesario para que cuando los médicos sientan ese apoyo, también puedan sentirse empoderados. Podríamos llamarle a esta red una cadena de sostén. Como cierre, les comparto este diagrama de Venn. Ahí nos encontramos personas que tenemos roles, pero que primeramente somos seres humanos y necesitamos una red que nos sostenga y contenga. Gracias por esta oportunidad de visibilizar lo que nos sucede. Quiero decirles que somos simplemente familias tratando de cambiar el destino de esta enfermedad.
Wow, thank you so much, Georgelina.
Thank you very much.
I feel incredibly inspired by your vision and what you said about working together step by step and hand in hand.
Thank you very much.
So we've heard a lot from the patient and family perspective, and I welcome questions in the chat or through the Q&A feature. We're going to hear now from Rachel Kramer, who is a licensed clinical psychologist with experience working with families with complex medical needs. Rachel, thank you so much for being here and for sharing your perspectives on how families can take care of themselves, how caregivers can take care of themselves, and how the provider community can support that as well.
Thank you, Liz. And thank you to everybody who just spoke. Christina, Jennifer, and Helena, those were just incredibly powerful and moving presentations that you just made. And I thought it was really deeply meaningful to hear you speak about your families and your experiences. Let me tell you just a tiny bit about myself. I'm a clinical psychologist in private practice in Concord, Massachusetts. I've been in practice for 26 years, and my practice is split about half the time I work directly with children from the ages of 5 until 23, so some who are young adults. And then about half of my time I spend working with parents, doing parent coaching and consultation. And I just want to begin by talking a little bit about, acknowledging just all of the stressors that are entailed in parenting a child with a progressive illness. And I think we've just heard a lot about that, just the challenges of managing medical and developmental care for children, of facing and managing fear and uncertainty, lack of control, and also acknowledging, the stressor involved in having the bandwidth to attend to the physical, social, and emotional needs of other children, if you have other children that you're parenting. And in addition, it's important to acknowledge that over the past 2.5 years, everybody has been parenting in the context of the global pandemic and all of the challenge and worry and uncertainty related to that situation. So, when I think about that, I think, how can I possibly ask you then to spend 15 minutes thinking about parental well-being? And a thought that came to mind is that in psychology, we often talk about the complexity of holding opposing ideas or emotions at the same time. And a useful phrase to describe that is to say 2 things are true. So I think in this instance, we can say 2 things are true. At times, it may feel really unimaginable to focus on yourself as a parent. And we know it's important to find ways to do just that. In order to be a calm and robust leader for your family, parents need to prioritize time to care for their own physical and emotional needs. Psychologists know that children do best when they have the presence of a calm and loving adult in their lives. And it's incredibly difficult to be that person if you haven't attended to your own well-being. When I think about the topic of parental well-being, I think about acknowledging the important basics of physical and mental health. And I've heard a lot of people speak about the challenges of sleep. And so I wrote adequate sleep on here, knowing that we have the context of sometimes that's just not possible, but just thinking about these building blocks when you can, and knowing that the building blocks of good physical health help support emotional health for parents as well. So I often speak with parents about, if there's a small action they can take, every day or most days to support their own well-being, that will support the psychological and emotional health of the whole family. And, this is not an exhaustive list, it's an example list. And I just want to acknowledge that you may be in a place right now where your child's physical needs demand every moment of your attention. So every parent who's listening right now is in a different situation. And what I hope is, if right now you can't focus on these kinds of ways to support your well-being, that you'll be able to at some point come back and think more about your own well-being. In the meantime, I find a helpful frame to be thinking about well-being snacks. So rather than a whole meal, rather than spending a half an hour focusing on well-being, can you take 5 or 10 minutes? So if you're somebody that likes to go hiking, that may not be realistic, but can you take a 5-minute walk? If you're a person who would enjoy reading a book but doesn't have time for that, can you, a couple of times a week, read 1 poem? If you like crossword puzzles, can you just play Wordle a few times a week? So just thinking about small ways that you can take moments to care for yourself, and particularly the idea of self-advocating with friends and family members, and setting some clear boundaries. So texting something like, I have 5 minutes right now. Are you free to catch up? You're really asking people for what you need. And I think sometimes focusing on well-being for parents means thinking flexibly. If what you need to do is to let your child, watch something or play on a screen for a few minutes, so that you can take a couple of minutes to talk to a friend or to do something for yourself, sip a cup of tea. I would encourage you to frame that action as a way of caring for yourself and your well-being, rather than feeling guilty about taking the time to do that. I think another important part of supporting parental well-being is thinking about your own information gathering style and the style of other family members or caregivers who spend time with your child. So as you think about all the complexity of diagnosis, procedures, complications, there are going to be some parents who are information seekers and who are really going to scour the internet, and that's helpful to them to gather as much information as they can. And there are other people for whom compartmentalizing and maybe just focusing on the child who's in front of you in the moment is the most helpful way for that individual to move forward. It's not that one of these styles is right or wrong. It's just that it's useful to be aware of your own style and then to communicate about what works best for you with the other adults in your life. And in particular, if you're parenting with a partner and you have different styles, to talk about that and to balance. So if you have one person who's an information gatherer, who's got a lot of information, and another person who prefers to compartmentalize, to be able to say, hey, before bed, I don't want to hear about the things that you learned in your internet searches today. That's stressful for me. So to try to find the time to talk about that. I think it's useful to think about your grieving style. And in this, I mean grief in the context of, I think actually all of the parents spoke beautifully about this idea of, hopes and dreams a parent may have, let's say when they're pregnant, and then the grief entailed in receiving a really devastating diagnosis. I think about the slide that Christina just presented with emotional challenges, and that speaks to this, I think, really beautifully. So, just to consider that you need time to process, you need time to grieve. If you're parenting with a partner, you'll need to gain some insight into each other's styles of grieving and to communicate about where you are emotionally on a given day of the week. And I think also to feel comfortable self-advocating with the medical team. If there's information that you're presented with that feels overwhelming, to say, I need a moment to think about this, or I want to ask questions, but first I'm just feeling such strong emotion hearing what you've just told me. Everybody perceives and reacts to this type of shocking and upsetting news in a unique way, based on their past experience, their cognitive style, their culture, their environment, their knowledge, and be respectful of your own style and try to develop the ability to self-advocate for what it is that you need. What I've seen in my work with families is that very often processing a child's illness means recalibrating what you're hoping for. So that can be a highly individualized process. You may have hopes, say during a pregnancy, or hopes in a child's first few days or weeks of life. And then after receiving a diagnosis, often parents are hoping just for different sorts of things. And it's useful to consider that individually and then also communicate, if you have a shift in perspective, communicate that to family members or to other people who are part of caring for your child. Something that I referenced earlier in talking about meeting basic physical and emotional needs was the idea of connecting with a community of people. And that might be extended family, and it might be friends who are playing a role in supporting your well-being. And I think, for some people have a large community of family members and or friends who support them. Other people maybe only have a smaller group with whom they're close. But giving some thought to who are the people in your life that provide a supportive and steady presence for you. And I really encourage you to self-advocate with those people, the people that you're close to, about how they can best support you. So, are there one or two people with whom you feel comfortable being really open about your situation? So not only the logistics, we went to the hospital and this happened, but also your hopes and maybe your deepest, darkest fears. Really explaining the illness to a few people, again, whether family or friends, so they know what you're dealing with. And something else that I've seen in some of the families I've worked with children who have very serious illnesses is that parents actually get to this stage where they're functioning what from the outside looks really effectively, they set up systems, they maybe have some outside supports, maybe a family member is coming in to help them. And what I see is that those parents have gotten really good at coping with an extremely difficult situation. But an outsider that doesn't have that awareness of how hard it is to build those systems might say, oh, they have a child who's ill, but they're doing great. So I think being able to be honest with people about how hard it is, how good you are at doing something really, really hard is important. And then self-advocating. Some families like it when a close friend just is physically present with them, can just come over and be in the house. I know that's more challenging. It has been more challenging in the context of COVID, but can come over and just be in the house and just be there as a calm, kind presence. You may feel like sometimes you want a person in your life, a family member or a friend, to talk with you more about what's going on with them, to distract you, to help you think about other things other than what's happening with your child who's ill. And some people may need you to almost to give them permission to say, no, I really do want to hear about your child. That's gonna help me to hear your stories, to hear about what your child's doing, what your family's up to. And then separate from that, I encourage you to accept offers of help and really think about, if people want to drive a sibling to school or offer to cook something for you, to think about how you can allow yourself to let those people in and let them help you. Another aspect of well-being I want to address is just the role that social media plays in parents' lives. Many of you may have read the really disturbing studies that talk about the use of Instagram in teenage girls and how it's linked to depression and anxiety. And we don't really have data about the impact of social media on parents, but anecdotally, we know that social media is a real mixed bag, and it can be fascinating. It can also be really toxic. So I just encourage you to, ask yourself some questions. Think about, let's say you're following people who appear to have the perfect child or a perfect family or the perfect life. Is that helping you, or is that, maybe it helps because it feels like it's a real fantasy, and that it's useful for you to look at that fantasy, and maybe it's really dragging you down and is a habit or a pattern that is not feeding you or supporting you. I've had lots of parents, just with all the different types of disasters that have occurred throughout the world, who get really stuck in doom scrolling, in this idea that they're on their phone and reading about something upsetting that's happened and then clicking on the related article. And it becomes so automatic. I think often we don't even realize that we're doing it, and we don't realize that it's making us feel worse and worse and worse. And again, not serving us. And similarly, I have some parents in my practice who follow parenting, experts, some of whom have real expertise and some of whom don't. But sometimes that the advice that they're listening to can make them parents feel really guilty. So as opposed to supporting them. So I encourage you to really analyze the pros and cons of your own social media diet and consider whether it would be helpful to make some changes in the service of your well-being. And, this strategy, point 3 here, this strategy of setting a daily time limit on some apps on your phone, some parents have found that really useful, like a time limit on Instagram or TikTok. And keep in mind that particularly, depending on the ages of children in your home, thinking about limiting your social media or structuring it is good for your well-being. It's also a really good model for your child. I want to just spend a moment thinking about some kind of cognitive behavioral strategies to coach yourself through stressful moments. I work with families sometimes who will tell me after the fact about a really unbearable moment with the medical team. And I think it can be helpful to have a script in mind, this idea of like, this is stressful, but I'm not alone. And I can tell my partner, my parent, my sibling about it soon. Or if you're having a really challenging moment with your child, I'm a good parent having a tough time and I'm going to do my best. And then giving yourself some scripts for self-soothing. So the idea of, I'm going to take slow, deep breaths and then reevaluate. This is an intense moment. I'm going to close my eyes and count to 20, just slowing yourself down and giving yourself that grace. And finally, When I talk with parents caring for a child with a chronic illness, I find it really helpful to remind parents about the principles of self-compassion. And these are the 3 principles. And in particular, notice that second one, this idea that I just implore you to try to treat yourself with the same tenderness that you would offer to a close friend. And I think that, Jennifer was talking about everybody's going to make mistakes, and she spoke about that so eloquently. And, if you make a mistake or a misstep, and you find the inner voice, like you're beating yourself up about it, how could I have done that? That was careless. I'm the worst parent, whatever that inner voice is saying, think about if it were a friend who were in the same situation, what would you say to that friend? You would say to your friend, oh my gosh, that's so hard. I'm so sorry that happened to you. That's really painful. What can I do to support you? So can we treat ourselves, can parents treat themselves and with that same kindness and compassion that they would offer to a close friend? I'm going to stop my share, and I just want to say thank you to everybody for inviting me to speak with you. It feels like a real privilege to be here.
Thank you so much, Rachel. It was really helpful to see those really tangible practices reinforced. And I love that piece of self-compassion, of treat yourself the way you would encourage a close friend to treat themselves. I know that when my son was alive, I took that a step further to treat myself with the same level of attentiveness and care that I was giving to him, understanding that when you're a caregiver, it is such a symbiotic relationship. And I really just appreciate your words of advice for folks to be as self-aware as possible about their own preferences and needs in all of these domains, because we do get so much information about what it should look like, but we're all unique individuals. So paying attention to our own needs in those domains is really important. The other thing that I would offer is it's a skill set to learn to kind of ask for what we need once we've become aware of it. And it's also a skill set to be clear about what we don't need. And I think that caregivers very broadly are just expected to have this incredibly expansive capacity, and we just don't. And no is a full sentence. No, thank you is also a full sentence. And so I would just encourage all the caregivers on this call to really, really honor your boundaries for your own capacity, because you are doing really, really hard work every single day. And Rachel, I appreciate you being here to speak to that. I'm going to pause really quickly in case anybody would like to pop a question in the Q&A. Anna asked, Rachel, do you recommend any books, blogs, podcasts, etc., which would be directed towards parents of special needs kids, focusing on support, some of the support strategies you talked about?
Yeah, that's such a great question, Anna. And the answer is not off the top of my head. You can shoot me an email, or I can email some resources to Sophia or somebody else at Cure Mito Foundation that they can distribute. So absolutely yes. And somebody's put, Liz, you put a podcast idea in there. So.
Yeah, there's a really excellent podcast called Once Upon a Gene that features stories from rare disease families and researchers and advocates, very specifically focused on children with rare and complex diseases. Any other questions? I know that there was some discussion in the chat about social media in particular, and how people have learned to moderate there. So definitely a thing that's just going to become more and more relevant to these conversations. Okay, well, I don't see any more questions, so I'm going to once again thank all of our panelists for being here today, for sharing their stories and expertise. And I am now going to turn it over to Jessica and Casey to wrap things up. Thanks so much.
Thank you so much, Liz. This has been amazing. I just want to say thank you to all of the parents who have been with us today. It's been a long day, and I know most of you are taking care of your children just like I am. So it's been a constant, put a feeding in, change a diaper, but I've learned so much. And I also want to acknowledge Jennifer and Christina and Jorgelina, their children, Nora, Lena, Malachi, Benito, and Attilo. I love them. I love all of our Leigh syndrome children like they're my own. Since Will was diagnosed, I have come across 134 different SIRF1 families, and let's make it a mission to get more mutations of Leigh syndrome on board so we can figure this out. I hope that we have empowered and inspired you all today here. I know I'm not the closing remark, Jessica has it, but I do want to say a special shout out, or give a special shout out to Sophia for getting all of this organized and getting all the technical things ready. We couldn't have done this without you. You are a blessing beyond. You'll never know. And Miriam is so lucky that you are fighting in her honor. So thank you. My heart is so full, and I'm going to hand it over to Jessica. Thank you all.
Thank you, Casey. So it's my honor to have the closing remarks, and I thought I would just hit a couple highlights and take home points from today's many really great talks from not only scientists and physicians, but also hearing from pharma companies and about clinical trials, and most especially the parents of children that are affected by Leigh syndrome. So as we started our day today, it's been 70 years since the first description of Leigh syndrome, and we've come a really long way. And there are still many challenges and barriers that we face, but we've also made a lot of important discoveries in the science. We have new preclinical models, including mice, the NDUFS4 mouse that really well recapitulates many of the hallmarks of Leigh syndrome. And we have new cell models and induced pluripotent stem cells that can be directed to differentiate into different cell types, including neurons and cardiomyocytes, that are opening up our ability to identify new drug targets and perform drug screening. In addition, we've also had many challenges in doing clinical trials for the new therapies for the treatment of Leigh syndrome, but that's also starting to see movement with pharma companies becoming less risk adverse, as well as several new drugs that are currently in the pipeline going through the phases of clinical trials and showing a lot of promise, particularly some of the antioxidants that were discussed today. And so one of the things that I think is really going to help move us even further is also the fact that the organization like Cure Mito has really identified two really important barriers to the movement of clinical trials. And one of those is creating a registry of patients in order to connect the patients and families with researchers and with clinical trialists to help move along the science and identification and safety and efficacy of new drugs. In addition to that, the registry and the surveys that the Cure Mito Foundation is doing will help us understand the natural history of these diseases, to understand the progression, and to better design clinical trials to better identify those therapies that are going to be most effective in improving the outcomes of patients living with this disease. And I think I want to leave you all today with how we started. Casey started us off highlighting the fact that we all need the community to come together if we're going to identify drug therapies and better improve the lives of these patients. And so I think this really requires us to come together, not only patients and families, but physicians, pharma companies, and scientists. And I think Casey and Sophia really effectively did that with our symposium today. And I would just like to thank them and all of the leadership at Cure Mito for hosting this really great event. And may this be the first of many. Looking forward to seeing you all soon. Have a good rest of your day.